Là, ça marche. Ici aussi. Alors. Ok, ok.
Aloja. Huomenta, huomenta.
Se,
No niin.
mitä täällä tapahtui, oli siis se, että meidän niin kuvayhteys mun tietokoneelta meidän näytölle toimi ja lakkasi sitten toimimasta.
Okei. Hei, mä näen tuossa nappula, jos lukee share screen, niin oikeastaan voisin tehdä sillä lailla, että jos mä laitan täältä PowerPointin nyt, että se on esitysmoodi ja sitten hetkinen nyt, niin tuolla on toi share screen nappula vieläkin.
Jos painat sitä, niin tämän pitäisi näyttää tänne sun näytte.
Joo, niin näyttääkö se nyt?
Ei.
Hetkinen, start sharing. Entäs nyt?
No nyt. Paitsi että nyt siinä tietysti niin päällimmäisenä näkyy Skype, mutta...
Okei, mitäs jos mä vien sen tonne?
Just noin, ja sitten jos mä laitan Skypen fullscreeniin täällä. Joo.
Ja tää on ehkä sit se... Ja PDF mä pistin sulle äsken, se oli helppo. Sitten vaan tuli melkein 40 meganen.
Joo, mutta se on niinku toinen vaihtoehto, mitä voidaan sitten tehdä. Eli täällä näkyy nyt täsmälleen...
Ainoa, mun täytyy tiedätkö, niinku tähän mun läppäriin keskittyä, enkä tähän vieressä olevan screeniin, koska sitten mä niinku ikään kuin puhun sinne yleisölle.
Joo.
Ja näkyy siis screenillä nyt se, kun mä hiirtä liikuttelen?
Joo, näkyy. Et siinä on tietysti pikkasen viivettä, että mitään sellaisia niinku ihan kauhean nopeita. Tää on nyt vähän niinku...
Ja sitten täällä pystytään näkemään tuolla ja tosta, tää on tärkeä.
Joo, sen näkee.
Ok. No mut hei, täällä me saadaan tää toimimaan.
Joo.
Ja se oli 11.15 sitten ja mulla oli nyt sitten 20 minuuttia.
Joo. 20 minuuttia ja sitten sen jälkeen katsotaan, jos yleisöllä on toivottavasti on kysyttävää, niin jutellaan vähän.
Joo, ja oliko se sillä, että kaikki tietää niin kuin fMRistä niin kuin melkein?
Mitä?
Tietääkö kaikki fMRistä kaiken? Siellä.
No ne ei varmasti kaikki tiedä kaikkea, että kyllä ne niin kuin...
Suurin piirtein, joo.
Joo, suurin piirtein tietää, mutta ei välttämättä ole mitenkään kaikki tehnyt fMRI-analyysiä itse.
Joo, mutta hyvä, niin mulla on myös yleisluontoinen, että mikä ikään kuin kristallisaa. Joo, niin, sano.
höpise jotakin. Mä käyn tuolla salin takaosassa kuuntelemassa ääniä.
Okei. Joo, selvä. No niin, yritetään jotain höpistä sitten. Bla 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 bla. Bla 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 bla. No mites Onerva, kuuluuko sinne salin takaosaan mitään? Kuuluuko? Vai onks kaikki huono?
de résolution avec lui là-haut.
Joo.
Eikö kuulu hyvin?
Kuulu hyvin. Sä et tietysti kuule mua sieltä, mutta...
Joo, no ei se mitään. Miten sitten kun tulee kysymyksiä, niin sit sä esität varmaan mulle, kun joku kysyy.
Joo, niin se on fiksuinta tehdä, koska et sä kyllä oikeastaan millään kuule niitä yleisöstä, niin mä voin sitten toistaa niitä sulle.
Joo, ok. Oliko se 5 minuuttia kysymyksiä?
No 5–10. Katsotaan vähän, että miten paljon niitä on ja kauan sulla menee siihen varsinaiseen esitykseen.
Joo. No mut hei, mä yritän olla sillä nopea ja tosiaan niin kivahan se olisi olla siellä Ranskassa, mutta näin se olisi ehkä mennyt vähän liian sitten. Nyt. No, Mariahan on nyt siellä Jyväskylässä. Niin sitten tosiaan, niin kun jollain on jotain yksityiskohtaista teknistä kysymystä tai haluaa niin oikeasti sen datankaan jotain tehdä, niin sitten se Maria, joka on mitannut ja analysoinut, sehän on sitten se niin kuin paras henkilö, mutta että sittenhän se voi niin kuin yhdistää justiinsa.
Joo.
Ok, mutta hei, olisiko tämä, mulla tulee tuommoinen uusi työntekijä 9.30. Onko se sillä, että mä oon sitten tässä Skypen ääressä valmiina.
Joo,
ja
soitan sulle sitten, kun...
Tehdään tämä sitten sama niin kuin hommeli sitten.
Joo.
Niin, hyvä, hieno juttu.
Kiitos.
Ja, alright, yes, moi.
We actually used to have an image connection from there to there, and then we closed it. I don't know why, but I'm pretty sure we'll get it back in some minutes. I hope so.
So I arrived too early.
No, not really too early. Definitely better than too late.
Il n'y a pas de raison que ça marche tout à l'heure, ça marche plus pour l'instant.
I change the cable maybe because we get the teacher computer
I
signal.
noticed. Yeah. Toi me voidaan muuten laittaa kiinni, sitä mä en missään tapauksessa halua. Toi mun oma esitys. Avaappas nyt mulle se. Kiltti pieni.
It's strange because your computer is supposed to do like a black screen or something to say it detects it, but
Yeah. It's weird cause
maybe on the screen configuration.
Let's see the settings. It's weird because it worked already, and then it disappeared for some whatever reason. The devices, displays. It just doesn't detect the other display, which is weird, because it already worked.
It's a refresh rate problem, I think. It should be in 50. Are we? Okay, no, I think I know why. Because here we are on a 50 hertz system,
Okay.
and you've got a 60 hertz system.
Okay.
So it won't work.
But why it worked in the beginning? That's what I don't understand. Let's try it. Apply. Keep changes, please. This would be 40 hertz, but no, it doesn't.
Is that a problem if you use this computer instead for you?
Tasty one talk via Skype.
Okay.
So
Okay.
that's why I would.
Because otherwise we've got another computer with 50 hertz on Windows.
Yes. And then also the sound is still working from my computer.
Yeah.
Okay.
But that's not a problem.
That's really weird. Tää on aina just tällaista.
En principio sí, por eso no. Eso no quiere decir que funciona ahí, ¿no?
Es el único que funciona.
Sí, no, exactamente.
No, porque
Sí, parece funcionar.
we need to tell it to keep the changes.
And there is no more display
No, I think
option?
I think these are all the options we have.
Because you're supposed to be able to tell him if it's double screen configuration or duplicated.
Yeah, but the problem is that this laptop, it doesn't recognize the other screen at all. So I basically can't change the configuration. Let's try to plug it back in. Okay, what we can do, which is not optimal, but it's probably what we need to do, is that we then use another computer, so that we can actually see the picture.
Yeah.
And then what we can do is that I have the slides for the...
Copy it.
Yes.
And put it on the other computer. Yeah.
And then we can use this one for the sound and the other one for the pictures.
Okay.
Possibly. Okay. Let's see if I can get this opened from
I will look for the other computer.
Yeah. Avaan nyt vittu se. Do you happen to have Skype on that one?
I don't know.
Okay.
Because it's the one from the informatics service.
Okay,
and
I
we have
see.
no right to install anything, and it's
Okay. Then
maybe you can try to download it on this one, but
I don't think we really have time to download it on anywhere.
What?
The problem is that for some reason the system first was able to nicely build a connection from my laptop up there to show the image, but then after a while it just disappeared and we can't get it back. And now the only problem is basically that because we have the Skype talk by Iiro.
oh no.
Okay, that sounded bad. So then I basically need to, if I plug this back in, do we? Okay. So now basically what we need to do, I hope that works, is that we need to call him from my laptop.
Okay.
So that we can get the sound. And then we need to have his slides locally in here and show them from the other computer.
I'll let you.
So that's why I asked if he has Skype on that one, because if he had, we would have called Iiro from there.
But will the quality be enough to?
Yes, it was perfectly okay just like 2 minutes ago when I tested it. Now I don't know from where the scratching is coming.
Okay. So it will need decent calculation, I suppose. Or,
Yes,
yeah.
yes, exactly.
There is no Skype because we need an update, but we have no administration.
But that's okay. We can just, or then we possibly could. You have Skype on your laptop, right?
I do.
So we could actually call him from there.
Correct. The amplification of my computer is awful. Sound quality is just
But
abysmal.
It's not a problem with.
because we will plug it into the.
Okay. Yeah, I'll let you.
Can
do
we
it.
try to connect it?
Yes, because it's a Windows one, right? Yes. I have this problem that most of the programs I use for research work better on Linux, but then most of the meeting room technology is really bad on this.
Yeah.
Okay. Could you try the sound as well? Could you? That's really short. Sorry.
Just put it here.
I see.
Officially not, but let's say you spill it all over and you can.
So I have Massimiliano writing here. They've been trying to buy tickets for the metro
They have
for the
been
last
trying.
15 minutes.
Okay, that's.
There's a little bit of queue. I'm waiting for 10 minutes.
Yeah. He's an airplane pilot. Yeah. He probably didn't get, he wasn't authorized.
Well, at least there's a big square for London.
When you say they have been trying, did that mean that they finally managed, or that they are still trying?
He at least the Italian phrasing would not suggest that.
Okay.
He said, "But we will make it."
Okay, I hope so.
They probably didn't understand that they can enter the metro without paying a ticket.
Oh, I didn't either.
It depends on where you are going.
Basically you can. Then
jump over the barrier.
Then one of us, in my last workshop,
Empezamos.
nonetheless was fined, I think, 40, 50 euros, because in fact, they had these multiple tickets that he bought.
So we clearly have a signal from here to there,
Yeah,
but
yeah,
it's
yeah.
not actually updating.
But now we have to look for the sound, or you can use the one from your computer.
Yeah, but it would be better to get it working
Yes,
from here.
because we get the
Yeah.
picture.
Yes, and because then we could actually use the screen sharing and Iiro could operate his slides himself.
Okay.
That would be better.
Mm-hmm.
Let's see. We have the David. Could you please come and have a look? I was trying to open Skype on your laptop.
No,
this,
that's
this
not the
clearly not
way
where
to-
we want to be. From here.
Yes.
Yes, exactly. Let's see if I can find any way of getting an A. I really would. Thanks.
Yeah, there's no A in my.
In your
Try
alphabet
again.
there is no A.
You'll be luckier.
Was now, please. Yes. Here we are. In Finnish we have pretty many A's.
Because we didn't test the sound yet. So.
That's true. David? Do you have some sort of sound file, any sort of music or anything on this computer? So
No.
that
Or
absolutely
YouTube or anything, a video.
we could.
Anything with sound.
Yeah, okay. Well, I suppose we can go on to the internet and
Yes,
and
exactly.
find.
Just to check that.
Because when you use headphone, you just plug it here.
Yeah.
Okay. And then, is there anything special to do, or
There's not much. I don't know.
anything?
Yeah, well,
even
I don't know
football,
if
with-
they're going.
Yeah, anything with sound would do.
Here we go. Tchaikovsky.
Okay.
Not the best.
Grammarly does more than catch errors. With Grammarly, you can find really good, no, perfect words that make your writing sharp or explicit or excellent.
Would you like more? Okay, so let's see.
Okay, that's good. What is not good is that it's not actually updating the picture.
No, I know.
Did you do that on purpose or? Let me finish my sentence in Skype. I'll let you back in.
Tell him you're writing without A's.
And SEs. Skype. Plumps. Okay, I just thought that when it's time for his talk, I'll call him from your Skype. Mä tiedän, sä aiot käyttää näytön ylösalaisin, älä kiltti.
So, let's see if it doesn't stay. So, no? Okay. No. There seems to be a problem. now it's not that it doesn't update, but maybe it's not properly yeah, no, it's connected because there's the image.
Yeah, it's
so
connected, but let me have a look, because it might be some sort of where do we have okay, this
challenging.
this challenging because these windows and in Spanish, but
Cuts a little bit thin, which is not going to be a problem.
because what we would like to have
If it represents
identificar. Okay, that's what I assumed. It would be nice to actually.
no, it shouldn't be a problem.
Yes,
because
it
started with.
Yeah, I'm thinking that because now it's actually using these as two different displays. So this one is display 1, and then we move over there and get to display 2, you see. And it would be better to have them as mirrored displays.
Yeah, well,
There is a little tick you can to mirror, so you only have one.
Yes, exactly, but I'm just trying to figure out which one of these it is. Desactivar configuración, color, recomenda, orientación, varios pantallas, extender, duplicar,
here we go.
conservar cambios y aquí estamos.
Bonjour, ça va? Tu vas bien?
So, is that okay?
Okay,
No,
it's
thank
okay
you.
now.
Let's see. Okay.
Porque lo asimilaron es que hace 10 minutos que se están peleando con la compra de los billetes.
Ese pilota los aviones. Hey, how are you? Very nice to meet you. Thank you for coming. early comers actually. It's the first day of school here, so there's chaos. So some people in mass media are watching that side. He just wrote me that he was queuing for ticket purchase. Yeah, just queuing. Yeah, no. Technically, he's an airplane pilot, I think.
I was asking because I hope most of the airplanes
No.
doesn't work with the same logic as the
No,
ticket sales.
I know. But
Yeah.
the fact that he's my friend doesn't mean that he's as clumsy as I am.
I'm not saying anything about him, I'm just talking about the ticket machines. That's not a problem, that's usual. Just in case somebody didn't notice, there's some coffee available and tea, if you like. If you think it's too early to leave without coffee. There is some coffee and tea. Ole siellä sitten.
You got the link on your email? You saw it? You saw the link on
Yes,
Okay,
yes,
you
I-
shared it? And
yes, I shared
Okay,
it.
perfect.
Älä käännä sitä näyttöä.
È obbligata a cambiare quello. Pantalone da passeggio.
Quello è il pantalone da passeggio.
Questi
sì.
sono quelli
Quelli da combattimento.
Sì, questi vanno un po' la lotta. Dura senza paura. My goodness.
And then if somebody prefers moving while talking, this one is working.
Yeah.
It's
So
just-
okay, so
If
then
somebody-
we'll see with times
Yeah.
and
If somebody wants to use it, we just need to switch it on.
Mhm.
I switched it off just to avoid any artifacts.
Okay, so I think we can now start with due delay, given the amount of Italians here, it's no surprise to anybody. So it's a pleasure to have you, to host you all today to this little workshop. As you know, it's a 2-day workshop. So the first day will be devoted to talks and the second one to discussions among us. And given the amount of audience, I think it might be open to all those who would like to participate. So the main idea of this workshop was to unite people working on a common or somehow related to a common denominator, which is studying brain signals. So brain signals, so the brain is one of, if not the most complex object or system, as you wish, that we can deal with, and it can be approached from many different lines of attack. So people can have different objectives. For instance, one may be interested in finding simply compact representations of what one observes from brain recording. Another objective can be a common one, can be mapping cognition or neurophysiology, or as some of us here, to build some, to try and build some brain physics, or generally speaking, describing, modeling, predicting, controlling, and so on and so forth, the object itself, which is the brain. And so likewise, one can start from completely different vistas. One can think of the brain as an information-related device, so information storage, transfer, and processing device. But equally, from a different perspective, from, let's say, a statistical physics perspective, one can see the system as a spatially extended disordered system, or more generally as a dynamical system. And some of us as you will see, can see the system as a topological dynamical system, for example. Or other perspectives, if you think of the energetic constraints on the brain, for example, you can think of it as a thermodynamic engine. This reflects itself in a variety of different aspects of brain activity that one can hope to analyze or want to analyze: dynamical, thermodynamical, information-related processes, and this can be done at different temporal and spatial scales. And finally, and this we'll see along this day, people can use different methods. Different recording techniques, of course, and different analysis. So this is what the sort of rationale for this workshop is. Now, so along the day you'll see speakers talking about time series aspects of data analysis, network-related aspects of data analysis from different perspectives: theoretical, experimental, and so on. So this for the general picture. Now there are a few practical aspects. The first one is related to this aspect. As you probably, some of you probably noticed, there's some sort of gender imbalance in speakers invited. So this is not to do with a gender imbalance in invitation. We invited roughly as many women scientists. We only had a rejection rate which was imbalanced. So I'm sorry for that, and we'll try to strive a little more next time to to have an acceptance rate which is balanced instead of a proposal rate. Finally, and before giving the stage to people who actually talk about serious things, I would like to thank all these people who, without whom this wouldn't have been possible, particularly Sabine, who bared with my tantrums with providers and so on, and mainly due to my lack of experience with organizations, particularly here. So many thanks to the three of them. So now since we're late, I think we'll proceed and go ahead with the first talk. So maybe you would like to present the first talk and the first. So we'll have a few sessions. The first one is devoted to time series analysis, and so I'll leave. So the first. So I'll let you.
Yes. Let's see. What is for the sound that we don't need? This one we got somewhere.
Boy, it
Yes.
worked right away. That's a pleasure.
Yeah. Do you prefer using this mic or the
Oh,
other?
that's, I think, will be fine.
That one?
Or,
Or
yeah, we can.
well, just switch this one. Yes. Here we are.
So thank you, Vadim, for coming along.
Yes. So, as David said, we will have some sort of chronological order of the day. So basically, we are following the order of normal brain network or any other sort of computational brain analysis. We start from the time series. We talk a bit about how to construct the network. Then what we actually can do with the network, with different network theoretical models, and then we finish the day with some applications. But now we will start from the time series. That is basically where the brain analysis always starts. And our first speaker today is Vadim Nikulin from Max Planck Institute in Leipzig. And he'll tell us about the long-range temporal correlations in neuronal oscillations. Welcome.
Yeah. Thank you, and I'm very grateful to organizers for having a possibility to present some of our research here. So we'll be talking today about long-range temporal correlations in neuronal oscillations. So the content-wise, since we have different backgrounds, I would like to introduce the idea of avalanches and criticality, and then show how long-range temporal correlations can be recorded with EEG and MEG, and show also their functional relevance and clinical significance. So as many of you know, the Ising model, the famous Ising model, where we have spins oriented in different dynamics, in different directions, and up and down in this lattice. So when temperature is low, we have a situation where, just a moment, sorry. Yeah, we have a situation that the interaction between spins are so strong that as time goes by, there will be only one direction, and there will be a strong magnetic momentum. At the same time, you might have a situation when you have a very strong thermal effects. So they kick out spins in different directions, and then there is this kind of random fluctuation, and you don't have a strong magnetic momentum. At the same time, you have an intermediate state, so-called critical state, in between, where the interactions between spins are counterbalanced by the thermal effects. And this is the most interesting state, the complex state, critical state, so to say, which shows the largest number of complexity, amount of complexity, which you can see is manifested in different avalanches. Domains of these spins are oriented in specific directions. So you can see that they have interesting temporal properties, and they have also interesting spatial properties. So, and for exactly that reason, So I would like also to show you the idea of power law distributions, because the Ising model, very many moments in Ising model relate to power law distributions. As you know, many objects and phenomena in the world can be quantified with normal distributions. For instance, the size and weight of apples is nicely captured by Gaussian. The same thing can be said about white noise, which has again Gaussian distribution and a more or less boring white noise spectrum. But many interesting phenomena and objects in the world are actually characterized by power law distribution. So for instance, if you look at the Roman cauliflower and see the probability of a given size of a flower, then you will see that it's actually power law distribution, which could be captured as a straight line in double log. The interesting point is that critical systems also display interesting signals, which are one wave noise or pink noise, which again show very nice distribution like this. So it could be captured as a straight line in this double logarithmic kind of scale. So now, the criticality has been thought to be very important in neuronal networks. I will show you why. So one of the most relevant questions nowadays is whether neuronal networks operate at a critical state. This is a very controversial kind of topic. Many people say yes, many people say no. What I want to show you, I don't want to show you necessarily the ultimate proofs. I want to show you signatures which are consistent with an idea of neuronal networks operating at a critical state. So now, basically, what I want to show you whether we can use at all EEG and MEG non-invasive measurements to detect signatures consistent with critical state, whether they are functionally relevant, and finally, whether they are clinically relevant. Okay, so we can discuss whether it's actually the proof of existence of criticality, but then we can also look whether it's functionally or clinically relevant, as theory tells us. So now, I would like to start with the classical work by Becks and Plants. Probably many of you know this. So the idea here is that instead of spins, we have neurons. So this is a slice of a barrel cortex from red somatosensory system. You have a grid of electrodes. So now how these avalanches are defined. So we don't have here spins. We have rather spiking neurons. So you have neighboring electrodes, and an avalanche is an event which occurs either simultaneously across many electrodes or in closed temporal bins. So for instance, when we have this event, this is an avalanche of this size. When we have many spiking in neighboring electrodes and in neighboring bins, we have a rather large avalanche. So now, following the logic of the Ising model, we should see very interesting distribution in the size of avalanches. So this was a very important work because it showed that neuronal activity can actually propagate, not necessarily in oscillation or kind of other form of unconnected spiking, but in avalanches. And indeed, what they showed that if you measure the avalanche and the size of avalanche distribution, then you see it's a beautiful kind of straight relationship in double log with an exponent of 3/2, which is consistent with Ising model. However, of course, it wasn't enough just to show, as you know, in criticality, that you have only one exponent. You need to show that a family of exponents exists there and they relate to each other in a specific relationship. So what you measure not only size, you measure duration of avalanches, and you actually measure also the average size of avalanche depending on duration. And importantly, they should all relate to each other through this equation. And actually, in some of these slices, using more sophisticated methods with 512 electrodes, they actually confirm this. So I will not go into details about the avalanche collapse and so on, but there is enough literature showing that in vitro, we might have an evidence for critical state being present in neuronal system. So now, another way how you can look at the criticality and to think about this in functional terms, particularly in neuroscience. So let us consider 3 scenarios for neuronal network simulations. In one case, we have a probability of any neuron in this network to excite any other approximately 0.5. As time goes by, the whole network gets saturated. As you can see, it's impossible to code information here because everything is up. On the other side, you can imagine another extreme when the probability is very low for any neuron to excite any other. As time goes by, there is a complete cessation of activity. So none of these 2 states is good for coding information. But in between, where the probability is approximately inversely proportional to the number of neurons, you actually have very interesting properties, such as the largest permutational possibility for the network to code all kind of information. And it's been shown in theoretical and empirical work with slices that at the critical state, we have the largest dynamic range. So for instance, our visual system can see one photon, and it can see the whole. You can actually look at the blazing sun. So we have also the largest information transmission and information capacity. So and if you would think, what are the required properties of networks to be functionally relevant, then these three are of paramount importance. And it actually occurs at exactly these critical states. So now I would like to show you that, okay, you can measure this in slices, but what about us, people who measure EEG and EEG, whether we can approach this also? So now what I will be talking about, not of course single cells, but I will be talking about LFPs. So this is when you record invasively activity from implanted electrodes and also from scalp EEG, where your spatial resolution is a few centimeters. Importantly, I will be talking not about cross-correlations, because whenever you hear the word correlation in the context of neuroscience and EEG, people immediately think about the connectivity measures. No, I will be talking about the outer correlations. So now what is outer correlation? Basically, it just simply shows how the signal is correlated with itself when being shifted by a specific amount of tau. So now we say that we have long-range temporal correlation when the correlation, outer correlation, decays very slowly according to power law. So in my talk, throughout my talk, I will be talking about signals, EEG signals, and specifically I would rather focus on amplitude envelopes of oscillation. So you see that this is a bandpass filtered signal, for instance, in alpha range. I will be mostly analyzing signals which represent an envelope of oscillation. Why it's important? Because it actually shows the recruitment of neurons. The larger is the amplitude of your oscillation, the larger is spatial synchrony of your oscillators. So now, in practice, we can measure, of course, outer correlation using Pearson coefficient of correlation. However, because of the non-stationarity in the signals, we rather prefer to do detrended fluctuation analysis, which captures actually this exponent through Hurst exponent. And for that, we use detrended fluctuation analysis, which basically analyzes the cumulative sum, the random walk of your signal. And as we know from theory, if you measure the variance of random walk as a function, as a window size at which you measure it, then usually we have this nice linear relationship in double log, where when our exponent is 0.5, it actually means we are dealing with white noise, and anything between 0.5 and 1 indicate persistent correlation, which we also call long-range temporal correlations. So now this is our work which we published with Klaus in 2001, where we used MEG to record magnetic fields generated by currents flowing in the brain, where we actually showed an existence of these correlations in amplitude envelope of oscillation extending up to 100 seconds. So basically, if any of you do EEG and MEG measurements, especially resting state, you would be surprised to know what happens now actually has a propensity to affect activity 100 seconds later down the road, which is quite surprising. Of course, a similar thing could be observed through calculating the spectrum of amplitude envelopes. This is, again, beautiful pink noise. And, of course, what I will be throughout referring to often, the detrended fluctuation analysis, seeing this beautiful linear relationship with approximately an exponent of 0.7 in this case. So now, in another theoretical work, it was also shown nicely that if you have network consisting of different excitatory and inhibitory connections, and if you measure the proximity of this network in terms of avalanches to the critical state, captured here with kappa exponent equal 1, then you see that the largest long-range temporal correlations are at exactly critical state. What is also important that it corresponds to a balance between excitation and inhibition. So your strongest exponents are here. So now why it's important? Basically, it means that now we have a little bit of a kind of a non-invasive biomarker for a proximity of measures such as long-range temporal correlation in amplitude oscillations of oscillation to the critical state. So now, since then, there was a rush, of course, to show that criticality is also functionally beneficial for the brain as measured with EEG and MEG. So what people actually wanted to show that the closer you get to a sweet spot of 1 for LRTC, the better is your performance. So I'll just show you two examples of previous work. Here, they used motor tapping. So people were tapping with approximately a frequency of 1 hertz. Of course, we are not perfect. There is always a deviation from you tapping at a perfect 1 hertz rhythm. So there are errors. So interestingly, if you accumulate enough of this, let's say over 10 minutes, you will see that behaviorally, your tapping is also showing these long-range temporal correlations. And so what they actually wanted to show here, that the arrow with which you are tapping relates to LRTC detected before the experiments, and they failed to show it. So what they only were able to show is that if you measure LRTC before the experiments, they will simply relate to LRTC in the behavior. So meaning that if you are wearing in a specific way in the brain, you will wear in a specific way in the behavior. But as such, LRTC were not predictive of the performance. So you see, that's not a prediction yet. So in another study, groups from Palva, they were using now sensory task. So near threshold stimuli, such as, for instance, visual and auditory. You can see here sometimes you detect stimulus, these ticks are up, sometimes you don't. Again, as you can guess, if you analyze behavioral results, they show nice LRTCs. Again, they were not able to show that you can predict with brain LRTC the performance in task. They only showed that variability in the brain relates to variability in the performance. So now what we wanted to show that in complex sensory motor tasks, such as brain-computer interface, we can predict your performance on the basis of LRTC recorded before the task. So for those of you who don't know what BCI is, so this is a possibility to basically control different programs and devices by endogenously modulating your brain activity. So in this specific case, it's a motor imagery. You are just thinking of moving left or right hand, and we can decode using machine learning your states, and then we can move cursor, for instance, in a specific direction. So this is kind of task which require, on the one hand, fast switching between neuronal representation, left and right hemisphere. On the other hand, maintenance of information. So you have to be, on the one hand, kind of flexible in switching, but also in maintenance of your information. Again, the traits which are best kind of existing with critical state. So now, okay, this task can also be done with C electrodes, which I invented. So I was fed up at some point with EEGs. As standard caps, you see these ugly things. So I developed, I wanted to develop an electrode which would be not felt by subjects, invisible to external observer, just they are kind of semi-dry, I call them, because you need only a tiny droplet of gel, and adhesive power is enough to keep it on the head. So you can see here human hair, 1 millimeter, so pretty small. This is a subject with 8 electrodes on the head. Do you see them? So he's wearing it. But I just tell you, you should believe me, there is for here and for here. And now I would like to show you the example of this brain-computer interface in action. So now this guy is wearing 8 electrodes. Maybe you see a little glistening of one of the wires here. So now he will be performing brain-computer interface task, and we wanted to predict performance of it with LRTC recorded before the task, okay? So now what he will be doing, he will be thinking about moving left or right hand. He will endogenously modulate his sensory motor rhythms, and we will decode it and move cursor on the screen. So you will see now how he's doing it. I hope it's possible to see. Do you see arrow and cross moving? No? It's, maybe we can lighten, dim the lights. So.
Yes. Let's see.
So actually he's doing it, okay, extremely well. I just, okay, you see how the cross moves in the direction of arrow? Okay, so now just to show you a little bit of magic behind this machine learning stuff. So what we do here, we use common spatial patterns which discriminate two conditions. Let's say what you see here is the spatial filters obtained with machine learning, which would maximize the difference between two classes, such as, for instance, imagining left hand movement, attenuation over the right hemisphere, and attenuation over the left hemisphere when you're imagining right hand movement. So you see this beautiful attenuation of rhythms. So now these are the signals which are used for BCI. So the interesting thing about BCI is that first you think about moving, not moving hand, but only thinking about it. But then as time goes by with training, you just forget even that you kind of have to imagine hand movement. The same way you don't think when you're performing hand movement, it just occurs naturally. So as I told you, yes, so this is this delicate balance between activation of different neural networks and maintenance of the state we wanted to predict. So now, again, just to show you also another manifestation of LRCC. So this is simply an envelope of sensory motor oscillations over 500 seconds, and you see a classic view of scale invariance. When you zoom in into this and you look at the temporal propagation of amplitudes of oscillation, you see that they qualitatively look very similar at different time scales. Very beautiful. So now, and this is exactly the proof of what I wanted to show you. So now these are 100 people sorted in an ascending order with respect to the accuracy in brain-computer interface. So the best subjects are on the top. The Hurst exponent LRTCs are depicted in color. What you can see is that for different frequencies, for LRTC at different frequencies, the best performers are those who before the task had the strongest LRTC. And this is exactly what we wanted to show, that the strongest, that the best performance in this complex sensory motor task will correspond to properties of networks demonstrating LRTC closest to critical state. So now this is sensory motor task. In another of our study, we wanted to predict, of course, cognitive abilities. So we use a battery of tests such as California verbal learning test, short-term memory for words, working memory, trail-making tests for executive control, measuring different properties of oscillation such as power, individual alpha frequency, and long-range temporal correlations on which I will focus here. So I deliberately will show you first the results of working memory. This is the largest cornerstone for any cognitive operation. It actually shows you the working memory is a short-term memory where you also manipulate objects in the memory. So in this specific task, we use n-back task where you are shown a specific number, and then you have to compare this number with the number which has been presented 2 stimuli before. You know, not the previous one, but you have to remember 2 before. So this task actually requires, on the one hand, switching and update of a memory buffer, but also the maintenance, and you have to counterbalance it. Again, as we wanted to show that this task will be performed best by subjects in whom even resting state activity shows the largest LRTCs. So, and this is exactly what we showed with 90 subjects. It was quite a large group of people. So the so-called switch cost score shows you how accurate you are and how fast you are. It's a composite measure. So the smaller it is, the better you are. So and these are long-range temporal correlations. Again, what you can see in people who have it largest, closer to 1, they have the best performance. When we do inverse modeling, we actually nicely show that the best prediction is obtained for frontoparietal cortex, especially over right hemisphere. These are the areas which lights up like a Christmas tree during the working memory task in fMRI, for instance. But what is important here, we are not recording activity during the task. We record it before the task. And we show that depending on the properties of neuronal networks, they will be more capable of performing this task in an optimal manner. And this is exactly what we show here. And now finally also to show you that that's not only good for functional consequences, but also for clinic. So I'll give you just 2 examples. I start with schizophrenia. As you know, that's a mental disorder characterized by abnormal perception and expression of reality. The hallmark is, of course, hallucinatory activity. So one of the most dominant neurophysiological hypotheses currently is disconnectivity, meaning that not that your connectivity in the brain is diminished, but it's rather very abnormal. So what we thought that it's not only spatial aspects of it which are kind of affected, but of course the temporal ones. And we hypothesized that LRTC should be diminished. I show you why. So here I would like to use this analogy with energy landscapes, where the activity of the neurons is represented as a position of the ball on the energy landscape. So the deeper is basin of this curve, the stronger is your neuronal representation, for instance, of a specific object. It also corresponds to high firing rate. As you can imagine, when you have a high firing rate, you have a better sustainability of information. At the same time, when you have a very sparse spiking, then your energy landscapes are very shallow. It's very easy to kick this ball in different directions. And actually, that's exactly what happens in patients with schizophrenia. They have diminished glutamatergic activity, as a result, diminished spiking. As a result, most likely, their energy landscape is flattening. If it's flattening, it's much easier to be kicked from one state to another. So you don't have this sustained activity over time, which in turn, as we hypothesized, would lead to attenuation of LRTC because they're switching too fast. And this is exactly what we saw. So compared to controls, patients have a massive drop of LRTC for both spontaneous alpha and beta oscillations, but in differing topographies depending on the frequency band. What is important that neither amplitude of beta or alpha oscillations show the differences. And this is again the fact which I emphasize over and over, that it's not the amplitude of oscillations, which is pathological, but the way the system behaves, which is abnormal. And now, finally, I would like to show you the Parkinson's disease. So as you know, this is a motor dysfunction which is characterized by degeneration of neurons in substantia nigra pars compacta. So now, as a result of this lack of dopamine, in motor system, what happens is that you have tremor, you have bradykinesia, slowing of movements, you have postural instability, freezing of gait. And now currently, one of the most dominant ideas about the neurophysiology and neuropathology of Parkinson's disease is the abnormal oscillations in beta range in subthalamic nucleus. So subthalamic nucleus is a structure in basal ganglia, which is the only excitatory structure, and very important one, because it then projects to GPI to thalamus, and the whole information is actually jammed between cortex and cortex loop basal ganglia. So therefore, imagine if this delicate processing usually occurring in normal subjects where you only see strong beta oscillations. So now, either for treatment of this disease, either you give a precursor to dopamine, levodopa, so that it can penetrate blood-brain barrier, or you do surgery. So you implant to subthalamic nucleus electrodes, and 24 hours a day for years with a frequency of approximately 120 hertz, you stimulate subthalamic nucleus activity, supposedly jamming the bad beta oscillations. So what I would like to show you here, patients with and without DBS, and so with this implanted electrodes. As you can see, without stimulation, there is a very slow movements and a different joint, so he cannot do it as fast as he does here. So, and this DBS on, it's 120 hertz bilateral for days, so he's pretty good. And then you can see it's not only distal, but also the axial muscles are good, and you will see that his walking is actually considerably better in on state. So now, however, what we reasoned, it's not the amplitude of beta oscillation which is bad. So what we did, we measured subthalamic nucleus through electrodes, because you not only stimulate, you can actually also record activity. And what you see here in with and without levodopa, LFP is recorded from subthalamic nucleus. So here you see a classic picture. When you give patients levodopa on state, bad beta oscillation drops, okay? For high frequency, it's reverse thing, because high frequency are considered to be prokinetic, good oscillations. So let's look at LRTCs. They are increased in both cases, okay? So now, but let's look at another patient. He shows a completely opposite to a classic view. His strongest bad beta oscillations are in levodopa on. So that completely doesn't fit into this simple idea that it's only oscillations, the amplitude of oscillation, which is important. Let's look at LRTC. So voila, you have this increase of LRTC in both cases. So what it means actually, that's not the amplitude as such, but rather the behavior of the system which discriminated. And with levodopa on, we move system closer to an optimal sweet spot, to a critical state. And also to conclude, because I added these two slides yesterday, because I understand that many talks today will be also based on fMRI and on connectivity. I would like to show you the dark side of LRTC. And the dark side is the following. So I here show you different processes generated with Hurst exponent 0.5, random Brownian noise, and then we have here the pink noise. So if I generate 2 time series independently of each other, but with a given exponent, and then if I calculate correlation between them, but I generated them independently, I will see lots of spurious interaction as my exponent increases. Because effectively, the effective degrees of freedom are diminishing here, because the samples are correlated in time. So now you see this, there is this, like up to 80% of connectivity could be spurious, okay? So now, what it means, because we know behavior has LRTC, brain has LRTC in oscillation. So imagine if I do a task, I present you a stimulus and you have to press a button, and I try to predict how fast you will press a button depending on pre-stimulus alpha activity, okay? So now, reaction times over experiment show LRTC. Amplitude activity shows LRTC. So now what I can do, I can take my EEG and I can predict your reaction times. And this is exactly what we did. So this is exponent from alpha from random subject, and this is the reaction time. And as in simulation, we predict correlations, as you see this right corner, depending on how close like your exponents are increasing. So why it's important for fMRI, you will ask? Well, guess bold signal shows beautiful one-wave signatures, okay? So I just show you one slide with our preliminary work where I take, let's say I have 2000 voxels, resting state fMRI. Here is a product of Hurst exponent from 2 voxels. And what you can see here is that I can predict from univariate properties, because basically I do not calculate here anything, I just multiply together 2 exponents from 2 voxels. I predict the magnitude of correlation between these 2 time series, okay? So basically knowing Hurst exponents from this and this voxel can tell you already that you will have rather high correlation. And this is a little bit scary, basically, because what we do now in this, we reproduce, for instance, default mode network connectivity on the basis of univariate properties. So meaning that we have a solution, we have a remedy for this. It's based on surrogate data, but I guess we leave it either for later today or for tomorrow, but definitely it should be adjusted. So these are colleagues who contributed to different aspects of the study. Thank you. I guess there is already some.
Thanks for this very interesting presentation, Mario. My question is about the statistical assessment from a statistical point of view.
Yeah.
Especially for the EEG. According to your experience, how can I assess the long-term correlation on attacks, and by excluding the effect of filtering currently used in EEG, not only the amplifiers filters, but also knowing that the volume, the school, all the different layers of the, below the scalp that provoke filtering effect. How can you exclude this on the long-term correlation properties?
Okay, maybe I can. Totally very relevant question. Thank you. So here there is one very important aspect to make distinction of. And this is, so for instance, there was a critique by Bedard and Alain Destex about one-wave signatures in broadband data. So if we are talking about broadband data, for instance, from DC, almost like 0.1 hertz to 40 hertz, broadband data, EEG, is indeed affected by filter properties of the tissue in ion diffusion. This is trivial thing. Basically, if in your brain you have white noise, just the brain tissue can filter it to be one-wave. But this is not what we are doing. So first what we are doing, we're extracting narrowband activity. Narrowband. In narrowband activity, the filtering effects of brain are constant. It's actually completely irrelevant. And we have, with British mathematician Duncan Blair's recent paper, where we actually show in analytic form how it's all done. But you can also intuitively understand if you have delta in spectrum, delta function like PAM, so you basically have only one frequency, well, with some neighborhood, and then you study amplitude envelope of this. And of course, this is not affected. Another thing, temporal filtering. You are very right. Let's say if you use FIR filters or infinite impulse response filters, you have to estimate the smoothing produced by convolution, for instance. And of course, we do so in simulations. Let's say you have white noise, you filter, and then you see where it starts bending and going exponentially to, asymptotically to linear relationship. And that's, of course, all of this we take care of course, not being naive. So we estimate all of these effects. So this is very important. But the most important thing which you said, the effects of the tissue and ion diffusion, it's rather relevant to this broadband.
Thank you very much. My question is about the time point that you are using, especially for task ones. How much time do you need pre-stimulus time, and does your results change according to time you are using? And
so basically, the majority of the studies I have been presenting so far are based on resting state activity, and usually it's 10-15 minutes. So the last study which I showed with predicting reaction times on the basis of pre-stimulus activity, we do it, let's say, half a second or 1 second before the stimulus. And yeah, I wasn't able to show many other interesting things we have, but because it's limited time. But like I would say, 500 millisecond, 200 millisecond would be good to estimate post-stimulus effect. Yeah.
Other questions?
I was wondering if you also look into the spatial distribution, spatial structure of the autocorrelation of the decay times of this autocorrelation, because you can see signatures that different brain regions have different type of autocorrelation. Some are going to be much faster and some are going to be much slower.
Totally. So of course we also analyze this. So what I would like to show here, okay, these are distributions of how LRTCs predict performance in working memory task. Of course, we do it both in sensor space and in source space. So we can build distribution as such of them where they are strongly manifested. So alphas would be mostly occipital-parietal, but with the central part also. But then you can ask, okay, which of them are relevant for the upcoming task? And then you can do it through this correlation like this. But of course, like the spatial aspect is super important, as well as for fMRI. This is how I told you that we can predict spatial correlation on the basis of univariate properties of voxels in a given location.
Any other question? Maybe I have one fast question. Did you look at long-range correlations of synchronized episodes?
So can you repeat the first part?
Did you look at long-range correlations not of local activity but of synchronized activity? Let's say.
Okay, but this is, for instance, what some people like Bachirova are doing. So they, for instance, they calculate the phase difference between two brain regions, and then they analyze the time sequences of phase differences. Mm-hmm. Of course, you can do that. And that kind of shows you how stable is your phase interactions between different brain areas. So then, of course, if you know, Podobnik and Stanley published a paper on cross-detrended fluctuation analysis, where you have two time series, and then you see how they are in a cross-detrended manner display or not polar distributions. I've been in touch with Podobnik also. This is an interesting case when physicists also take two sensors from the brain, thinking that they represent a unique and separate information. But of course, we know the strongest offender is volume conduction. And wham, you will have beautiful results just on a trivial fact because you have volume conducted signals. So, like there is this, as I mentioned, at least these two I know people try to do.
Okay, many thanks.
Thank you.
It's doing something.
Yes. Okay.
Do you have a pointer by any chance? I'll use a cursor.
Yes. Now we.
It's all black. There we are.
Here it is. Yes. Do you need sound?
No, for the. No.
Okay. Excellent. So our next speaker will be Emmanuel Stamatakis from the University of Cambridge, and he'll tell us about the large-scale brain networks in consciousness. Welcome.
Thank you very much. Good morning. Thank you for the invitation. So what I will do is try to take you through our journey to consciousness, and I will define what I mean by that in a second, and hopefully I will not make you unconscious in the process. So by means of introducing myself and the lab, here's what we do. We study networks with almost exclusively with fMRI, although we started working a little bit with EEG recently. And what we do is we try to understand how large-scale networks interact during resting conditions, but also during tasks to produce behavior and to produce what we understand as consciousness. Our cohorts are
in healthy controls, of course, but patients who suffer from brain injury. We have people who are conscious as well as unconscious with minor deficits and also with major deficits. And we also work a little bit with patients with stroke. For today, for the purpose of today's meeting and for this talk, I will concentrate on consciousness and unconsciousness. And by that, I mean two things. People that we make unconscious with an anesthetic drug called propofol while we scan them in the MRI scanner, and people who are unconscious or in a coma or minimally conscious following brain injury or brain anoxia, so on and so forth. So here's my definition of consciousness. I'm simplifying consciousness a little bit, trying to stay clear of the philosophical definitions of consciousness. And I do with this slide, but somebody else did, Stephen Lauris from Liège in Belgium, concentrate on the content of consciousness on this axis and the level of consciousness. So we have people in coma who are not wakeful at all, and we have people in vegetative state and minimally conscious state who have cycles of wake and sleep. As we go up this axis, we have, my cursor has disappeared, it doesn't like this slide. Up here, we have fully conscious, wakeful individuals, and down here, as I said earlier, we have vegetative state patients. So these are the comparisons. The red lines reproduce the comparisons that I'm going to try and make today. So I'm going to compare people who had general anesthesia or had a sedation with conscious, wakeful people, and conscious, wakeful people with patients in vegetative state or minimally conscious state. We do this by looking mostly at resting networks, and the discussion on resting networks started around about 95, and this is a bit of work that is relatively unknown, and I'd like to highlight a little bit because Barry Biswell was the first person to talk about resting networks identified by fMRI by comparing the activation of a person doing a motor task with correlations of those areas, of the primary motor area, when this same person was not doing anything, was simply resting. And as you can see, the two patterns on A and B are very, very similar. The field, of course, exploded if you're into resting networks with fMRI. Around about 2005, when the people in Oxford published this paper, which identify a group of networks that we've seen before while people are doing tasks. But the interesting thing was that these networks now are present, correlate, these areas, brain areas correlate with each other when people are resting. When they're scanned for 5, 10, 20 minutes, these correlations seem to appear. And of course, given them names based on what we know these areas do from tasks. So we have somatomotor areas, we have attention areas, we have auditory areas and visual areas. Now, why would the brain do that while we're resting, and what does it all mean? There was an interesting paper by Cole, Bassett et al. In Neuron in 2014 that showed if you take 7 tasks and build this correlation matrix, breaking up the brain into 250 areas, and you take 56, you concatenate together 56 minutes of resting state, you get this extremely high correlation. So the brain architecture that is present while executing a task highly resembles the resting state network. So these networks that we detect at rest reflect what we do or we have done during tasks. Here's our little example that we never got around to publishing because we kind of got spooked by how high the correlation was between the two. So we have a person doing this. It's a finger opposition, thumb index opposition task. And these are the areas that they activate here. And this is the resting equivalent from primary motor area. So you see there are high similarities. The connectivity is stronger while you're doing this task. And the point I'm making is that this resting networks have some kind of relevance, and they highly correlate with the networks that we see when we're doing tasks. Now, one network that, when this whole resting field exploded, one network that we had not seen before, and we didn't know what it does, and there have been various speculations ever since, is what people call the default mode network. And the initial kind of reaction was, because it was more prominent while people were being scanned with no task, was that this is the set of areas that you use to ruminate, to think of your shopping or what you're going to do tomorrow or what you're going to do when you leave the scanning session. Interestingly for us, this is a network that has been shown to disconnect in consciousness. So what are these plots here? The first bar shows people fully awake, healthy controls. And at this other end, we have people in coma, so we have minimally conscious people. So we have a scale of consciousness, if you want, from fully awake to comatose people. And what we see is in various areas, PCC, posterior singular here, all the way to the front, actually, in fact, parahippocampal areas as well. You see this graded linear disconnection as you go from fully conscious awake to people in a coma in all these areas that we call default mode network. So this kind of aroused our curiosity, and we did this little experiment in Cambridge. We started with these pictures that you see on the top row there, and you can see some of these brains are distorted. Some of these people have been in severe, had severe brain trauma. So the experiment that became famous from Cambridge in about 12 years ago now, I think, is where you put people in a scanner, minimally conscious people or visually state people, and you ask them to think for 30 seconds that they are swimming or they're playing tennis or they're doing their favorite sport, and then rest for another 30 seconds and so on and so forth for a while. And what was discovered was that some people were responsive. So supplementary motor area went up in activity while they were thinking that they were playing tennis or they were doing their favorite sport. What we saw here is that the people that have this long-range front-back medial connectivity are the ones that are responsive. So they don't just need to, the idea that we're trying to kind of embrace here is that you don't just need your supplementary motor area to think that you're playing tennis, but you need some kind of higher integrative network to be working to be able to do that. And I'm showing this picture because of all the 4 patients that you see here, the only one that was responsive in this tennis task is the one that had this long-range medial connectivity intact. So we proceeded to try and explain what this default mode network does and what it does, especially during task, while we're doing a task, rather than just looking at resting state. And we have, we've published quite a lot on this, and we show that even when you're doing a simple motor tasks, your default mode network gets engaged and engages with the areas that are involved in the task. And the kind of idea that got more publicity than anything else is that when you're doing a task and you learn the task, you have learned the rules, then your default mode network engages most. Now, the important point here is that it doesn't engage because you have learned the task and you can now do the task and think about your shopping as well, but it engages, the connectivity in the default mode network relates to behavior, to how well you perform in the task. So you need this network to do this task. Another idea that we're working on at the moment, and we haven't, we're just about writing it up, is that there is this hierarchy in the brain from sensory networks to higher level networks such as attentional frontoparietal control networks, the default mode network. We think that the default mode network sits at the top of this hierarchy, and we're not the only people supporting this, but we think that the posterior aspect of the deformer network in the precuneus here is the one that modulates inwards, outwards interactions, thoughts with experimental, with stimuli that we're experiencing on a daily basis. So what the idea that we're trying to push here is that as information comes in, you engage attentional networks and perceptual networks, and the information then is processed at this highest level by the deformer network. The ventral precuneus has access to a lot of memory systems, medial temporal lobe systems. The dorsal precuneus has a lot of structural and functional connectivity with attentional systems. So you have this constant kind of interaction whereby you receive information and you execute tasks, you respond to this information, and you interpret this information by this constant kind of loops that are happening in the brain. Now, the idea that some people in the field of consciousness are trying to push was that you either have your attentional systems engaging or you have your deformer network engaging. We're coming to this, to understand this idea that it's really an interaction of everything, interaction of all systems in the brain that creates consciousness. It's not a binary one or the other situation. It's more like this seesaw action. Now, interestingly, this resting state networks have been almost always studied in this narrow band from about 2005 that the paper appeared in PNAS and said, look, there's this opposing forces in the brain, the deformer network and the dorsal attention network. Either one, when one is on, the other one is off. What we have shown in this paper is that if you extend your range a little bit and kind of don't take any strange frequency range, but the kind of frequency range that you look at when you're analyzing an fMRI task, what you see is that these anticorrelations that we have been talking about for years and years and years reduce a little bit. So if you extend to the high kind of end of this frequency range, you see that the anticorrelations reduce. So it's not a case of either or. Again, interestingly, what we showed is that if you consider this higher frequency, you get a correlation with the reaction time, your response to tasks. And also that this increased positive connectivity that you see between these networks that we consider always anticorrelated is more present during tasks when you consider higher frequencies. To go back to hardcore consciousness, we did this experiment where we gave people propofol. We had 4 levels of sedation: awake, mild sedation, moderate sedation, and recovery. Moderate sedation is where people fell asleep, but you could wake them up with loud shouting if you want. And what did we do? In the first kind of, almost a few years ago, I think this was around about 2010, we did what everybody else was doing. So we looked at correlations with the posterior cingulate, and we found indeed the posterior cingulate being an important part of the deformer network, and we found it did this graded change in connectivity within and across, within the deformer network, but also between other parts of the brain. The connectivity was changed in relationship with the amount of propofol one had. Interestingly, we took blood samples from those people where they were being sedated, and we found that the connectivity correlates with plasma concentration levels, especially with this part of the brain in the brainstem that is part of the arousal system that and I don't have time to talk about today, but that's what we we noticed at the time. Now, this is not a simple picture, as I would like you to believe from this previous picture here. What we did next was use partial least squares and look at the entire brain by splitting it to 90 or 95 areas, if I remember correctly, regions of interest. And for those of you that are not familiar with the partial least square method, you get some latent variables out of the data, and they correspond to a set of weights. Of those, we got 4 latent variables here. One of them was significant and showed this pattern that you see here. So we have awake here, mild, moderate sedation, and recovery. So we have a suggestion that some areas increase in connectivity and then come down back to normal, and we have the decrease in connectivity and then back to the normal stage. And you can see this is a very complicated picture, basically. If we even look just at the default mode network or the interactions between default mode network and attentional frontoparietal networks, you see that within one network you have many increases in connectivity, but also decreases, and you have all these complicated patterns. How do you make sense out of any of that? Well, I don't know, we're still trying. Robin Carhart-Harris in London, who does research in psychedelic drugs, proposed this idea of complexity and entropy and its relevance into looking at brain patterns. So we have this kind of, the hypothesis that we have this point of criticality below which, and at a very low level, we have, this is above it at high levels. We have high entropy, and we have psychedelic states, we have psychosis, and at the very other end, we have low entropy, and we have coma, anesthesia, and sedation. And we try to experiment a little bit with this idea. So our first attempt was to look at connectivity entropy. So how does, with sedation, how does the entropy of connectivity change? So we looked at degree, which is a basic graph theoretical property. So degree is how many areas one area would be connected to. So the area I'm showing you here now, N5, will have a degree of 2 because it's connected to 2 other areas. So we looked at the distribution of this and what we called the fingerprint of the distribution, and we looked at areas that have a rare number of connections or a high number or a frequent number of connections, i.e. If an area is connected to 5 other areas, we looked into whether 5 is a rare or a frequent number of connections a part of the brain has. What we saw is that the entropy that we measured confronts a pattern like this. So we have to an inverted U shape. So we have the brain around about here. We have a random network around about here. And we have a very modular network next to the brain there. And we have a scale-free network there. So the theory then that was, and here's the actual result. So we're looking at the entire brain here, and we have total entropy of connectivity, and what we see is that it goes down as you get increasingly more sedated, and it comes back up when you recover. It doesn't come up to that level there, but we know that 10 minutes after recovery, after propofol has been withdrawn, you're never going to reach that same level there. Your brain is not the same. Our method is able to distinguish awake from moderate sedation, so the extremes, if you want. Interestingly, what we noticed was that if you look at the frequent number of connections, you don't get this effect. It's only the rare connectivity that shows you this effect. So it's only the areas that are connected to a number of other areas that doesn't appear very often that are powerful in showing this effect. We then isolated individual networks and used the network names that I've already discussed, and by removing some nodes, if you want. And what we found was that Our beloved DNM showed that effect. And the limbic network, and these are now network names as discussed by Thomas Yao et al. And also the limbic network is one that displayed this effect strongly. What we saw with the DNM is an additional kind of significant pattern here. So between moderate sedation and recovery. So it had that extra bit of discriminating power. Here what I'm showing is the width of each colorful bands. Each band is a network. So we have the deformer network here. We have frontoparietal network, somatomotor. It's how they change in the number of their connections as we go from awake, mild, moderate sedation, and then recovery. So you have a lot of connections here. They don't seem to change very much. We don't know anything about the nature of these connections. We're just talking about the number of the connections there are. And we see very little change. However, what we see here in the frontoparietal network, as you become increasingly anesthetized, your kind of number of connections changes, it reduces, and it doesn't come up very much when you recover. If you see awake there, it's got that reddish color, and moderate, it's got that yellow color, and we go across to this, and the only, when we compare the two directly, the only significant effect, again, is from the default network. Now, talking about predicting behavior from connectivity, these people were not only scanned doing nothing at rest, but they were also scanned during doing tasks. So we would have 6 minutes of resting state in the very beginning, and then they would do a semantic decision task. So they were hearing words like cow, table, and they had to decide, they have to press a button. Well, they had two buttons, and they decided whether what they were hearing was a living or a non-living object. Now, the reaction time, so we found strong relationships on the number of rare degrees, so the number of connections that were not widely present in the brain, and this is at awake. So their awake state, the number of rare degrees of their awake state predicted how fast they're going to react when they became moderately sedated. And when we kind of removed all other parts of the brain and kept the DMN, that sort of predicted that as well. These are very, very early results. And to go back to this idea of Robbins of the importance of entropy and complexity in the brain, we have the connectivity complexity, so the connectivity entropy, and we have persons who took LSD up here. We have awake people are in their moderate sedation, as I described it earlier, and we have people with diseases of consciousness down there. So back to this diagram, but now, how am I doing for time? Have I got?
Yeah.
Yeah, I've got some time. I can carry on and talk, so somebody must stop me. So now I'm going to talk about interactions between complexity and integration. So we're going to this other theory, great theoreticians, Tononi and Edelman, who also talk about consciousness very famously, and they talk about, well, especially Giulio Tononi, they talk about the importance of integration and segregation in the human brain, and especially for consciousness. And Andrea, who is amongst you in the audience, did this work where he took people from a different data set who were this time they were fully anesthetized, they were not just sedated. And these people you can see in this column here, so these people had anesthesia, and he calculated their ICC. What is ICC? It's intrinsic connectivity contrast. Basically, you calculate the degree I was explaining earlier for each voxel in the fMRI picture. So the brighter voxel is, the more connected that voxel, the more connected it is to the rest of the brain. So Andrea calculated which parts of the brain reduce in ICC as you become fully anesthetized, and which parts increase in ICC. So again, we keep seeing this complex pattern. It's not just one way. Your brain starts disconnected when you're becoming unconscious. And we have people who are fully anesthetized here, and we have patients with diseases of consciousness here. You have the overlap between the two, driving towards a kind of consensus of brain areas that are important for consciousness. So you have the areas, and I'm showing you the default mode network in healthy controls. We have, so I'm still talking about connectivity of each voxel in the brain. We look at the overlap. We see the posterior cingulate, a little bit of the precuneus, and we see this medial prefrontal cortex here that is commonly loses integration when you consider patients with disease of consciousness and anesthesia. We then looked at sample entropy, and we have, again, diseases of consciousness here, and we have anesthetized people here, and that's reduced voxel-wise sample entropy. When we take the overlap of the two, we get parts of the default mode network, and when we then do the ground overlap, we have the posterior cingulate and a little bit of the precuneus and a little bit angular gyri. So parts of the default mode network in people who are either anesthetized or suffer from diseases of consciousness seem to be important for consciousness. What Andrea then did is trying to separate this data from a whole big kind of a continuous scan into states that we called either integrated or segregated. There are some definitions here, how you do that. You identify modules within the data, you calculate participation coefficient, and within degree Z score, basically roughly higher mean participation coefficient means that you have an integrated state. Lower means you are in a segregated state. Interestingly, what we saw here is a kind of partial answer to a question that has been discussed in the context of consciousness for quite a while now. Is consciousness all about cortico-cortical interactions? Is the cortex important for consciousness, or is further down important for consciousness as well? So is the thalamus, and we have a lot of physiological data, but also post-mortem data and all kinds of data that suggest the important role of the thalamus in consciousness. So what Andrea found was that thalamocortical disconnections in states that we called segregated were important for consciousness, but also cortico-cortical interactions, so integration, while these areas seem to be mostly talking to each other, is the cortex seems to be more important, and segregation, the thalamus, seems to be quite important. The other thing that we did with this data is trying to see whether this separation and this idea of dynamic segregation into this dynamic separation into these integrated or segregated states was to calculate properties, graph properties such as small worldness or entropy, again, trying to capture the principles by proxy of integration and segregation. And we saw the importance of, if you look at the entire scan, you get different results than when you look at the states that we call integrated or segregated. I briefly talk about one more study, and then I'll stop because I think I will have run out of time. Yes. Okay, so we looked at the temporal aspects of this data in a slightly different manner. We used hidden Markov models, we concatenated data, and then we used the PCA to find some components. And we defined concepts such as fractional occupancy. By states here, I mean it's more or less the same principle as networks I was discussing earlier. They're not defined in the same way that networks were defined, so they're not exactly identical networks. But so we defined these concepts of fractional occupancy, i.e., the mean probability that the state is active during the duration of the scan, and switching rate, how more active one state is over the other. And we see results in consciousness in the sedation model. In the sedation data that I showed you earlier, we see that there's a state 6 here that seems to be underactive when you become moderately sedated, and a state 4 that seems to be taking over. Interestingly, this top row is activity of those states, and the connectivity is on the bottom row here. You can see, for those of you who've seen the DMN before, the default mode, you see parts of the DMN in the medial frontal areas, connectivity of the precuneus and the PCC here. So we see that switching between 4 and 6. Remember, state 4 is really important when you become unconscious. State 6 is really important when you're fully awake. Predicts, again, the reaction time of the task you're doing when you're asked to do this task in the scanner. Okay, I shall stop there with this last plot here. So the switching rate between the states that I showed you earlier, and we have awake, moderately sedate people, and people with diseases of consciousness. The more ill people in your cohort, the more variability you seem to have in your cohort. It's the same that we see over with brain injury. The more injured people are, the more variability your data has. Anyway, thank you very much for your attention. Here, somewhere here, are all the people that did this work. Thank you.
Hello, thank you. Very engaging. So I very much like this diagram which you showed, the entropy and how psychosis is on the one side, depression is on the other, and like when we use this idea, and the criticality is in the middle. And that's what I've been talking in the first talk.
Yes.
And that's exactly what we see in schizophrenia. It's attenuated. In depression, it's enhanced.
In depression, it's enhanced.
And normal subjects are exactly at this parabolic height.
Interestingly, we see enhanced connectivity in the DMN in depressed samples.
And because of this, my question actually is that whenever I hear talks about fMRI connectivity, people present this connectivity matrices, but do you actually analyze also the signals themselves, whether the spectra are changing, whether they're becoming more flat, whether they show nice one-wave signatures, because nobody is even looking at this. And given what I presented in the last 2 slides, like how connectivity is drastically affected by statistical property, I'm wondering whether you...
We haven't. We always felt the poor relatives to EEG and MEG, and kind of felt if we looked at spectra, people would tell us, "How dare you?" but we did a little bit. We did a little bit when we studied the frequency bands that people use for resting state and whether it's important to extend a bit at the high end. And we did observe a pattern in the direction you're describing, but again, we weren't brave enough to put it in a publication.
Thank you.
So we'll move to
that we detect
Tulee rakkaani. Okeipa. Okay, I don't think this would read.
Siamo nella XX Nabo University. Mi sono frenato in tempo per non dire che
Ehkä mä sit avaan sen PowerPoint. Ihan sama. Mä oon hirveän tyytyväinen aina, kun täytyy puhua PowerPointista, mutta ehkä mäkin sitä.
So I think we are ready for session 2. Moving from time series to analysis to networks. And our first speaker is Onerva, who so co-organizer, who will present herself better than I can do.
Thanks for the very kind introduction, David.
You're welcome.
Yeah. So yes, this is our second session, and this is now about how we actually move from time series into networks. And as said, my name is Onerva Korhonen. I work here under the supervision of David, who doesn't know me. And and the topic of my talk today is the quest for consistency. What's wrong with the nodes of functional brain networks? This will be mostly based on my doctoral thesis that I finished in Aalto University, Finland, last year, actually around 1.5 years ago. So, what's wrong with the nodes of functional brain networks? Typically, I start this by defining what is a network. Now, I assume most of you actually know what is a network. Network is a model of interactions, model of connections, consists of nodes that are connected to each other by links. That's all you need to know at this point. What are then brain networks? If a network is a model of interactions, model of connections, a brain network, of course, is a model of interactions and connections in the brain. Basically, what we know is that all the way up from the level of single neurons, up to the level of brain areas, even up to the level of different subjects, all the cognition is based on interactions. We need to have signaling between neurons, we need to have a collaboration of brain areas, and then we also need to have interaction between people to be able to perform normal cognitive tasks. And today I'll concentrate on functional brain networks. Typically we divide brain networks into 3 groups. So we talk about structural networks that are networks of brain anatomy. Then we talk about functional networks that I will cover today. They are networks of temporal co-activation. So basically when we have a link in functional brain network, that means that, okay, that area got activated in the same time with that other area. Basically this means that we can't tell if there was any causal interaction between our areas. We know that they behaved similarly, but we don't know why. And then the third group of brain networks, that would be effective brain networks. Those are actually networks of causal interactions. I'm not going there. I'm concentrating on the functional ones. As, by the way, most people working on brain networks do. Some of us tell openly that we don't see causations. Others claim that we do. Actually, we don't. All the networks I'll show you today are from functional magnetic resonance imaging data from fMRI. For those who don't have experience on fMRI, this is basically how we obtain a network starting from the fMRI data. So we start from the fMRI bold signal. This is the blood oxygen level dependent signal. So basically what fMRI measures is the level of oxygen in different parts of the brain. And the assumption is that those areas that have more O2 are more active, more busily doing something. So we have the signal. We need some level of pre-processing, removing of all sort of artifacts, movement, whatever, eye blinks, heart, and also technical artifacts from the fMRI scanner. Then we are going to construct a network, so we need a definition of nodes. And in most cases, the nodes of our fMRI network are not the measurement points, but they are some sort of combinations of measurement points. So we talk about coarse graining of our data. We combine the measurement points into some sort of areas, and those areas are used as nodes. Then, when we are armed with definition of nodes and definition of links, we can obtain the network. In fMRI, the definition of links is almost always a Pearson correlation coefficient. It could be some other measure of similarity, but very typically it's Pearson. So basically we obtain the correlation matrix between all nodes, and this is now already a network. It's a fully weighted network. Very often, however, some level of thresholding is applied, so only the strongest, most significant, if one wants to say so, connections are kept to actually obtain the final functional brain network. And today I'll concentrate on the definition of nodes. And in particular, I'm going to ask, should we use the regions of interest as functional brain network nodes, or should we use something else? Basically, the options in fMRI are voxels or then the regions of interest or ROIs. Voxels are the fMRI imaging resolution. They are little cubes of the brain matter, and that's actually where the bold signal comes from. Sometimes people say that the signals of single voxels are noisy, that they need to be somehow filtered or averaged to get rid of the noise. In a normal brain, we have space for around 10,000 voxels, which means that our network, if we use voxels as nodes, is actually somewhat huge, depending on what sort of computational resources you have. If you have a supercomputer, then it's of course small, but if you have a normal old laptop like this one, then 10,000 voxels might be a bit much. So you might want to downscale a bit. The other option are the ROIs. They are collections of voxels, and they are defined either by anatomy, so close by voxels form an ROI, or by function, voxels that activate in the same time or during similar tasks form an ROI, or by connectivity, voxels that have similar connectivity to other voxels form an ROI. Independent of how we define the ROIs, we assume that they are homogeneous, meaning that all the voxels inside the region are doing the same thing. And based on that assumption, we then average the voxel time series to obtain the time series of our region. We assume that the ROI time series present the voxel dynamics, but we actually don't know how violent is this averaging. I'll today walk you through two studies. Besides, I'll annoyingly refer to a third study that I'm not going to cover in detail. And finally, I'll give some pointers to a direction where we could move in the future. So both of these studies are included in my thesis. Both papers are available online, open access, so you can search them up if you want. The first one is titled Consistency of Regions of Interest as Nodes of fMRI Functional Brain Networks. Work from Aalto University with Heini Saarimäki, Enrico Klerean, who should be here today but is not, Mikko Sams and Jari Saramäki, published in one of the first issues of the Network Neuroscience. In this paper, we basically asked two questions. What should be the nodes of fMRI brain networks? Should they be ROIs or voxels? And then we ask, are they functionally homogeneous? We used two sets of fMRI data. Today, I'll only show you results from the in-house subjects. Results for those from the APIDA dataset were very similar. Our time series were 215 points, that's around 6 minutes, and it was resting state data. We used ROIs from 3 atlases, 2 anatomical ones, the Harvard-Oxford, and the automated anatomical labeling. And then we used BrainAtom, which is kind of a top-notch parcellation at the moment. It's connectivity-based. And investigated connectivity both between voxels and then between ROIs. We started by a simple question. Are the voxels inside an ROI actually correlated to each other? So here we have the distribution of voxels belonging to different ROIs, correlations between them shown in green. And then we have in blue the correlation distribution of voxels belonging to the same ROI. We can see that yes, inside ROIs, voxels are a bit more correlated than between ROIs. This is not very convincing actually. There is a huge overlap and a lot of low correlations also inside the ROIs. So we investigated this in a bit more detail. Basically, we defined spatial consistency as a measure of functional homogeneity. It's just the average person correlation coefficient between voxel time series inside a region. It is pretty straightforward to calculate. It's very easy to interpret. Basically, if we have spatial consistency of 1, that's a perfect ROI. If we have consistency close to 0, what is the opposite of perfect? Totally unperfect ROI, a random collection of voxels. And what we see for our 3 commonly used parcellations is that for all of them, the spatial consistency peaks around 0.2, telling that, yeah, in these atlases, these commonly used atlases, ROIs are not functionally homogeneous. This is the basic assumption of all ROI atlases, and it doesn't hold. What does this mean for connectivity then? Here we have connectivity at the level of voxels, so basically the mean correlation between voxels in this ROI and that ROI on the left. And on the right we have connectivity between ROIs, so correlation between ROI time series from here to there. So basically on the left, average of correlations, on the right, correlation of averages. At the voxel level, we see that the strongest links are between ROIs that have pretty high consistency. That is the mean consistency of these 2 ROIs on x-axis. And in particular, we don't see any links between ROIs that have low consistency, which is understandable. At the level of ROIs, however, there are some links between ROIs that have very low consistency. And in my opinion, these links are pretty hard to interpret. If these guys are not correlated and those guys are not correlated, how can we have a strong link between them? What does that mean? Nothing to me. I would say these are spurious. So to conclude, our ROIs are not functionally homogeneous. We see correlations between ROIs that are not functionally homogeneous, and I dare to say that these are spurious. What we don't know is why we have this low homogeneity. Are our ROIs badly defined? Or is it just that voxels are so noisy that when we calculate the average correlation between voxels, it's low, just because of the noise individual in each voxel? Or could it be that our ROIs, for some reason, are idling, and they might actually get activated and have higher consistency in some other time window? That's what we address in the second study, called, these titles are very boring, but I think they are informative, Regions of Interest as Nodes of Dynamic Functional Brain Networks, done basically by the same people as the previous one, plus Elisa Ryyppö and Elvira Prattico, who authored the data, also published in Network Neuroscience. So we asked, if we let our network change in time, how do our ROIs behave? does the spatial consistency change in time? Again, we had 2 sets of data. 13 measured in-house, listening to music freely, and then the 28 subjects from APID. And again, I will show the results for the in-house data. The same 3 parcellations, BrainAtom, Harvard, Oxford, and AAL. I will mostly show results for BrainAtom. This time, we divided our data into time windows because we wanted to create a network changing in time. So we used time windows of 80 samples, that's 160 seconds, and they were allowed to overlap by 1/2. And then, to investigate how the structure of the networks changes, we defined for each ROI a closest neighborhood. That was the 35 other regions to which our ROI had the strongest connections. We used 3 different measures. First of all, the spatial consistency, defined as earlier, the measure of functional homogeneity. We used spatio-temporal consistency, which is the time dependency of spatial consistency. The formula looks ugly, but it's just the mean relative change in spatial consistency between time windows. And finally, we defined network turnover that measures how much our network structure changes, and that is just 1 minus the mean Jaccard index over time windows. So basically it tells how many percent of the neighbors of an ROI change on average between time windows. So what we see is that spatial consistency indeed changes in time. We can see that yes, in some ROIs and some windows it goes down, in other ones it goes up. And this is then reflected by the spatio-temporal consistency. So we can see that there are some areas that keep their consistency pretty stable, and then there are others whose consistency is fluctuating a lot. And same applies to the network structure. Here we have the Jaccard index, so basically which percentage of neighbors of the ROI stayed same across the time windows. And we can see that even for those regions that had the most stable neighborhoods, up to 1/2 of their neighbors changed between time windows. So the network structure is really fluctuating. And that is quantified by the spatial turnover, the network turnover. Here you can see that especially the subcortical areas were really changing their neighbors a lot, which might partially be related to data quality. fMRI has pretty low signal-to-noise ratio for these areas, so it might be that we actually just don't detect their networks properly. But so, we know that network structure changes, spatial consistency changes, but we still don't know why it changes. So we looked what happens inside the ROIs. For this end, we picked 4 example regions. We have 2 with high spatial consistency, the 2 upper rows. We have 2 with low spatial consistency, the lower rows. And these are now voxel-voxel correlation matrices inside the ROI. As you can see, for the high consistency ROIs, the voxel-to-voxel correlation is on average pretty high, meaning that the matrices are pretty yellow. For the low consistency ROIs, correlation is pretty low. They are pretty bluish. And what we see inside the regions, we have this one poor region where the voxel-to-voxel correlation is pretty uniform. It's just low. It could be that this area is badly defined. There is nothing that actually connects these voxels inside this region. But for all the other regions, both for the high consistency regions and the remaining low consistency region, we actually see modules inside the ROI. So, and these are changing in time. They are appearing and disappearing. Here we ask the question, could this be related to network topology? Could it be that the role of the ROI is somehow changing? In one window, it might be a bridge builder. It needs to be able to connect to this direction and also this way. And this why need different internal modules. In other window, on the other hand, it might be more peripheral. No need to build bridges in different directions. And now I'm annoyingly referring to the third paper. Actually, we that is not published yet, but we have actually shown that yes, based on the spatial consistency and spatiotemporal consistency of an area, we can predict what role it plays in the network topology. Is it a hub? Is it a connector? Is it a peripheral node? So yes, these do relate to network topology. To conclude, basically we have shown that spatial consistency changes in time. This may reflect changes in activation of regions. We have shown that these changes are because of changes in the internal connectivity structure of ROIs, and these changes may relate to network topology, and spoiler, they actually do. So the open question is there any way to define a stable set of ROIs that would have optimal functional homogeneity, that would have high spatial consistency all the time in all subjects, all tasks, all time windows? I don't think so. Which, of course, means that I'm saying that all the ways you nowadays use for defining brain network nodes are wrong. Sorry for those who actually do brain networks, but that's what I say. So, what should we do then? This is now just a framework. I'm not going to show any actual results. This is ongoing work with Mikko Kivelä, who is up there, and with Tarmo Nurmi, who is just starting his PhD in Aalto University. And the idea would be that, okay, if we can't have good, stable network nodes, let's go ahead and introduce flexible nodes. This model is based on the theory of multilayer networks. Mikko will tell you more about them in the afternoon, so I'm not going into the details. Basically, what we need to know is that multilayer network is a network that has multiple types of connections combined into a single network. And these multiple types of connections are located on different layers. In our network, for fMRI data, the layers correspond to time windows. They could also be frequency bands in MEG and EEG. In fMRI, they really can't, because fMRI doesn't have a proper frequency spectrum, but they could. Inside each layer, we will define the ROIs on a way that optimizes their consistency. This basically means that we need to cluster the voxels so that voxels with similar time series belong to the same ROI. This is a network clustering problem. If any of you have any background in network science, you know that this is one of the favorite problems of all network scientists. So everybody wants to come up with a clustering method of their own. I want to do that in future as well. I can't present it now, but I promise to present it this time next year, for example. Okay. But so the idea is that we define the ROIs inside each layer so that they may differ between layers. So they are no more fixed, no more stable. Then inside each layer, we define the links as similarity between ROIs, just as we normally do. In fMRI, that would probably be still Pearson correlation coefficient. That's not optimal. So one day in the future, we might actually introduce also a new definition for links, but give me some time. I first need to optimize the nodes, and then I can move on to the links, and in 10 years, I may actually be building networks. Let's see. And then finally, between the layers, we will define the links as spatial overlap between ROIs. So basically, we want the links between layers to tell how much did the ROIs change, because that basically indicates how much changes we had in the connectivity between voxels. So this combining the connectivity between ROIs in the links inside layers to the connectivity between voxels that is reflected by the weights of links between layers. This is something we are working on. I'll let you know when we have some actual results. The preliminary results show that yes, indeed, the ROIs do change between time windows, between layers. And what we would like to see is that are the changes more sudden? If something happens in the stimuli, does that mean that our ROIs change suddenly as well? Or are the changes more gradient, slow, fluctuating? Let's see. So, to conclude, it is not at all trivial to construct a functional brain network. If you want to build one, know your methods, be aware of the possible problems of your methods. The methods used for defining nodes today produce ROIs that are not homogeneous. I'm sorry if you are using Harvard, Oxford, or Brainetom, or AAL, or Craddock parcellations, or any parcellation you can name. Your nodes are not functionally homogeneous. Which means that when you obtain the ROI time series, you lose data. And also, unfortunately, may mean that when you obtain a link, that link may be spurious. Sorry for that. Functional homogeneity changes in time. These changes may be related to changes in function, changes in topology. As a spoiler from the annoying third paper, yes, they are. And finally, because there is a connection between topology, activation, and changes in homogeneity, the low functional homogeneity can't be fixed by introducing any new set of static nodes. It's just not possible, in my opinion. So, to actually accurately model the brain as a network, we need to come up with models with flexible nodes. Thank you for your attention, and if you have any questions, comments, I'm happy to answer.
Thank you. Well, before moving to questions, I would like to do a little recap, actually. Here we've seen, and in this section, in this session, we'll see we're moving from the first main point of the first one, which was connectivity, especially long-range connectivity, both in time, as Vadim showed, and in space, as Emmanuel showed, is important. Here we are adding some sort of structure to this. So there's a lot of connectivity, and this connectivity is not random. So it has some structure which is non-trivial. So this is one main point, which is really terribly important. And then there's an added element, which is when you add structure, what does it mean? And does it mean something? This is Onerva's main point, and so
I'm basically asking if anything means anything.
Exactly.
Okay, thanks for that.
Exactly. So
yeah, but this is actually a rhetorical issue also, because I think sometimes people we don't know what we are talking about. People talk about connectivity or networks or network neuroscience or modules or clusters or whatever, and it might be that when I say connectivity, I mean something completely different than what you mean when you say connectivity. Also, when you say network, you may mean something totally different from what I mean with network. I think the field would really need some sort of standardization of terminology.
Yes. Well, staying within this sort of... sorry for interrupting.
If you ever have a reviewer who tells you that I don't like your terminology, that's probably me. Sorry.
No, there are many people. But speaking of this, so I come to my questions, actually, before I move to the more legitimate ones.
I promise to listen to you now.
So you sort of said at some point that an ROI wasn't functionally homogeneous or something like this.
Yes.
In fact, I think that that's a remark, but also a question, that in that particular instance, you meant dynamically homogeneous, because there was literally no function.
Yes.
So that's one remark. And my main question is, and that's probably a philosophical question, what is an ROI? What do you ask? What do you demand of an ROI?
Okay, before I answer, please take 2 steps to that direction to avoid stepping on my laptop. Thank you. That's a very good question, because ROI is actually not. It has nothing to do with the brain. I have once given a talk with the title "ROIs don't exist," because while we most probably in the brain we have some sort of systems of neurons and systems of brain areas doing the same thing, working on similar tasks, we don't really have ROIs. They don't have any meaning. They are just an analysis tool. Basically, the idea is that because we don't want, for whatever reason, to analyze the network between all our voxels, we are definitely not able to analyze the network between all our neurons, then we need to have some sort of more coarse level of modeling the whole system, and that's what we call ROIs. And of course, we want them to be as good as possible, meaning that we don't want to lose data because of using them, or let's say we want to lose as little data as possible. But actually, they don't have any functional meaning per se. And that's why I also don't like the idea of giving them some sort of like functional names, because ROI is just a collection of voxels. We can't promise that it's actually doing a certain task.
Okay. So that's, yeah. I think that...
I disagree mildly, not completely.
Okay.
I mean, we have to define ROIs in a context, and some of them are based on histological findings, some of them are based on functional findings, some of them are based on functional findings at rest, others at task. So it's a kind of fluid definition, it's not a very solid definition. And with that, I think the wrong assumption is of these networks and these ROIs that they are rigid, that they never change.
Yes,
and perhaps we're
exactly.
assuming the brain is dead or something like that, because that's the only situation that will never change. But, and I come to my question now, which is maybe we should use your homogeneity index to study what the brain is doing, rather than whether an ROI is correctly defined or not. To define what is the state of the brain at that time, rather than to say the ROI is wrong or right.
Yes, yes, these are kind of two sides of the coin. Either one can aim to maximize the homogeneity inside some areas all the time and then build a network, or then one can fix the areas first and then see how the homogeneity changes. And there are actually methodology, for example, the regional homogeneity that is actually used for detecting activated areas by finding the voxels that are most similar to their neighbors in a certain time. Yes, definitely that can be done. But then you don't actually build a network. It's just like more regional, more local thing, so to say.
So thank you for your interesting and philosophical talk. I have not really objections, but I would like to put to your attention some consideration. Mm-hmm. From a statistical point of view, the problem of reduction from voxels to ROIs is not just a problem of memory, but the problem is that using a huge number of nodes with short time series with hundreds of points, you introduce a huge statistical bias just by combinatorial effects.
Yes, that's
so that's
true.
the first problem. So that's why we need the ROIs.
Mm-hmm.
Second, I don't think that ROIs have no meaning, because if you build ROIs based on architectural arguments or anatomical arguments, you are building something related to the structure of the brain. Indeed, I'm against building ROIs by functional methods, because if you require homogeneity, mm-hmm. functional homogeneity inside ROIs, you are destroying functional relationships between different ROIs. So you are introducing other statistical bias. So from my point of view, it is much better to build ROIs on a structural, either architectural or anatomical point of view ROIs, because you need from statistical point of view to mm-hmm. reduce all the statistical bias. And you don't want to introduce uncontrollable functional biases.
Yes, that's an option, but then, and I agree that of course if we have an area defined by anatomy or histology, then it does have a meaning. The only problem then is that if we want to investigate the functional connectivity of that area with other areas, we need to be careful to retain some meaningfulness in the time series of that area, because if we average voxel time series that are not correlated at all, what we get is white noise, and we don't want to study correlations between white noise time series.
Thank you. Very good critical attitude, pun intended. But so what I'm thinking is that if you were to give this talk in another community, meaning in decoding, with decoding people, mm-hmm. they would say, wait a minute, all of our decoding like clusters are working only because the activity is to a certain extent decorrelated. So basically what you are saying is that what they are doing is kind of doesn't make sense, right? But also, actually what it means is that depending on task or condition, there is a very minute but still changes in each specific voxel. But these are two different opposing views. Like decoding people, they are just praying for these little differences in ROIs. You are saying this is kind of, well, not good. Did you understand what I mean?
Not totally.
Like for instance, imagine like what they do with Searchlight. So they have a voxel, let's say 7 by 7 by 7, like a sphere.
Yes.
And the only possibility for them to decode anything at all, if in this very small, it's not even ROI, it's much smaller, there are considerably strong differences between voxels, such that they start discriminating between different conditions or even neighboring time segments. And while what you are saying, it's actually a huge disadvantage.
Yeah, but I don't mean that we should get like rid of the differences between neighboring voxels. I only mean that if we have neighboring voxels that are different, we shouldn't claim that they are similar. Because that's basically what we say if we combine voxels into an ROI and say that the time series of this ROI represents all these voxels, then we are saying that these voxels are to some extent similar. Do we still have time for questions? Do you guys want to have lunch at some point?
Yeah, no, I like very much the provocation. I think it's pretty cool. I think it would help a lot the case to have some sort of killer app. So like find one of the examples where you define, for example, flexible intelligence or whatever that actually breaks down under sort of proper renormalization, proper redefinition, because that actually would point to something that it's out there. Because I completely agree that this is what happens, and I like very much the idea. It's also very true that apart from the statistical issues raised by Andrea, there's also another fact. This is a very good tool for interpretation, however you define it, right? So if the results functionally or the cognitive scores and so on that you can correlate sort of are the same, people are going to tell you exactly like in the coding literature, well, but it works. Yes. So who cares, right? And
that's the problem of the
field. And also having a lot of, for example, I mean, I would love to have dynamical redefinition computation on like forms that compute, but it's a bitch to do mathematically, right? So it's very hard. So you don't want to do that. At the same time, it's very good that you raise this point. So that would definitely help very much in this. And the second point I forgot that I had, but it's gonna come up to me eventually.
Yeah. We'll have. I think we found for those of you who were worried about the discussion and it's not having topics, this is probably one that we'll have.
Yeah.
So.
Yeah, no, that was this is a very nice line of inquiry, and yeah, it's interesting to see this discussion. But yeah, the things that the brain is multi-scale, so every time you are enforcing a certain scale, a certain set of ROIs, you're committing some errors, and you're just having to accept that, well, this is the level you are interested, and there will be some errors there. But it's true. What's the best way of dealing with these, and what's the It's very interesting question and way of thinking, and and then also this is also important so in the cortex is more accepted, but you can also think on this in the subcortical regions where how you define, how you split this in subsets. So this is also a very complex issue.
And then there is always the question of data quality that for the subcortical regions. We typically don't see them with MEG and EEG at all. With fMRI, we see them poorly. So it's very difficult to define them so that they are actually...
I remember the second point. It's
Okay.
about ecology, and this is a problem that is actually in ecology too. The problem they have there very often in ecological networks is that they have species, like food webs and so on.
Yeah.
But the truth is that the animal, so the duck here is not the same as the duck there, right? And so at some point you would like to have this population of ducks be considered as one, and other times the whole population of ducks to be considered as one. So it's related to the issue of scale, but it's not just in scale in terms of how big it is, but how large spatially and how encompassing that is. And so, for example, apart from the points you raised that are very good, it might actually be interesting to think not at how can we optimize at every timestamp or whatever for it to be maximally coherent, but to have something that allows actually to zoom in and out of through scales, both temporal and spatial, and use that, keep the whole information rather than, I guess it's more for tomorrow than anything. But
yes. Also, we don't know what is the correct number of ROIs. We probably know that the 10,000 is too much, and then we are quite sure that 15 is too little, but there is quite a range between.
Okay, so this we're only scratching the surface of the problem. I mean, we didn't even talk about the meaningfulness of connectivity, because we take it for granted. Could be collectivity, for example, as some people say. So, but we'll talk about that later. It's time to thank Onerva for the excellent. So we move to the next one, which is given by Massimiliano Zanin. Massimiliano Zanin works in Spain, it's fair to say, although it's difficult to say where he will be working next year, because he has many options. Today, he's a multifaceted scientist, and today he's going to talk about one of his many research streams based in inference in complex networks. So I'll leave it to you.
Thank you, David, for this introduction, and also thank you, Nerva, for your talk, because it's very nice. You started like destroying complex network or the functional complex network, and I will try to finish the job. So I don't know, probably at the end of the day they are going to kill us, but in any case. So first of all, what is due, I think that I have to acknowledge the help of a lot of people, which is quite bad because somehow the screen is cut in the lower part.
Have you got collaborators?
No, in this case it was not. Yeah, actually there is one that is missing. I'm very sorry. So I hope he's not looking at the streaming right now, because otherwise he will kill me. But I don't know. Let's just. I don't know why can it be. No, we still have the same problem.
Okay. If you try to mirror the displays instead of using that one as a second one.
Okay.
I know that's annoying, but.
Maybe it works better. No, we still have the same problem, because it's the format is like 69. Well, okay, I will try. Let's see what happened. So if there is something missing, I will just try to tell you to remember it. So okay, so as you can see, a lot of people have participated in this. Actually, I've done. Well, I mean, it's not that I've done nothing, but I've done not that much. There are actually a few people that you may know, like David Papo, Onerva. So there are people here, Ilinka, and so forth. And as you can see, we spread across Europe and even to New Zealand. So quite nice. So what is the main idea of this set of works that we have done? Okay, let's consider that we want to create some functional networks on the brain. What we usually do is that we have time series corresponding to, let's suppose, EEG sensor, MEG sensor, or fMRI data. We have some time series, and we usually calculate correlation. Just for the sake of simplicity, let's consider correlation at this time. So there are two approaches to do this in statistics. The first one is what is usually called the frequentist statistics, or the frequentist approach, or whatever. Is the common one. In the common one, we think that there is a true correlation, like for instance, there is a true correlation or causality or whatever between two regions of the brain. We want to calculate to get that specific correlation, and therefore we use what is called, okay, this is the part that is missing. Below is the sample correlation of this, according to the data that we have. There is another approach that is exactly here on the right, that is the Bayesian one. The Bayesian one, what it says is, okay, data are the only things that are fixed. That is, I don't know if there is a real correlation or what is the value of this real correlation between parts of the brain. The only thing that I really know are the data that I can record. And this is the really important thing that I have. So what I can do is that I can calculate a population correlation that is not really representative of the true one, because I don't know, I cannot access the true one, I can just see what the data are telling me. And what is more important is that, as you can see here, or you should, actually I'm thinking, if I... You
can modify the size of monitor.
Exact, or let me see.
Go to... Yeah, you have 111% of zoom.
No, but this is just for the presentation here. So for the so it's just this. Still is not. Okay, it's really ugly. No. So, okay, 1 second.
Time is up.
Thank you. So I think this is really ugly, but at least you can see something. You can see the whole slide, which I think is quite important at this point. So the point is that when we are in the Bayesian framework, we don't just have one value. We have a full distribution of probabilities, because you can calculate what is the most common correlation or the most probable correlation, but actually you have other correlation coefficients that can be possible, just they are not very, let's say, frequent or easily found in the data according to the data that you have. So the important point here is that, now I lost this, so I have to go to, so the important point here is that when we use the standard approach, the frequentist approach, we just have one value. So we say, and this is something that we will have seen a lot of times in functional network, you calculate the correlation between two regions of the brain and you say, the correlation coefficient is 0.3. Yeah, okay, it's 0.3, probably this is the most common value, but the point is that we don't really know, because there is some uncertainty, and what we should do is work with a full probability distribution of this correlation coefficient. Now, why is this important? Well, let's go back to how networks, functional networks, are created and analyzed. So what we usually do here, as you can see, we have several time series corresponding to the activity of the brain at different sensors or voxel or region or whatever. Then we calculate this correlation coefficient, which is just one number, remember, which actually corresponds to the most probable value of the whole distribution of probabilities. And then we use this value in an adjacency matrix. We can binarize it, or we can put a threshold or whatever. Then we calculate some network metric, and then we get some nice results, like that the clustering coefficient is higher in some subjects or things like that. Now, what is the problem? The problem is that, the fact is that here, the calculation of the topological metric is equivalent to applying a very complex and nonlinear function on the adjacency matrix A. So we have an adjacency matrix A, we apply a very complicated function, and the result is the clustering coefficient or whatever else that you want to calculate. Now, this is a problem because there is a very, very well-known fact in statistics that is so very well known that actually I wasn't aware of it, so I mean I have to recognize it, and that this is wrong. Because what we are doing here is we have a distribution of probability distributions, we extract the expected value, that is the expected value of each correlation, which is the correlation coefficient, we use this to fill the adjacency matrix, and then we apply to this a very nonlinear function. This is wrong, and any statistician will tell you that this is the opposite way of what you should do. So first of all, you should apply the nonlinear function to the distribution, and then calculate the expected value. And this is something that in theory is like the first year, the first course of statistics, but once again, I wasn't aware of it. I mean, I wasn't really aware, let's say, of it. So the point is that, a very simple example to explain this. Let's suppose that you have a set of data, like you know that you have something that is, whose value should be 0, but you are not sure, so you have some uncertainty, and then you say, okay, this value, this thing is 0, is a normal distribution centered in 0 with some standard deviation. And then the nonlinear function that you are applying is just the absolute value. Of course, if you apply the absolute value on the expected value of the distribution, which is 0, the result is 0. But what you actually have to do is to apply the absolute value to the whole distribution, and then calculate the expected value. And then the expected value will always be above 0. So they are not the same, and actually the first way of doing it is wrong. So, okay, what does it mean from the point of view of networks? So what we have usually, as I've said, is this, the frequentist approach, in which we have some time series. We just calculate one correlation value, and then we use this correlation value for each pair of channel or time series to create a network. Well, what we should be doing is the following. So for each pair of time series, we calculated the full distribution of correlation, or let's say the distribution of correlation probabilities. Then we sample from them, and we create a lot of networks that are sampled from this distribution. We calculate the topological metrics that we want to study, and then we will have a probability distribution of this topological metric, and then if we want, we can calculate the expected value, the average, whatever you want from this. So this is quite different, as you can see. Last year, we published a paper on this. We said, OK, let's try if this is really important, because maybe the effect is none. So we took several EEG data from this data set that is public. Essentially, these are very short recordings, 1 second, 256 data points, 64 electrodes, and in this case, we had 450 instances. It was, if I'm not wrong, a very simple task, a memory task or something like that. So what we did was, OK, let's apply the standard approach and the Bayesian one, this second one. And how do we measure differences? Well, we just created a very simple bias measure, and the idea is, OK, let's just measure where the frequentist, the value given by the frequentist approach, is located with respect to the Bayesian distribution. So if this value is 0, it means that the frequentist is completely on the left, all the values of the Bayesian are to the right, and therefore that the frequentist approach is subestimating the value that is given by the Bayesian one. 1 is the opposite, 0.5 is like they are essentially the same, so no harm done. Well, these are the results for 6 topological metrics, 6 common topological metrics. You have each one of these, this is a box plot, each one of these box corresponds to a different, we tried with different link densities, with different sampling, with different filtering, so we just considered different bands, alpha, beta, and so forth. And essentially what you can see is something that is quite startling, and is the fact that we never have 0.5. 0.5 will be exactly in the middle of each graph. So here, for instance, you have that this metric, that is the efficiency, is quite underestimated. And here you have that the modularity is overestimated. Also, well, the clustering coefficient is super overestimated. And also the small worldness, quite interesting. Now, why is that? Well, it's very simple to understand. Imagine that you have a structure that is really clear. That is, you measure something, and the frequentist approach tells you that this is like a perfect star or something like that. When you consider the Bayesian one, and you consider that there is uncertainty in each link, you cannot be sure that that will be a star, because just those links are probably the most probable, but there are other combinations of links that are consistent with the data that you have. And therefore, that star might not be as sharp as you think. And actually, all the metrics tend towards random networks. So for instance, if you think about that, the efficiency in random networks is actually higher. That's why the Bayesian says, no, you are measuring something that you think is much less efficient than what it is. Because actually, your network that you think has a given structure is actually more towards randomness. You cannot be sure of that structure. You have to consider that probably is more fuzzy. And so forth with the other metrics. Now, at this point, this was our first work on the topic, and we were a little bit wow, this is bad, right? Because essentially what we are saying is that every paper that has been published using functional network is overestimating the topological metrics that is calculating, and especially the one about small worldness. So as you know, there are a lot of papers out there about how important is small worldness in the brain and how this may change between one disease and another. But actually, the small worldness is probably the metric that is more overestimated here, as you can see according to this approach. So. What we were thinking is this really a problem? That is really uncertainty just a problem that we have to solve somehow, or is uncertainty something that we can use for our benefit? Well, the idea is quite simple, and actually the answer is yes to the second. So let's consider that we have a very simple network like this one, in which we have 3 nodes, and let's suppose that we want to calculate, for instance, the maximum spanning tree or something like that. So you know that we will just keep 2 links out of this network. And here, as you can see, we have that each one of this link is defined by a probability distribution for the correlation, which is 0.5 here, here is 0.5 minus delta, and here is 0.5 plus delta. So essentially, these two distributions can be collapsed to the center to 0.5, or they can just get farther away. So the point is that if you use a frequentist approach, you will always have the same result. It doesn't depend on the value of delta. That is, if delta is not 0 but very small, or even if delta is very big, you will always have that this link is the most important one, this one is the second, and this one is the third. So the maximum spanning tree will always be this structure here. Now, what happens if we add the Bayesian part? Well, in the Bayesian part, it's quite different because when delta is close to 0, these three links have essentially the same probability distribution. So every structure can appear out of here. On the other hand, when delta is quite high, is quite large, what we have is that the three distributions are completely separated, and therefore, even the Bayesian approach will say, okay, this is the structure, the most probable structure is the one including the two original links. So what we have here is that somehow frequentist approach, or the standard approach for analyzing, for creating functional network, will tell you the most probable structure. But the Bayesian one will also tell you how probable is that probable structure. So it will tell you, we can encode somehow something about the uncertainty in the data that we have. So it's a different point of view to the data. Now, we said, okay, let's try this. Let's try if it's really important. So we took a similar data set. These are EEG data from an object recognition task. We have alcoholic subject and matched control subject. We have a lot of trials, and this is very important because we have 4,000 plus 4,000 trials, and the same 64 channels, 1 second recordings, and 256 data points. So second, what we do, we calculate the networks, and as I told you before, just for the sake of simplicity, we have considered linear correlation. And in principle, I will show just the results for broadband signals, but as I know that there are some people here that like to see the things in alpha, beta, and so forth, we also had the same results for alpha and beta, and they are essentially the same. So it doesn't depend actually on the frequency that we are analyzing. Once we have this network, we calculate the maximum spanning tree, that I guess more or less you know what it is. And then we calculate 4 topological metrics on these spanning trees: leaf proportion, diameter, degree divergence, and efficiency. So why do we use, in this case, maximum spanning tree? Well, first of all, just for doing something different. And second, because this simplifies a lot the analysis. Because the idea here is that we just have a 4 metrics. So when you analyze a functional network, you can calculate, I don't know, like 100 of different topological metrics, and which one do you want to choose? That's quite a complicated problem. The advantage of maximum spanning tree is that beyond these 4 metrics, I think that there are none that are really relevant or that are not really correlated with this 4-1. So with 4 numbers, you can describe quite well this network. And then what we did was, okay, let's, well, first of all, let's see the differences. So you have the 4 topological metrics here. In green, you have the frequentist approach, and in red, this probability distribution given by the Bayesian one. So as you can see, as in the previous case, these vertical bars are the values expected in a random case. So essentially, we are moving towards the random case, which is exactly what we expected, what we have seen in the previous result. So up to here, no surprise at all. The surprise is when we say, okay, let's try to classify this. That is, I have 6,000 trials that are from control people and 6,000 trials that are from people with alcoholism. So if I use these 4 metrics that I have here, these 4 topological metrics, and a random forest, for instance, classification algorithm, which is a very standard one in machine learning and very well known and quite effective and so forth, am I able to discriminate between the 2 groups? Well, we did that. And this is the result when we just consider the frequentist approach. That is, these are just using the 4 topological metrics that we get according to the frequentist approach. The classification score is around 60. It's not amazing, but on the other hand, you have to take into account that this is a very complicated problem because we have just 4 metrics and not a lot of features, clearly, just 4 of them. And also the problem is quite ill-defined because identifying the difference between a control subject and someone with alcoholism just in 1 second is quite difficult. So, I mean, 60% is not actually that bad. Then we calculate the same with the Bayesian data. That is, we calculate all the features according to the Bayesian approach. We put these values inside the same classification model, and we get a slightly better, but essentially is the same classification score. Now, this is interesting because what it's telling us is that the quantity of information that is useful to discriminate between the two groups in the frequentist approach and in the Bayesian one are essentially the same. So when we include uncertainty, because the Bayesian approach is essentially including uncertainty in the network reconstruction, we are not losing information about the differences between the two groups, which is quite nice. But then we said, okay, let's merge both of them. And if we merge them, we get a nice jump in the classification score. So essentially what it means is that, what I said before, the frequentist approach tells you what is the most common or the most probable structure in the network. And you may have a pathology in which there are differences in the structure between patients and control subject. But then there is the Bayesian approach that tells you, okay, not just this structure is the most probable, but I can tell you how probable is this structure, specifically this structure. So how much uncertainty I have on this. And it seems that this uncertainty is also important to discriminate between the two groups. So it's like maybe some pathologies are not just defined by a difference in the way the brain is connected, but also with the sharpness of this connection. So how these structures are really well defined or when they are more uncertain because maybe they are evolving very fastly and then we cannot fix the image there. So there are quite some differences. And as a last point, now we are working on the following question. So what about fMRI? So we have seen first with Vadim, then with Nerva, that probably fMRI is not, it's not that simple to analyze. And what about the problem specifically here that we are analyzing, that is the uncertainty? Well, it's essentially the same, just way worse. Why? Well, first of all, because of the time series length. So the uncertainty that we have, for instance, in the linear correlation between the two time series is inversely proportional to the time series length. If you have a very long time series, you will have a very small uncertainty. And usually, I would say that an fMRI is like 2 orders of magnitude slower than an EEG or an MEG. So shorter time series, higher uncertainty, bad. And second, we have the problem of the spatial resolution. So usually you have around, 32, 64, well, let's say 100 sensors in EEG. In an fMRI, you have tens of thousands of voxels. So this is kind of a problem. So we have higher uncertainty. So what we did was compare, let's first start by how you calculate functional network in fMRI in a standard approach. In a standard approach, what you will do, you will start from some time series per voxel. So you have a lot of time series. As we have seen with Nerva, the standard approach is to average this time series inside rows. So you have now one time series for each row, which is just the average of the signal that you have underneath. Then you calculate this point-wise correlation, which we have seen is wrong because doesn't take into account the uncertainty. And then you calculate some topological metrics. If you want some very nice mathematical formulation, this will be somehow the process. Now, there are two problems here. The first one is that, okay, we calculate the point-wise correlation, that is just the R, just the value, one number, 0.5, instead of the whole probability distributions, as we have seen before. But there is another problem here, is that we average time series. This is not the actual correct way, Bayesian way of doing things. And so don't ask exactly me why, but Javier Cano is an expert in Bayesian statistics, and he was working on the topic and said, no, the actual way of doing this in this situation is the following. You start, of course, with time series 1 per voxel. Then, for each pair of voxels, you calculate the full probability distribution of the linear correlation. So you calculate the distribution of rho. Then you average the probability distributions, this probability distribution, for each pair of regions between the two rows for which you want to calculate the correlation. And then you sample the network as we did before, and then you calculate your topological metrics. Now, this is very nice. There is just a very small problem, and this is the small problem that, so first of all, you have to calculate rho instead of R. That is, you have to calculate the full probability distribution. And if you have ever worked with Bayesian statistics, you know how time-consuming and it's quite complicated to do anything. And second, that instead of working with networks of 100 nodes that are very nice, here you have to work with all boxes that you have, so all pairs of boxes. So essentially the point is that the computational cost is like infinite. But more or less we have managed to keep it under control by using all the possible tricks that we could think in the programming phase. And these are the results that are just preliminary, so we are still working on that. But essentially the idea is that in blue you have the evolution of these 8 metrics according to the Bayesian approach. And in gray, you have the standard, the evolution of these metrics according to the standard approach, that is the frequentist one, as a function of the threshold that we applied to create the network. So essentially, on the left, you have fully connected network, and on the right part here, you have a completely sparse network. Now, what is important here, don't look at a specific value because we just have analyzed a bunch of subjects, so we need still to work on this, but there are huge differences. So this is the clustering coefficient. Look at how different is the evolution in the frequentist case and in the Bayesian case. So I don't know, information content also, this is actually just the opposite. Here we have a lot of differences. And actually, we have some results comparing control subjects and patients, because in this case, this was a data set was ADIB2. So we have control subjects and papers suffering from autism, thank you, my co-author. And so the point is that we looked at the difference, and it's quite nice because if you use the frequentist approach, you don't see a lot of differences. But when you move to the Bayesian one, you start seeing more differences between patients and control subjects. So there is something there. So just to conclude and not to extend myself too much, because as technically I'm one of the co-organizers, it's not very nice that I will use more time than what I should. Just to say that essentially there are 3 messages here. So first of all, uncertainty, we cannot disregard it. I mean, it's quite surprising that in I think all instances that I've seen of papers in which they analyze a functional network, they just calculate one value and they say, okay, the correlation is 0.2 or it's 0.5. That's all. Well, yeah, but we cannot really know if it's 0.2. This is just our best guess. And then underneath, there is a probability distribution that we have to take into account. But no one takes that into account when we use functional network, and I think this is quite important. Second, uncertainty is not as bad as it seems. So in the first part, you have seen that it is quite bad because it's just biasing all the metrics, the topological metrics that we are seeing. But it's not actually that bad. It's a different way of looking at the data. So maybe inside uncertainty, we have some information that could be used as a marker to understand differences between control subjects and patients in several diseases or pathologies. And this is actually what we are seeing for alcoholic people. Now, what does it mean? What does this exactly mean in the context of, let's say, more biological context, or understanding what is happening in the brain that is making this uncertainty increase or decrease? That I still don't know. This is something that probably we'll have to discuss all together. And the third point, sorry, is that, well, okay, especially if you go to fMRI, this is a pain in a painful place, honestly, to calculate everything, but it's not impossible. We are doing that. And actually, we are planning to create a library and to let's say, to make this public in order to, I mean, if you want to join this idea. So finally, some references. The first actually is the only reference that I can give to you because it's already published. The other 2 papers are submitted, or we are working on that. In any case, if you want some more information, I will be happy to share a preprint with you at any time. Okay, that's all. Thank you.
So I'm sure that there are questions.
Yeah, thank you. But you were a little bit fast in presenting this EEG study. So I guess for connectivity, you used again correlation for this scientific reports paper.
Exactly. Yeah.
And then let me get it right because it was too fast. So you create the distribution by partitioning data into different chunks. Basically, you calculate it in different epochs, right? Is this how you create distribution of values? Because you said that it's not like 0.5, but it's many, right? It's a distribution of connectivity values.
Technically, what we do, let me show it here. So what we do is, we first calculate, whoa, this is very strong. So we first calculate for each pairs of channel, the full probability distribution. And this is Bayesian. So actually there is a frequentist approximation that was proposed, I think, by Fisher or something like that. And you have the expected value and the standard deviation of this distribution. So that's easy. This is for each pairs of channel. Then what we do is, instead of taking just the maximum value and put it inside the adjacency matrix A or the weight matrix W, whatever, what we do, we make several replication, several like instances of the same, and each value is sampled from the corresponding probability. So like here, for instance, you have that in the first, this connection between node 1 and 3, I guess it is, in this case specifically is sampled by, is represented by this value, then by this other value, and then by this other value. So you will have a bunch of networks instead of one.
Yeah, I thought whether you are using actually a bootstrap procedure for that. No.
No, not in this case. Yeah.
Okay, good, thanks. Yeah.
I'll quickly ask something also related to EEG.
Okay.
This is actually a question from Matteo Francini, who is following our live video stream, and he asked if you used as nodes for obtaining the MST, the sensors or the sources, and did you compare networks obtained at the sensor level and source level?
Everything is at sensors level. Yeah.
Okay, this is my own continuous question. Why? Like, why don't you just inverse transform?
I don't think I understand your point, but I don't think it's relevant in the sense that you will have exactly the same point, the same problem independently. So the point is that every time that you use two-time series and you try to calculate the correlation between them, you don't have a value, you have a distribution. That is something that, then if instead of that you reconstruct sources and you do exactly the same with sources, that's perfect. It would be very nice to do that, but I guess the result will be the same because the principle behind that, the main error is the same. I'm afraid of that, that will be the answer, but okay, thank you in any case for the one that are streaming.
I agree with you that clearly reducing just to a single correlation value the similarity patterns possible between two different time series is risky and reducing. But I have a mathematical problem because correlation matrices have a strong non-local constraint. They have to be positive semi-definite. How can you take into account this feature when you just reduce just to single pairs? You extract single pairs by single pairs the coefficients of the matrix, and so that you do not satisfy this non-local constraint.
Yeah, that's a good point. Actually, we don't satisfy. We don't do that. I know that there are techniques to do that. Actually, we never explored that point. That could be a future work, an interesting future work. Yeah.
So I agree that it's better to have the whole distribution rather than a single point estimate, but also you have the prior choice you do for the posterior distribution you get. So it's also a lot of variety and possibilities. So your estimates depends clearly on the prior you consider. So this is something to discuss in the Bayesian formalism. And then when you did, there are two questions. Then when you did the machine learning classification, the trial you were using as instances, the trial were correlated because they were coming from same people, no? So you don't have real independent instances for the classification problem.
Okay, good points. First of all, what you said about the prior distribution, all of that, that's a good point. Actually, just to simplify things, we consider the uniform, so no informative prior distribution. And actually what we did was to compare this with the approximation, I think it's the Fisher approximation, that is just a normal distribution in the tangent. There is a transformation that you have to apply.
If you consider the uniform, it's
Exactly, yeah, but the point is that we recover the, let's say, the classical approach, just we have the uncertainty associated with it. So let's say that the maximum value, the expected value of the correlation will be the same in the classical and in Bayesian, just we add some uncertainty. And we actually thought about the possibility of using some more interesting prior distributions, but this is, I feel this is not that simple. It's not simple at all, actually, I guess. It can be quite a mess. So about this of the classification, I went really fast because I know this is not an audience about machine learning and so forth, but there are several things. Here we have used a cross-validation in which we excluded in the testing of in the testing of the trial of one subject, we excluded all the data of that subject. So the validation is not... Exactly, yeah, but they are correlated. We consider that the trial are independent.
But they are not.
They maybe not, okay, but I don't think that will be.
One person is doing several trials.
Yeah.
Those trials are correlated.
Exactly, but we don't use the trial of the same person to validate and to train. That's important. So from a point of view of machine learning, this is what you should avoid. And then the fact that you have correlated things, that's okay. The point is that you cannot use the same subject to validate and train the model at the same time. This is what we are avoiding.
So I think I missed the methodological detail in your comparison of frequentist and Bayesian approaches. You described that when you go from these R correlation matrices to binarized or weighted matrices to people do that to calculate topological properties and metrics. What I missed, and I'm sorry if you said already, is did you apply any thresholds? Some people apply small thresholds, big thresholds. Some people apply a range of thresholds and take an average.
Exactly.
Which was your approach?
Yeah, in this case, it's not really important. I mean, for us, it's not really important. So what we did, we just considered that we applied a threshold. And as you can see here, so here you have different, this is, yeah, these are thresholds. No, these are link densities, actually. So we kept like the 30% of the link, the 35, 40, and so forth. But the point is that it doesn't really matter. Because the point is that if you consider like 50% of the links or just a 20%, the point is that is all the steps that you have done to reach that. So as you can see, we have the same bias in the topological matrix, more or less the same bias, independently on the number of links that you include. Now, this is true if you apply a different like thresholding, like you use a fixed threshold instead of a link density, or you do something based on machine learning or whatever. So this will still hold.
No more questions? Okay.
Okay, thank you.
So our next speaker, I think, will be Fabrizio Vico Fallani from the Institut de Cerveau de la Moelle in Paris. And he'll continue the session on network construction, so building networks. I don't know the exact title, but
yeah,
I think that's going to be the flavor.
I changed it from the...
Okay. Here you go.
It was a little bit too much generic.
If you prefer this one.
Okay. So thank you very much for the opportunity of being here, and thanks for the organizers for having invited me. It's actually my first time in Lille, so I'm quite happy about that. And I think that the last questions we had on the previous talk was it's a very nice assist. Will allow
Somebody has one. I don't. But
Okay, yes.
welcome back. Nobody's complaining, that's good. So let's see, now we have covered some details of time series. So we have data, we know what to do with the data, we have some sort of idea how is the data. Then we have concluded that it's impossible to build a brain network. Basically, you can't define nodes, you can't define links, you don't know how to threshold. But now, let's not get depressed because of that. Let's move on to the third session of today. And now we, for a while, assume that we actually have a brain network. And this session will include some analysis one can do using the network, and also different, a bit more advanced models for modeling the brain as a network. And our first speaker in this session will be Daniele Marinazzo from University of Ghent, with the title of Redundancy and Synergy in a Network Perspective. So or networks in a redundancy or synergy perspective. Do you want me to make more jokes or?
Is it like that? I
okay.
was actually torn about what to say.
So our next speaker hasn't yet decided what he's going to talk about, but anyways.
It's actually the same, right? So.
Do you prefer that or this one?
Okay, well, I don't know.
Welcome, anyways.
Thanks. Okay, so thanks. It's a pleasure being here. And so we'll try to discuss some concepts which are ubiquitous, yet elusive in their definition. That is redundancy and synergy, when we want to talk about a system made of different units. So whether it's one or the other, we don't care. But it's more important to define information in this sense, because redundancy and synergy will be defined in terms of information. So information also is a quite complicated concept to define, but we can define it in terms of the amount by which one variable, which can be an answer to a question, a signal, certain measurements, reduce our uncertainty or surprises us about another variable. So typically, this is quantified using a probability distribution. We have a very boring case of the one six-sided die, or still boring, slightly less, two six-sided dice, or something that my friend Joe loves to use to introduce information theory is the guess who game. In this case, it's about the strategies of guessing the character. So we have a measure of the amount of information to describe a guess. So in this case, in this case is the entropy. We still don't know, but let's say that there is a lot of information when we pinpoint the exact character. then in this original version the game was not gender balanced, and so there is a little less information knowing that the character is a female, or even less information knowing that the character is a male. Now, what's the best question to ask first? And then once we know the answer, what's the second question? So for example, we can ask whether the character is male or female, then we can ask about the color of the hair, and this is the concept of joint of mutual information, or joint entropy if you want. So for example, there is no joint information from identity and sex because it's all contained in the identity. On the other hand, you gain some bits extra of information in knowing that the character is blonde after knowing that is male or female. Now, this was for the old version of the game. Now they made a new version of the game which is perfectly balanced between colors, male, females, or whatever, and the game is a disaster. It's completely boring. So my kids have been playing. This is a real picture of my kids' game, and all the summer, no one was guessing first. They were all guessing at the same time. So it's good to have diversity, but not for this game. Now, we can also build distributions probability from the past of the system. So if you have a dynamical system in which we have the future and the past, we have the arrow of time, we can refer to a target system, which can be any part, univariate, bivariate, or multivariate of the system itself. And so, in the most general case, we have a dynamic process made of all the other actors of the system and the target. And we define two quantities, the present of the system and the past of the system. So now we go to the case of just two variables or two groups of variables, but we can, and actually we will, extend it to more variables. So in this case, if we consider the flow of time, we can study directed dynamical interactions within processes and between processes. And the two quantities, information-bearing quantities, are the past and the present of the system. Now, sorry, I could not get rid of the game. So Granger causality is a family of methods to answer this type of questions. On the other hand, the same ideas are true for the so-called model-free estimation, even though even a probability distribution is per se a model, so there is no such a thing as model-free, at least in this case. The idea is that the Granger cause precedes in time its effect, and then the Granger cause contains unique information about the future values of its effect. So in the simplest case of a dynamic process of x and y, one driver and the target, the information flow graph looks like this, in which you have the internal dynamics, so the memory of the variables, and then we have the directed dynamical interactions at zero lag or lagged interactions. So in this case, our notions of conditional independence or conditional mutual information help us to define this type of Granger cause relationship. So for example, we can detect a directed dynamical interaction from x to y by comparing about the future of the, or the present in this case, of the target, given the past of the driver conditioned to the driver to the past of dynamics. So we have to somehow decompose this h quantity that we define at the beginning. So the information generated by the target is called the system information in this case. And this is the information contained in the presence of the process. In this case, we can first decompose it into quantities. First of all, we have the predictive information about y. So this is the information contained in the past of the system that can be used to predict the present of the target. And then we have the unexplained information about the present of the target given the past of the system. And so in this case, we have a first part of the entropy decomposition in prediction entropy and unexplained information. Now, this prediction entropy we can set up to further decompose it And in this case, we can decompose it into quantities. One is what we call the self-entropy or information storage, which is the information contained in the past of the target that can be used to predict its own present. And then we have the transfer entropy, which is the information contained in the past of the candidate drivers that can be used to predict the present of the target above and beyond the information contained in the past of the target itself. So this is important. We need something that adds information. So in the simplest case of a Markov process of two variables, in which we have internal memory of only the driver, and then we have the strength of the influence given by the parameter gamma, the information storage, the target, grows with gamma when we have different values of memory of the driver. On the other hand, what happens with the information transfer? In case of no noise, so in case of perfect memory of the driver, when the coupling becomes too strong, driver and target became the same thing. And so basically we don't have any more transfer. So the correlation would go to 1. On the other hand, the transfer entropy goes down to 0. This is not the case when we add noise or we remove memory from the system. So we have this situation, which can be extended in the case of multiple drivers, in which we have the total information in the target, the information storage, and then the information transfer. If we have at least 2 drivers, we can talk about transfer from one driver, transfer from the other driver, and then information modification. So the total information is decomposed in storage, information transfer, which is the directed connectivity, and then the information modification, which is the interaction between the information provided by the 2 drivers. And then we have the so-called new information, which is non-predictable dynamics. So this is the most general scenario. How do we compute these quantities? We can either compute it with information based with the probabilities, or we can compute it using a reduction in surprise and reduction in variance. We will see later. And so in this case, we have the entropy, which is just the logarithm of the probability distribution. Then we have the conditional entropy, the mutual information, and the interaction information, which tells how, let's say, it's a difference of the 2 mutual informations minus the joint mutual information. In this case, we can define all these variables. So the present information, which tells us about the uncertainty about the present state of the target, then how much the target is predictable from its own past, which is the information storage. Then we have the information transfer, which are the interaction from all the sources to the target. And then we have the new information, which measures the complexity of the target given the past of the whole system, let's say. Now, we are interested in the joint information, in particular to the joint effect of 2 or more drivers on the target. And so in this case, the framework of interaction information decomposition defines redundancy and synergy as to whether the 2 transfer entropies overlap or don't overlap. If they overlap, then we say that the joint transfer entropy is smaller than the sum of the 2 individual transfer entropy, and so we have redundancy. On the other hand, when this quantity is bigger than the sum of the 2 transfer entropy, we have what is called synergistic interaction. Now, this is not a perfect definition, we'll see it later, but still it's something which is already relevant when we want to detect interactions between different components of a system. Indeed, redundancy and synergy need to be taken into account when we compute interactions between dynamics. So we can, when we want to reconstruct a network, sorry about all the multiple quotes, but it's the purpose of this meeting, I guess. So the first example is the collider. So the lower part of the matrix is the pairwise interaction, and the upper triangle of the matrix is the conditional interaction, so the multivariate interactions. So even in this simple case, in which we have 2 random variables, and the third variable, which is made by 2 uncorrelated variables plus some random noise, this dependency induces a false positive if we compute the conditional, the partial correlation or the conditional mutual information. On the other hand, this is not revealed in the pairwise interactions. On the other hand, if we take into account the non-negative interaction information, we are able to detect this one as a spurious link. Another case is the case of synergy. So when one variable acts as a suppressor for the other variables, as it happens here for x2, which acts as a suppressor for the influence between 1 and 3. So for example, here in the pairwise interaction, we don't see this type of interaction. On the other hand, here we see all the correct direct interaction, but we would also see a spurious interaction between 2 and 3, but this one is correctly labeled as a suppressor if we take into account the shared information. And then last case is the case of redundancy. The simplest case of redundancy is a chain-like behavior, in which, of course, the pairwise interaction give all non-zero values. On the other hand, the multivariate would also give some false positive. On the other hand, if the non-zero interaction information is taken into account to flag these possible interactions, then we are able to correctly reconstruct this interaction in directed and mediated. Now, apart from the computational aspect, then also synergy and redundancy can reveal some mechanism of the system under study. So let's take a brain, but it can be anything. Two regions send both information to A, so both contributes to the prediction of the future of A, but what about the joint influence of the two of them on A? So we go back to our framework of information dynamics, in which we have information storage, transfer, and modification, which can be expressed either in terms of entropy or in terms of variance. And then the interpretation of information modification in terms of redundancy and synergy. Now, do we really want to reconstruct a network from the observed dynamics? Maybe yes, maybe not. Let's say that most methods, in particular this type of methods, are meant to disclose the effect and not the mechanism. So they can tell us something about what we want to look at, but maybe they don't tell us about what the actual network looks like, if ever there is a network underlying there. Then the effect can involve links resulting from non-trivial dynamic effect, so higher order interactions. And then if we don't have a perfect generative model, and if we don't know all the possible confounders, then you can say, okay, here there is a spurious link. Okay, but the spurious link can tell us something about whether there is a confounder or not, or whether the global dynamics has the same properties of the target system or not. And then, of course, ROIs are bad, don't exist, etc. So now, let's try to, first of all, solve one problem. So from interaction information, let's go to partial information decomposition. So in interaction information, synergy and redundancy are seen as mutually exclusive phenomena. So you have either synergy or redundancy. So it's a net effect. You cannot have both of them. On the other hand, we would like to have a situation in which we can allow redundancy and synergy to coexist in this case. And when they coexist, they have this property. So the total entropy is the sum of unique contributions plus redundant and synergistic contributions. The individual transfer entropies are the sum of the unique contributions plus the redundant ones. And the interaction information is equal to the difference between synergy and redundancy. Now, we need one extra relation to have this framework. Because in this case, the partial information decomposition components cannot be obtained through the classic information theory, simply subtracting conditional mutual information terms. So the classical channel information theory does not univocally determine the decomposition, allowing synergy and redundancy to coexist. And so in this case, we need to make a choice. One convenient choice is to define the redundancy as the minimum of the individual transfer entropy. And this choice is convenient because prevents us to define as a redundant transfer entropy the simple correlation between the two source processes. So that would be a trivial example of redundancy. Information dynamics is also relevant close to transitions. So if we model even a very simple binary dynamics like the Ising system on the structural connectome of the human brain, we see that in the critical regime, the correlation between spins is maximized, the transfer entropy among spins is maximized. On the other hand, the link-wise correlation between transfer entropy and structural connectivity is minimized. And also the similarity, link-wise similarity between the functional correlation matrix of the spin dynamics and the structural connectivity underlying. So when the system displays a richer repertoire of dynamics, to some extent it has to forget its underlying structural connectivity, at least at the level of individual links. It's less true in terms of modules, as the work of Jesus, which we'll tell you about later, will show. Anyway, something happens close to transitions. Another thing that we expect to happen in transition is that when the system is completely not synchronized, the transfer entropy will be minimized, but also when the system is completely synchronized. For what I told you before, we also have a drop in information because we don't need anymore, or we don't need so much the driver to predict the target. And so this is actually what happens in a system of Kuramoto oscillators on a lattice, in which we have the synchronization which goes from 0 to 1 in case of explosive synchronization, and also a total transfer entropy also goes immediately up and then goes down again. Now, what comes first? The transition to order or the peaking information? In a paper of a few years ago, it is shown that the global transfer entropy peaks in the paramagnetic phase, so just before something critical happens. And so this is good as a biomarker or a marker in general of a warning sign of a transition. But on the other hand, this involves computing transfer entropy across all the possible variables of the system, which is a bit unfeasible. On the other hand, using partial information decomposition, we found that the responsible of this peak in the paramagnetic phase is the synergy. So the redundancy, for example, peaks exactly at the critical value. On the other hand, the synergy peaks in the paramagnetic phase. Here it's inverted, this is the inverse temperature and this is the temperature, but what comes before is after actually. And so the synergy is responsible of this warning sign. So this means that we need maybe as few as 3 variables to have a warning sign of a transition in a dynamical system. Now let's visit quickly a few applications. The first one is psychological networks. So it's now a hot topic in psychometrics to define psychiatric disease or conditions by looking at the interaction between the different responses given by all the subjects to questionnaires. Or, for example, I don't know, do you manage to sleep well? Do you want to kill yourself, etc. And so by looking at the probability distribution across subjects, they build a network of symptoms and they try to target, for example, the symptoms which are more central to the disease. So just to take an example, this from some colleagues, is always good to shit on colleagues' work. No, I'm joking. But in this case, they have 1973 young adults, and they look at a questionnaire based on different items describing empathy. And they are divided in 4 groups: fantasy scale, perspective taking, empathic concern, and personal distress. So if they retrieve the network from the study, they get this picture, which is nice, as all networks are all nice pictures. On the other hand, that doesn't tell us so much more than the original distribution in 4 communities given before the network was computed. On the other hand, if you take into account the non-zero interaction information are mostly expressed in this way. And so if you condition the network to this non-independent interaction, you see that there is only one group of variables which now is central, which is the perspective taking. So it looks like perspective taking is more central in understanding empathy in this case. This framework can be extended over multiple time scales. So we can, using the state space model and the discrete algebraic Riccati equation, we can obtain an exact multiscale decomposition of interaction information. And we applied this epilepsy in a case in which we had 64 cortical electrodes as a target. And then we had 2 depth hippocampal electrodes, which were number 11 and number 12. They were both candidates as putative foci of epilepsy. So these are the 2 drivers, and this one is the target, the cortical grid there. So in this case, if we look at the joint transfer entropy, this is before and during the seizure, the individual transfer entropy and the interaction information, they are both modulated by the presence or absence of the seizure, but we don't see any difference between channel 11 and channel 12. On the other hand, if we look at the partial information decomposition, we see that the quantity which discriminates between ictal and preictal phase and separates 11 from 12 electrode is the unique transfer entropy from the electrode 12. So this only using partial information decomposition, we can tell the 2 different dynamical contributions of the first target and the second target, namely ascribing to the second target the most unique information. And this is what we observe at different time scales, in which the unique transfer entropy from the electrode 12 mostly separates between the pre-ictal and ictal stage. Now I will conclude with 2 other perspectives. The first one is synergy and redundancy seen as network themselves. So if we look at the matrix of redundant information, of pairs of variables which are in the same redundant duplet or in the same synergistic duplet, we see that these matrices have a hierarchical structure. And this hierarchical structure, this dendrogram, is very different in case of redundancy and in case of synergy. Also, the distribution in this case is average data on the human connectome project data. We have a different distribution of synergy and redundancy across the brain. More importantly, if we now use as node of a network the independent components, so neither anatomical regions nor functional ROIs, but individual independent components, and we apply synergy and redundancy at different time scales, so the smaller time scales and then up to longer time scales of a dozen of seconds, we see that we have different structure of this dendrogram. And if we look at the individual leaves on the dendrogram, the colors reflect the intrinsic connectivity according to EOS parcellation. So we see that indeed we don't have a perfect separation of regions in the intrinsic connectivity nodes when we take into account both the synergy and the redundancy. On the other hand, we reobtain the EO networks if we use the zero lag information. This tells that in this case we have an alternative decomposition which is both hierarchical in time and space of the intrinsic connectivity in the human brain, and also a different structure, between synergy and redundancy. And now we conclude with synergy and redundancy among networks. So now let's imagine to have a multi-layer network, which is what Onerva introduced, and maybe someone else will do later. In multi-layer network, we usually compare layers pairwise. So we have, for example, mutual information between layers, or we have edge overlap, and so on. sometimes we use information-based measures, and sometimes use mutual information, or also spectral entropy and the Kullback library divergence are convenient because allow to compare networks at different resolution of the network. Now, hopefully I will develop a framework to use information. So far I was just playing with an extension of the Jacquard index. So let's suppose we have 3 networks with the same nodes. And we have 3 cases. The case in network C has nothing to do with A and B. So in this case, the Jacquard networks, on average, will be the same if it's evaluated just on the links where C equals to 0, because it doesn't have an effect on A and B. On the other hand, if the network A influences the network B with the mediation on network C, if we remove the links of C, the Jacquard index will go towards 0, because the similarity will decrease. On the other hand, the synergistic case would consist in the fact that having a link of B depends on having a link in the union of the subset A and C. And in this case, removing the links of C will enhance the Jacquard index between A and B. And so this is what will happen if we compare the Jacquard index with the pairwise Jacquard index. So in this case, we have independent uncorrelated relations. So the 2 Jacquard index are the same. On the other hand, in the case of mediated interaction, the global Jacquard index is greater than the pairwise Jacquard index. And in the synergistic case, the global Jacquard index is smaller than the pairwise Jacquard index. And just to make a test, I used the C elegans connectum, in which we have 3 layers. The functional connectivity, so the correlation, the electrical synapses, and the chemical synapses. So in this case, I computed the Jacquard index, the difference between the 2, for the actual values and for 1,000 random permutation, and see that indeed, in this case, the chemical synapses mediate the relationship between electrical synapses and Pearson correlation. It's not a very profound result, it's quite obvious, I would say, but on the other hand, it's just a proof of concept of how we can define this higher order information between networks. So thanks my collaborators, and thanks to you.
Any questions?
Thank you. Well, in practice, of course, if you are talking about MRI or EG signals, we have a problem of, volume conduction. That's clear, where you spuriously get this. But then there is also signal-to-noise ratio.
Yes.
So the problem is that you might have one source, which is visible by 10 electrodes. But because of different signal-to-noise ratio, information will always flow from strong SNR to weak SNR. Do you see what I mean? And no.
I see.
No, the question. No, I'm just formulating the question. So, for instance, what to deal, especially in case of fMRI, where you cannot directly measure signal-to-noise ratio. But even imagine if there is a third source. I'm not referring to the problem of third source as such, but I use it to build an example. So it kind of drives 2 voxels, which are literally the same, like they have the same activity, but there is more noise in one of the voxels. So therefore, of course, you will predict activity of this voxel with a voxel with better signal-to-noise ratio.
Yeah, but so they're not equal anymore. If
exactly.
One is more noise than the other. So then, of course, that's why I was saying that these measures look at the effect, not at the mechanism. So if you have 2 voxels and one has a higher noise content, then in this framework, they are not equal anymore. Indeed, we have a study just completed in which we compute the partial information decomposition in the joint effect of the global signal, which is thought to reflect noise, let's say in the sense of movement, but also it's by definition a ROI containing all the neural signal. So that's why sometimes people say, okay, but the global signal contains surprisingly neural activity. What the fuck? It's really the brain itself. And so in this case, you can disambiguate the effect, even if the global signal and the signal in vessels are correlated up to 0.99%, you see that they have a differential contribution on the cortical ROIs. So even if one is thought to carry low frequency fluctuation, and the other one is thought to carry anything by definition, then in this case you can have that they have a differential effect, which is mediated by the difference, for example, in blood arrival time at different regions.
No, it's good. It's just that your transfer entropy example with epilepsy, I like it because it's kind of a biomarker, but it could be biased by the signal-to-noise ratio and volume conduction because transfer entropy is biased by this.
Sure. So in this case, you have, let's say, two electrodes which are really a few millimeters away, and also, let's say, their spectral content is the same, and also even the individual transfer entropies look the same. And then on the other hand, when you compute when you see not only their own predictability, but how they influence the predictability of an ensemble of electrodes on the scalp. In this case, maybe since the target is the average of all the cortical electrodes, in this case, maybe the noise is also, let's say, average out in this case.
Okay, thanks.
Further questions?
How does the composition differ from what people find in standard techniques?
Which one?
The decompositions that you had, the two ones that you had in the end, not before on the cortex. Those ones.
Yeah. So if you apply this to a zero lag interaction, you will see that the leaves on the dendrogram will perfectly order all the regions. Will order the intrinsic connectivity networks by definition in the default mode network, the Salic network, the visual networks, and so on.
You get the right, okay.
Any further questions? Anyone? No? Okay.
Thanks.
Thank you.
Great talk.
Brisbane, Australia, and he will introduce us a measure for network flexibility, a thing called functional repertoire. Let's see, what's that? Welcome. This one, do you prefer that one?
You know. I can use this other one.
Okay.
Yeah, thank you very much. Thank you for the opportunity and for the invitation. Thank you, David. Yeah, it's great to be here. It's a great conference and workshop. It's really nice I look forward to discuss lots of ideas tomorrow as well. Yeah, it's a nice get together of interesting people and interesting lines of work. So yeah, I'm gonna propose this idea of this functional repertoire, which is very simple. And yeah, so it's gonna be kind of a proxy to estimate this network flexibility. So this brain network flexibility would be important for cognitive flexibility. So a requirement for cognitive flexibility. And we want to apply this, we will apply this for a case of Parkinson's disease later on. So yeah, this is one question that could be a topic for tomorrow. So what makes the brain healthy? It's a big question, but yeah, I'm gonna just focus on a key essential things. So elements, so one is these efficient communications between regions. And the other one is this efficient reconfiguration of patterns. So yeah, it's going back to the repertoire, to having a flexible repertoire. You need to reconfigure the brain and efficiently. So I've been working on this topic for a while. My first contribution was in this special issue that was organized by David and Javier and Stefano and Boomor in 2014. So yeah, the idea there was that we were trying to look into this problem of this flexibility, and I was looking in terms of frustration, which I'm going to introduce now. So I started looking into this from the modeling perspective. So you can simulate the activity of the brain. So you can assume that each brain region, so you have to assume an ROI, so each brain region, it has excitatory and inhibitory neurons. So it can combine to populations of excitatory and inhibitory neurons. So they can interact according to the connectome, and one can look into the emerging dynamical patterns that come from this brain. Instead of looking to the whole brain, what I was doing first was just looking into the brain and also in the motif. So how do you understand? Can you look into subparts of this network, these building blocks of this network, to understand better what's going to be the emerging brain activity that can be very complex. So in this case, we are looking to this, and one very basic feature was if you look into the coupling strength, if you vary in the coupling strength, and if you have a very weak coupling, regions, so these are all the possible regions in this motif. So you can look into the white dots are going to be when they are synchronized. So this is synchronized at 0 lag. So if the coupling strength is very weak, you don't see much synchronization, and this synchronization only appears sporadically. But then if you have a too strong coupling, then you have a lot of synchronized states. But then the diversity of these patterns is not very large, because you often get these guys synchronized. So those are within these models, and within these models become synchronized very fast. So this has a lot of synchronization, but doesn't have a lot of diversity or variability. And here there is an optimal coupling in which you have a larger possible variability in these patterns. So yeah, this was one case for this particular motif. But then if you look into the whole brain, this is even smoother here. So this was a macaque brain of 30 regions. It's also a very coarse division of this macaque brain. But just to illustrate this point that there is an optimal coupling strength, that you get a larger variability. So people has looked into this problem also in terms of criticality. So you can think, for instance, in the branching ratio. So yeah, Vadim was explaining a bit the criticality in many contexts, but he didn't focus too much on the branching ratio. So the branching ratio, it's this very simple idea that if you have a branching ratio that's bigger than 1, your activity grows in this system. So one active element generates more and more and more, and eventually you have a lot of activity. So this is a supercritical system. And then you can also have another case in which this sigma is smaller than 1, and regardless of how much you have at the beginning, this activity dies out very quick, because this is a subcritical system. And there is a critical system in which, in average, you get one active region after each other, but it's a stochastic process, so this activity can die out as well. But you can have very large bursts of activity. You have these that have many different durations, many different sizes, and these various orders over several orders of magnitude. So yeah, this has also been applied in terms or in the same idea. So kappa is also this indication of criticality, how far you are from the critical system. So this equals 1 would mean a critical state. And then in this case, you have, this is just the synchronization, that you are at the edge of this network that starts to synchronize, and you get the maximum entropy, the maximum variability of these patterns when you are in the critical state. So this is the framework that we are trying to look into this. But instead of focusing on this modeling, I want to go beyond that and look into this more in real data, in human brain data. So, well, we can look into non-invasive brain imaging methods on many different ones. So you have
a problem that's very hard to get both high temporal and high spatial resolution. So you either have to choose one or the other. And then in this case, in order to get what we thought was the optimum, the best temporal and spatial resolution, we were focusing on the MEG data. So this MEG data was acquired in Naples. So this is the very similar system to the Human Connectome project that they have in Chieti. So those are actually 163 magnetometers that are used to acquire this data, which is a very high quality MEG data. And we are going to compare this data of healthy brains, well, healthy people, and Parkinson's, and patients with Parkinson's disease. So this data set has 38 healthy controls and 39 Parkinson's disease, Parkinson's patients. And yeah, well, this has been covered before. So this is the substantia nigra, and then we have patients that have a big reduction in neurons there. So this is a problem in the dopaminergic circuit. But yeah, so this you can see a clearly signature here, but this affects the activity of the whole brain. It's not just at this region, but at the whole brain. You can see patterns that are affected, and that's a bit of how we want to look into this, not look into the spatial temporal patterns, not only at a specific site, but more at the whole brain and the complexity of this. So in this data, this is the protocol. So it's a very simple protocol, just a resting state protocol with eyes closed, and each participant and patient lies there for 3.5 minutes. So this is, you have half a minute just to calibrate the system, and then you have 3.5 minutes. And then you have a break just because during this acquisition, the patients or the participants are meant to stay still. So you do this again just to get more data. And we combine all this good data that doesn't have much movement or artifact of both healthy controls and patients. So, well, we have this raw data, and then we do the source reconstruction. We also have to assume a parcellation. So in this case, we used just the AEL parcellation, which is a very standard parcellation for this, and it's the most common one used for MEG data. So the time series are going to be, well, for each region, you have time series that can vary a lot. And we are trying to identify the regions that have these peaks in activity. So these peaks, we are going to focus on these peaks. We are going to focus on the avalanches that are caused by these peaks. So these peaks we identify as moments in time in which the activity has a z-score above 3. So it has 3 times above the standard deviation. So those are the high, or the moments of high activity. It could be high or low. We just do the absolute value because we are not sure of what's happening there. We just want to capture when there is a consistent activity happening at this region. Yeah, so another important thing to take into account is that we have to define these avalanches as moments in which there is activity. And eventually there is no activity, there is no region that's above this threshold. So this is when the avalanche finishes. So you can look into what happens between these events in which there is something happening. So this is the schematics. We have this, and we look into what's happening here. So we have all these regions, and we can see events in which regions were active and moments in which there is not activity in terms of this high activity. So there is activity, but not strong enough. But we are going to look into these patterns of this high, consistent activity. We are going to focus on these avalanches, on these patterns of these avalanches. So this is one example. We have an avalanche here, and we have regions that are active, and we actually combine all of them, and we look into these patterns. So we are going to just do this very simple method that we are proposing here, just to quantify or to characterize these patterns that we observe in patients and in controls. So yeah, this is the idea. We have this. We combine these regions. They can be above this threshold for a while. But then we combine it, and the pattern is made of all the regions that were active for any time during this period of activity. So you combine all these regions that were active, and this generate these patterns that we are going to look into the statistics of these patterns. So we can look at this in different ways. So we can look at this at the individual level, but this is just the combined patterns that we observe across all patients and across all controls. So in this case, we have above 600 different patterns in patients, but we have much more patterns that can happen in controls. And this is the main statistics that we are going to look into that. So this will be, so we can look into this in MAG. We have this high temporal resolution, so we can look into this in different frequency bands, but the results are all consistent. So yeah, the broadband is probably the most informative one. It's much more complex here. So in general, there is a clear difference between this number of patterns that we observe in patients and in controls. So in controls, in general, you have much more different patterns that you would find in patients. And then as you look into across different frequency bands, slow frequency, you have less patterns because you have the duration of the events, or the duration of the recording doesn't allow that many things to happen. But when you have high temporal resolution, you can see much more patterns there. But there is a significant difference across all different frequency bands. Regardless of which frequency you look, you always find controls having a more complex type of dynamics than patients. So there are many possible reasons for why this could happen. And one first reason we checked was perhaps there is more events that happens in controls than in patients. But that's actually not the case, it's actually the opposite. So in general, there is more events that happens in patients than in controls. So those are the number of events. So the number of switches, if you just count how many different activations you find, so the rate of these activations, you find a larger rate in patients than in controls. So it's just the pattern. So the flexibility of these patterns that occur, they recombine in much more different ways in controls than in patients. So yeah, this is the main idea. So you can also, for instance, look into combine these different patterns into modulus, and you find the patterns in patients tend to be more similar than you would find in controls. And another thing, one can look into this, and can look into different patterns. So the patterns that are common to both groups, or the patterns that are unique to a different group, to a specific group. So if you look into this, the patterns that are unique, they often involve these regions that are more well known to be involved in Parkinson's disease. So you have the thalamus, the pallidum, and the putamen, which is consistent to how we would expect to have these regions involved in the different patterns. So these regions, they appear more often in patterns that are specific to different groups and not common to the two groups. So yeah, one can explore these in different ways as well. So one way that we would explore, would like to look into this. So this is the UPDRS score. So this is a most common, most basic way of quantifying the severity of the disease. So the larger this score is, the more severe it is. So this is based on motor symptoms. So if you have very strong score, the motor symptoms are impaired. And this is also correlated with the number of patterns that you observe. As patients have more impairment, the patterns are reduced. So more patterns indicates a less severe symptoms in these patients. So then we were trying to understand why this has happened. Can we shed any light on the reason for this? And then in order to do this, we actually went to this. It's a phase linearity measure. It's the PLM. So you have two-time signals, and you look into this interferometric signal, and you look into frequencies that are common to the two, and look into this area. So this indicates that, well, if regions are very synchronized, you have a large value of PLM, and if you have weak synchronization, you have a low value of PLM. So this is a recent proposal to characterize and to quantify synchronization. We can go through these if you guys want. But this is just in a way of looking and estimating the synchronization in this system. So it actually, we were looking into this into the beta band, because beta band is also known to be involved in Parkinson's disease. And what happens is that this functional repertoire in patients, as they decrease, so if you look into this, the patients that have a lower functional repertoire, they also have a stronger synchronization. So this would be one reason for why they have this more restricted repertoire. That would be because they have a more stronger synchronization. So that doesn't allow them to explore much the possible states. So they are over and over in the same, following the same type of states. So yeah, this will also be consistent with the idea that you have more switches, you have a stronger rate of activation, but it's always or very common in the same type of patterns that you'd find in these patients. So another assessment is this Montreal Cognitive Assessment. So this is related to attention. And as in patients as well, patients that have a stronger repertoire have a better attention and better scores in this test. So this also, it's a way of going beyond the motor symptoms and going more on assessing brain activity in general, and not just focusing only on the symptoms that are observing motor activity. So yeah, this is I went too quick. But yeah, so yeah, this is the main idea that I want to propose. So this measure this functional repertoire, and yeah, I would like to propose that this can be useful to better understand brain activity, and can be used in this context of Parkinson's. So yeah, this message is that healthy controls have more flexible patterns of these avalanches, and then we can ask questions what this really mean, and those are deeper questions that we are just assuming that this is a proxy for estimating this flexibility of brain activity, which is also in a fast time scale. We can also think of what would that mean in a slower time scale, or can this be extended to a slower time scale? There are many questions that can go in this direction as well. So yeah, these symptoms correlate with this functional repertoire. So this indicates that this functional repertoire can really be meaningful and important. So this is the main scale that is used in the clinics. So this can be appealing to clinicians as well. So yeah, this is our explanation lies in this direction, that there is a hypersynchronization happening in Parkinson's patients. So they would also have a lot of activity, but this activity, it's very, very often happening the same way. So they cannot really explore all the different patterns as they could, as it happens in aging matching controls. And yeah, and this is also, so this functional repertoire is also associated with this attention. So it's not just with the motor symptoms, so it goes a bit beyond that. And that's an interesting way. So yeah, those are the basal ganglia regions that are associated that appear more often in the patterns that are distinct, so that the patterns that these these patients have, they are also often involving the basal ganglia regions. So yeah, those are the main conclusions of this study. And yeah, I would like to thank the collaborators who acquired the data, and especially to Pier Paolo Sorrentino. So he's my colleague, and we've been working on this data for a while now. And yeah, I'd like to thank you for your attention.
Thanks. So finally, I got a chance to ask somebody who is using this EEG MEG avalanches. So for non-invasive measurement. So Bex and Plant started with really spiking. So then other people went with LFPs. And in LFPs, when we have a negativity, we can say that it relates to spiking. But it's already a stretch. From that, there is a kind of a jump of faith to non-invasive measurements, where you have no say with respect to spiking at something completely else. So I also see now that at least you're using narrow band, because oftentimes people use broadband, and then it's everybody's guess what lies above this red line. So, but what's your understanding of this avalanche, basically? Because, I mean, it definitely doesn't relate to spiking. It's just basically thresholded activity. Or what kind of neurophysiological meaning do you put to this?
Well, at the end of the day, this is going to be moments in time in which you have this region that's consistent, that the activity of these regions consists. So you have neurons that are going to be activated together or nearly together in this brief time window. But it's true, yeah, the advantage, or the things that all the results hold for all the frequency bands and also for broadband. So it is a bit stronger to say that you don't need to filter in a specific band, and it holds even in the broadband. But it's true. There is, well, yeah, people might think that broadband it's too broad and it's not specific enough. There is a never-ending discussion on this, what that really means.
It's just that ideas, especially in criticality, they were developed for really unit activity, where you can really infer the unit activity, but this one is something else. So it's
yeah,
very
so
hard.
The idea of this avalanche, so yeah, the idea is that there would be in a specific region this consistent activity, but this happens like in specific time, so across different regions in specific moments in time. So it's like once one region has this spike in activity, it's more likely that other regions would also have this spike in activity. So that's why you see these clusters and you don't see this homogeneously distributed here. So you have these specific avalanches, and you can look at the spatial temporal patterns, and yeah, we are focusing more on the spatial patterns here. But yeah, it's a very basic thing, just looking at this flexibility, this how many you find there.
Okay, thanks.
Yeah, thank you.
Thanks, Leonardo, for this talk. I wonder how robust is your method against the threshold that is used to define, not say avalanche, but your extreme event.
Yeah.
I mean, I know that, of course, if you fix a very high threshold, you will have a very, very sparse number of patterns. But I wonder if even on this case, the organization of those patterns remain the same.
Yeah, no, that's a very important question. That's true. This is, of course, as we did here, this is just an arbitrary number. But yeah, we did vary this, and at the end of the day, it doesn't really matter much how you put this. Well, if you vary too much, it's true, it doesn't hold anymore. But there is a big range from 2 to 3.5 in which you still see a power law in the distribution of the size of the avalanches. And you also see you can also compute the branching ratio. That's going to be 1. So we choose this because it's very close to 1, this branching ratio. So yeah, that indicates that the system is critical. So this is also touches upon, well, this, of course, this work in which they actually define neuronal avalanches as only avalanches that have this critical exponent. But yeah, but I don't know. I find this a bit too specific because it doesn't need to be like this. But yeah. But yeah, in our case, it's also a critical system in this sense that you have this branching ratio of 1, and you have this power law with this exponent of 3/2. And
same question with respect to time. How does it change for how, depending on how long you observe?
Oh yeah, so this is actually an issue here, because so we have these recordings that that are with eyes closed, and we wanted participants to be awake, so we couldn't have this for a very long time. So it's kind of a trade-off here. You have to hope that they don't fall asleep within 3.5 minutes.
But for example, if you calculate the size on 1 minute, and 2 minutes, and 3 minutes, the difference between the two groups you find is the same, or separate speed? How does it work?
Yeah, no. So well, we tested all these variables, but yeah, it's very robust in the sense that, well, because the basic measure is how many different patterns you have, and of course, if you have a much longer recording for one group than the other, you are looking to the bias case. But yeah, in our case, it's very similar. We can force the periods to be always the same, and it also holds. And we can also do the longest periods we can have, or the longest periods we can have that are consistent across groups. But yeah, it always holds all these results. So they are very robust in this sense. But yeah, it's very important to check all these details, yeah. Well, this is an important detail.
So thank you, Leonardo. It was a very nice talk. So yeah, just have 3 very technical comments, but didn't get, and I couldn't follow. So the first one, already Daniele told me something, but I would like to double check. So when you do the source reconstruction from the MEC to, and then you put the AAL partition, how you, I mean, go from the very, let's say, dipole level to the, so just the average, or?
Yeah, no, that's a very important point, because in MEG, well, the deep sources are problematic, so you have to take that into account. But yeah, it's true. Well, we in our case we just had, well, did the best we could. So actually we didn't look into the regions of the cerebellum, because we thought that would be too noisy. But
basal ganglia is very deep, no?
It is. But yeah, it's just because it's an important region. But we could also remove that and see what happens. But yeah, in our case, we just trusted that this would be, well, meaningful, and we have these magnetometers that are the best way of acquiring this data. So it's more sensitive. So it's true, it does decay a lot, well, decays fast with the distance, but it's still the best way of doing this.
But I meant also for each AAL ROI.
Yeah.
How you get a single value per ROI?
Yeah, so in that case, we are looking to the centroids. So we have the centroid of the region.
So you put dipoles in the centroid of the regions, yes, and then you do reconstruction, or?
Yeah, well, yeah. So this is we do the beamformer at the centroids of the regions, yeah.
Okay.
But yeah, it's true. There are different ways of doing this.
Okay, that was one question. Another one is when you do template pattern, I really like it, the fluctuations around this pattern. How you calculate this pattern? It's just in the...
so you just combine all of the regions that were active. So here, say, for instance...
But what means combine? Union?
Yeah, because some of them are going to be... Yeah, we didn't look into the order in which they take place.
Okay.
We just
the union of all of them, no?
Yeah, that's the thing. One could go into much more detail, but that would require much more data to be confident that these results are meaningful. But it's true. One could also look into how this really takes place, but that was not our goals. We're more like, okay, these regions, well, presumably interact during this period, and without taking into account the order in which they interact, but which regions were active during this, and that's the only.
And just the last one, when you do the basal ganglia, you show in the very end that there are bigger differences between the two groups.
Yeah.
For the basal ganglia.
Yeah.
So, is this meaning? I mean, this is after multiple comparison, or you go one by one and you see that the per region activity?
Yeah.
And it's more different there.
So we can look into the distribution across groups. So if we look into the distribution across groups, they are significantly different. And then in this case, we are focusing only on specific patterns that are not common across different groups. So it's just the different types of patterns, and we look into the regions that are more present more often within these different patterns. So these non-shared patterns across the two groups.
Okay, and this is the number of times in which each region appears, something
Yeah,
like that?
So those are the ones that are much more, that appear much more often.
Very nice talk, really. Many things that really dovetail with Parkinson's disease literature, so this is really a sign of the goodness. My question would be, did you look at the dynamics of pattern repetition? So how they, say, come one after the other?
Yeah.
And whether they
Oh yeah, no, we didn't look into that, but that's a very good point. That's a very good question. That's true. Because there are many things that one can do. Yeah, look into this history dependent and interactions, and also what Jesus was suggesting, to pay attention to the order in which they appear. It's true, there are many things that can be done. This is just the first work in this direction, but it's true. Those are great suggestions.
Some people did it for microstates.
Yeah.
Something like that.
Yeah, this is just a very coarse first approach to this.
Okay. Thank you.
Do you want to use this thing here?
Good. Yes, that would be great. The size, does it help? Doesn't like Apple. No, that's rotation. Scaled. Let's try this way.
or do it the Zanin way?
The Zanin way, yeah, but Zanin has a very safe, had no animations. It was a very good idea. Haha, magic.
That's beautiful.
Thank you.
Okay, so our next speaker is a great.
It's fine, it's just this there.
Okay, who was actually able to overcome. Okay, who will most probably be able to overcome the curse of Apple? It's, let's say we don't have anything personal against Apple, it's just the local system here. No, for real. Our next speaker is Giovanni Petri from the ISI Foundation in Turin, Italy. And he'll tell us about the functional shapes of the brain. So basically, what can we learn from the brain from the viewpoint of topology? Welcome.
Thank you for being with me. Thanks, David and Onerva, for the introduction, the invitation, and everything. So let's get to this. What I'm trying to convince you over the next 25 minutes or so is that there might be a role in using some sort of topology, at least, and I'll explain what I mean by topology in the analysis of brain networks in general. Most what I will talk about is fMRI, but the tools really scale. So what I'd like you to move in your head from networks to the concept of shapes. Okay, so this TDA that you see up here stands for topological data analysis, but really think it's about shapes. And why is this important? Because the same way we do with networks, we would like to be able to say, okay, this network has a certain set of properties, a certain shape, and I'd like to be able to say that this guy, for example, is very different from this guy, and this guy is very similar, and this guy is more similar to this other guy in some way. Now, you might ask why I'm trying to sort of sell you this idea of shapes. And actually, it's not really weird, because the idea of shapes really comes back to the branch of mathematics of algebraic topology. And topology is that part of mathematics that tells you that cups, like mugs, have the same shape as a donut. Now, you might ask yourself, why do I even care about this? And the reason why you care is that typically, if you do have some topological information in the data set that you have, in the data that you're looking at, this is very robust. Exactly because the topological information is encoded in such a way that if you stretch, change the shape of your thing, it's still going to be there. Sometimes it might not be there, but if it is very robust. Also, there is something else you should think about, which is, if you go back to the beginning of graph theory, I mean, the graph theory, this is the Königsberg, the problem of the Königsberg bridges, so the idea is how you go through using the bridges only once, really is a topological problem, right? You don't care about the shape of the islands, you only care about the connectivity, so really the problem that you're solving is this one, okay? And even if you think, and this is like a simple graph theoretical problem, in a sense it's the simplest topological problem you can really think about. And this is actually something you do every day. If you think about navigating from Seven Sisters to Fulham, you really don't think about the geometry of the space, but you think about the topology of this object and how you can navigate that, okay? But why are we talking about this, and why is it even meaningful? Well, imagine that you have something that looks like this. This is your system. And if you're a network scientist, you're going to look at the number of nodes that you have, number of degrees, and where they are. But then I ask you to sort of look at this configuration, and this changes in time. If you're a network scientist, now you're at a loss, right? Because it's going to be very hard for you to pick up the differences from the statistical and with the usual tools that we have to differentiate the configuration between the closed one and the open one, okay? We can do it because we have this notion of shape, but even the notion of shape that we have sort of encoded in our brain, somehow it's a problem. For example, over here, what is the relevant shape? Is the big sphere, the little spheres? There's going to be noise in the data that you have. So how do we manage to actually quantify this kind of structures, okay? Also, if you're really interested in using these kind of pictures, go to conspiracy websites. They have the best really crazy pictures, and this is from there. This is supposed to be like the structure of the universe. Now, topology has been used over the last 10 years in a bunch of different systems, in a bunch of different applications. It was originally motivated by sensor networks, and the idea is that you can find holes. And there is a lot of work, more recent work in neuroscience. And the purpose of the next slide is just to flesh it and show you that there are many works dealing with this, and that therefore it should be interesting for you. Really, what does it mean in practice? There are two main tools, two main ideas that are defined, that are sort of in current use in terms of algebraic topology within neuroscience. The first one is persistent homology, which is the one we're going to be talking about, and really is the idea is that you start from some sort of data set and you are able to approximate it. So the data set you have to produce a series of approximations that are finer and finer, and then you have some indicators that you can extract from these guys, and then they allow you to compare different shapes and so on. And the second one, which I'm not going to talk about really, but I'm just going to mention and put it there, is something that's called mapper, and it's really about sort of extracting topological robust backbones from data set that you have. In the interest of time, I'm not going to talk about this, but it's actually pretty important, and I can point you to some of the applications later. The reason why we focus on this is because it's the one that is mostly used and also has this kind of higher order interactions that will be relevant later. So the general question is, can topology quantify shapes, whatever these shapes mean in our cases? And to do that, I need to introduce a little bit of simplicial dictionary. Now, if you are a network scientist, you are used to the dot, the vertex, and you're used to the edge. So we stop here, okay? Now, if you go up in dimension, you need to generalize this concept and go, for example, to the triangle. Now, this triangle here is full, right? It's complete. It's like it's a tile. It's different from the sum of the three edges. Now, and each of these guys is called a simplex, and then we put them together and you make a simplicial complex, which is supposed to look like a early 90s video game. And the idea is that this object is gonna approximate the data set that you have the same way you would do with a correlation network. Okay. The problem is that how do you go from the data that you have to one of these approximations? Well, the typical case is that you take points in some space and you fatten them, and then when two fat points touch, you put a link. When three fat points touch, you put a tile, and so on. Obviously, the next question is, how fat should my points be, right? Because if I take them thin or if I take them really, really fat, the separation complex that I extract is going to be very different. Now, persistent homology is called persistent because it does exactly, it twists, it sort of puts this question upside down. Instead of saying which one is the right scale, it looks at all the scales. So you produce a sequence of approximations of your data set, one after the other, and you end up with something that looks like this. You see this one is containing this one is containing this one, and so on. And then you calculate these objects, which are called barcodes. Now, barcodes are very intuitive. If you look at them, these are the connected components. You have many disconnected components, then merge together, and you only have one living through. And instead, these guys here start getting into the actual topology. So these guys here refer to the one-dimensional cycles that live through. You see that you have these holes, and these holes go through. Each of those bars lives as long as one of these cycles. And then you can go higher, for example, in 2 dimension, okay, in 3 dimension, and so on and on and on. Imagine the inside of a football, that's going to be a 3-dimensional cavity, okay? Interestingly, these objects, they are themselves a way, you can use themselves, as points in a metric space to compare shapes. The trick that you use here is that you go from these barcodes to something that looks like this. These guys are called persistent diagrams. The reason why all the points are above the diagonal is because all this each bar has a birth and a death, and typically you're first born and then you die. I don't know, Mother of Jesus Christ or something, so you're above the diagonal because this is the birth axis and this is the death diagonal. So what really happens in terms of pipeline is that you have some data set and then you extract these kind of persistence diagrams from the data that you have, and then you can measure distances between these guys, and then you have some sort of measures. There are distances, there are kernel similarities, and so on. Okay? Now, to show you that this actually can work and how it works and whether it's stable, whether we can actually use this kind of tools to make a little bit of science, I'm going to walk you through an application that we are currently running up. So we took 100 subjects with 2 recordings each. This is the sort of the shard data set, let's say, resting state. And one of the first questions we asked ourselves is, well, can we tell apart the same subjects? So if I'm recording myself twice, am I more similar from the point of view of topology than I am then I'm similar to the others? And what this plot is telling you here is exactly that you can take windows in terms of the data set, like how much data you take in terms of the correlation matrices that you build as long as you want, but with an overall for like reasonable length of the time series that you consider, most of the time you're more similar to yourself than someone else, so you're actually picking up in individual variability. This is the same result actually, just in terms of counting how many points are here, and this means that it's actually working very well. Another thing you can ask yourself is whether these measures are stable. And for what you see here again, I mean, and you have to believe me, is that down here I'm using all the data that I have, and here I'm taking windows of a certain length, of a certain width, and I'm calculating the distances. So basically these are similarities, so going up means that you're getting closer, so it means that the more data you get, the closer you converge to the final one, which is sort of obvious. But the interesting thing is that you can, for example, ask yourself, what is the minimum similarity threshold that I need between the full matrix that I have the full topology and the one obtained from windows, from shorter windows, and you can sort of see that already for 200-300 points you're pretty much well off. We reproduce it in the different sort of cases, in different recordings, it's pretty much the same. Question is, why do you want to do this? Is because you would like to know if you have a certain amount of scan time, you might want to know that you need at least 300 TRs, but you don't need more to have the actual final resting state topology that you would see, and this is actually what you find there. Interestingly, what we find is that if we look at how many bits of information we need, we get these results with logarithmically less information. This HC stands for homological connectivity, which is the topological invariance that we found. So the matrices, the function connectivity matrices that you have are always the same. Instead, this number here depends on how many dots you have in those persistent diagrams that I showed you. And these are logarithmically less, right? So you get pretty much the same results in terms of, for example, of classifying, of distinguishing whether it's you or another subject with a lot less information. And this is because what these objects are doing, these techniques are doing, is basically distilling the essential shape out of the data set that you have. Now, can we do more? Well, yes. For example, you can ask yourself, can we do something with dynamics? Now here, what you see is the classic sort of distance. So each axis is time, each dot is one time, and if you're off the diagonal, you're basically measuring the distance between the correlation matrix at time t and the distance correlation matrix t plus 1. Okay, so if you have blocks and diagonal, what you're saying is that you're staying in a similar state, and you jump to something else. Okay, naturally, this thing actually has a lot of variability across subjects. These are 3 examples just to show you what happens. But the interesting thing is that we can do the same thing in terms of homological structure that we extracted using that sequence of approximation of simplicial complex approximations that I showed you. And if you do this for the same subject up there in green and blue, you end up having something that looks like this. Now, this is an example that doesn't look particularly well, but you already see that basically what's happening here, if you're thinking about these like yellow parts as states, what's happening is that you have a bit of like a jam of clusters. You don't have clear transitions from one state to the other. Here, at least in some cases, you seem to have a little bit of a better discrimination. This can be put in numbers. For example, what you can do is that you can measure the distance, so how far you jump from here to here, from here to here, from here to here, and compare it to like a random jump. So in the blue case, the blue lines in both plots are for the homological connectivity dynamics, and for the functional connectivity dynamics, the real data. And this is ordered in such that if you have a tail in that direction, you have big jumps, okay? What you really should look at is this green one, which is if I just pick a random element of the diagonal, what this is telling you is that these jumps are typically much larger than the ones that you find here, and instead, which is basically how persistent you are, how close you are to the states where you were before. And these other distributions here instead are the same thing for a couple of standard null models, autoregressive models, phase randomization models, and so on. So what this is really telling you is that in both cases, what we're picking up is the fact that typically you have a series of small jumps, so you're staying around an attractor, and then you have a big jump. So these are actually Levy walks in a sense, okay? Instead, the random one is just picking up the jump ones, the random jumps, and so this is why it's above. And instead, these kind of autoregressive models and the phase randomization models are basically staying where they are. They are staying put all the time. Turns out that using this sort of distribution of these things, you can pick up the age of the subjects. These subjects are different age. And the interesting thing for us in terms of the use of topological dynamics is that if you plot the Earth moving distance, which is a distance between distributions, and you compare how far the blue one is to the red one here and the blue one to the red one here, it turns out that up here is systematically larger. So in the homological jumps, the distance in homology seems to be more discriminative than the one in using pure sort of functional connectivity dynamics. More interesting is that you can even do something else, which is, well, you remember, so we started from the functional connectivity. So here we have networks and we have net distances between networks, maybe using like the correlation between the upper diagonal of the matrices. It doesn't, whatever norm you use, it works. We can calculate the corresponding homologies and can calculate how the distance in the homology, so in these shapes, evolve in time. And there is something that, and then you can also build a new backbone, and I'll explain how this is built in a second later, but just imagine it as like it's called a scaffold, because the referees wouldn't have us call it backbone, but really is a topological backbone, okay? And so what you see here is that these are graph distances between graphs, these are distances between shapes, and we found a way of building, again, graphs. And then what you see is that this thing is the evolution in time, if you want, of these graphs. Interesting thing is that if you look at the correlation between these matrices, they're all very high, and they're they're much higher than you would get if you just randomize times, for example. Okay, so what this is showing you is that you can predict the evolution in time, the functional connectivity dynamics, as some called it, by using the homological information alone, or actually projecting it back to the graph some way. Okay, now. This was the pitch in the very simple case. So does it work? Yeah, so the answer seems to be yes, it seems to be working. But it's more interesting if we actually have some cases, okay? Can we detect altered states? Now, we go back to some work that we did a few years ago, and now we are extending. So we had originally just one data set. We had people that were injected with psilocybin. So psilocybin is the active principle of magic mushrooms, so it's psychedelics. And these other guys instead were given LSD. Okay, so all these guys were very, very, very high. This is data by Imperial College, the group of Robin Carroll-Harris that was mentioned earlier. So we ran the machine on this thing, and we were very disappointed in the beginning. So for the LSD, we had 3 conditions, and it's these 3 columns here, LSD 1, 2, 3. For the psilocybin, there is only 1 scan, and the pictures that you see here are all coarse topological invariants. So we count the number of holes of a certain dimension, how long they live, the length of the boundaries of this of the cycles, how big these holes were. And we counted how many there were at each possible thresholding, and so on and on and on, okay? And what you see here is nothing, okay? So there is no difference whatsoever. And this was very boring, right? Because it seemed to be saying that these kind of tools were not picking up any difference in homology, which is a pretty refined topological object, was not picking up any information here. Actually, it turns out that you need something slightly more precise to pick up the difference. You remember those persistence diagrams, those plots with the dots in the upper, so above the diagonal? They are points in another metric space. You can measure distances or you can measure similarities between them. And this is what we did. We did that instead of just taking coarse invariance. And so first look at these blue and red boxes. This is for the psilocybin, and this is and the same thing here is for the LSD. So you can measure the distance, so the similarity between people that are in placebo state or the people that took the drug. Okay? What you find for the psilocybin is that the similarity between people in the placebo is much smaller than the similarity in the group that took the drug. Okay? So if you take the drug, you're more similar to the other people that took the drug than when you are in placebo. LSD, now you don't really see the colors well, but you see that the order is different, right? It goes down, okay? So when you take LSD, you're more heterogeneous with respect to the placebo. Now, the second part is that this box here is telling you how far you are from yourself, how similar, actually, in this case, is how similar you are from yourself when you yourself take the drug, okay? Between your placebo and your drug condition. And what happens is that in the psilocybin case, you go further away, so you're less similar than in the case of LSD. So what does it mean is that when you take LSD, you move a little bit, but you move random shape space, right? When you take psilocybin, instead you do a long jump, but all subjects go in the same direction together, okay? Now, this is interesting because the original works were actually thought, the motivation to investigate some of these drugs and these psychedelics is because there is at least behavioral evidence that might be used for drug-resistant depression. Okay, so if you want to use a drug, if you want to have a drug, you'd rather have it sort of work and do the same on everybody, rather than, or at least have the least possible variance across subjects. Now, this is all very nice, and I'll show you later that actually is not completely random, but one of the questions that I usually get at this point is this really topology? Really topology? Because it seems like an awfully complicated subject for that. And this is where we have to go back to those backbones, which are called scaffolds that I showed you earlier. Now, this is from the original, the first paper we did, and it's just to show you that this network here is different from this network here. Now, I'll show you in a second how these are built, but on the left, what you have is the placebo scaffold, and on the right, you have the psilocybin scaffold, okay? So this is a lot more messy, and I'll explain why this happens in a second. Now, remember we had these simplicial complexes, so these, these video games from the 90s, okay? Now, so topology, shapes really are about the holes, right? So you want to classify where these cycles are in 1, 2x dimension. So what you do is that you select them, and then you collect all of them, because you might have multiple. Then you put them all together, and you sum them, okay? These now are cycles in a graph, so they're edges in a network, like in a normal case. If you have repeated edges, you just sum them. But actually what the sum that you do is proportional to the length of the persistence. So how long that cycle lived, that's a number, that's a scalar, and you can just sum them. No? Okay, in the end, you have a bunch of cycles. You put them all together, you weight them, you sum them, and you end up with something that looks like this, okay? Now, the problem is that this encodes in a network format the homological information that I was talking about. With more specifically, the one-dimensional topology that I was talking about, right? Because we're talking about one-dimensional cycles, not the higher order. The reason why we didn't go into the higher order is because we didn't find any in this case, but it doesn't mean that it don't exist. To be sure there is topology, we need to compare this with something that is absolutely non-topological. And the best measure to do this, or at least one of the best candidates, is curvature. Okay? Curvature is absolutely a local concept. So the point is, can we find correlations between them? And what happens if we measure this? This Ricci-Foreman curvature has been reintroduced for networks recently. So we cranked the machine, we calculated, and we ended up with something that looks like this. 3 conditions for the LSD, 1 condition for the psilocybin, blue is placebo, green is drug. This is for the edges, this is for the nodes, because you just sum the local the curvature on all the edges, okay? So what you see here is that blue is below green, and in the LSD, so it means that the placebo is less hyperbolic than the drug, okay? The problem is if you look at the psilocybin, the effect is the opposite, right? So this sounds a lot like what we find in the other case. And we're very scared, right? Because it seems that psilocybin is making you more hyperbolic, which is actually related to navigability. But luckily, these values here are values on the edges. So we can actually correlate the values of curvature that we found for these graphs with the values of the scaffolds, or the weights of the scaffolds that we found before. And the beautiful thing is it was exactly no correlation whatsoever. So this is perfect for us, because actually what it means is that we are picking up two different effects. One is sort of mesoscale or global in origin, which is the topological part, and then there is also some local modulation, which is probably which is actually related to what has been already observed before, and that's the geometric part, the curvature part. Turns out that you can actually go a little bit further down this rabbit hole, and you can ask yourself, well, okay, now I have a graph, a new graph, for myself in placebo and for myself in under the drug. So I can ask myself, how much does a single edge change? And what you find if you basically plot here the weight of your edge in placebo and here the weight of the edge in the scaffold for when you have the drug, in the case of the LSD, you find some positive correlations. So there is something that is conserved. While in the case of the psilocybin, basically you have no local conservation, so the edges will change, which actually it's consistent with what I told you before, right? Because in the LSD, you change a little bit randomly, but you change a little bit around. While instead, when you move from one state to the other, well, from the placebo to the psilocybin state, in the case of psilocybin, just jump quickly in one direction, right? So you disrupt the local information. Now, I have a draft that I can show you, but we don't have a preprint out yet because it took some time to do this, and in the meantime, Robin actually has MDMA and DMT coming up, so at this point we're basically putting it all together to see whether this is a consistent result, or is a consistent, yes, I would say result. I was very suspicious of those results in the beginning because, you know, you have one case, you have two drugs, so it's not really clear what's going on. But it seems to be holding. I mean, so we have this evidence that I showed you in terms of, you know, this effect, this different effect at the global level, at the mesoscale level between the two drugs. Although this goes against the narrative that in psychedelic literature has been going on for a while. That basically all these drugs pretty much do the same. And actually, what we're showing here is that they do the same at the local level, but they don't do the same at the mesoscopic, at the sort of the tower of correlation level. There is no particular localization. These are different regions that we can select, you know, macro regions in the brain. But the interesting thing, and here you have to take my word for this because I have only these plots and they're ugly, is that the same thing happens when, the same thing as with psilocybin happens when we look at patients with bipolar syndrome that were treated or non-treated with lithium. In particular, what we find is that bipolar patients that were treated with lithium have a topological structure that is much closer to the resting state of the healthy controlled, but also them as a group, are much more homogeneous. So effectively what it means is that the drug is pushing them in the right direction, but is also sort of forcing them in a smaller set of, so they are less variable. This is actually very interesting because it would be, yes, it would be interesting to see whether the repertoire changes. These two things are not mutually exclusive, actually, because this is about the correlation structure rather than the phase space volume. And so this is interesting, and actually we pretty much can do the same thing in, for example, in tasks, in cognitive tasks. This is a paper that is out, I can point you to later, and what we found here is that using the same tools, you can pick up the difference between, in a task, in an experiment where you are asked to imagine both a kinesthetic and a visual, to perform a kinesthetic and visual task. You can pick up the differences between basal and task condition and between imagined task and real task conditions, and how much you restructure functionally from the basal to the task corresponds to the effort that you report subjectively, and how well and your self-reported vividness of the imagination actually correlates with how close you go to the actual configuration in space. So this was beautiful, and it was actually super surprising because the pipeline was extremely simple. Okay, so question, does it generalize? It seems to be yes for the moment. Naturally, there is a lot of more work to do, but let me just, before I use 2 minutes to go to tell you what's next, what we did here, what we are able to do is homological fingerprinting. Instead of doing functional network fingerprinting pretty well, we are able to track the evolution, and we can detect states. Now, this doesn't mean that we can detect all states, but this shows that at least in some of these cases where we have reason to believe that there are mesoscale reconfiguration of the functional network, we are able to pick this up, or at least this is a good tool, it's a good candidate for picking that up, okay? If you have changes that are very specific, very localized, this is not a good tool. You can say it up front. Now, what's next? Well, there's one interesting thing here is that you remember here when I told you that I have these graphs in time and their distances and these other graphs, these scaffolds in time, and this thing correlates to this other guy. What I can do is I can actually calculate, compute communities, temporal communities through time here and through time there. And the interesting bit is that despite the correlation between the dynamical evolution is very high, the correlation between the cluster structure is actually basically non-existent, okay? So this means that the scaffolds are able to track, to have the same type of evolution in time as the full matrix without having the same community information. Now, this seems to be going against most of what we know about, modules and so on. Actually, I don't think that is exactly that. I think what is happening is that since if you look at a shape with a bunch of holes, imagine Gruyere, if you actually focus on the dynamics of the whole, it's still a much sparser representation, and it's able to tell you how the large-scale structure of the system evolves in time. You don't need all the blobs, okay? Now, there is something else which is interesting I think, for the future, and this is an example, which is what do we really mean by higher order, okay? There is a lot of talk about higher order, and really higher order can mean many different things. In this case, what we try to do is we try to generalize the spreading process where you can be infected by just a neighbor to the case where you can be infected by the two neighbors through the edges, but also you can have compound effect through the triangle. So if you have this triangle, you have an extra term here, okay? Now, the interesting thing is that even in very simple cases, this is an SIS model, so just like susceptible infected susceptible model, the introduction of these higher order terms create a bistable regime, and you have discontinuous transitions and so on. And as far as I know, there is very little work done on more generic and more general dynamical systems, and that would be interesting. First of all, to be able to measure, because we don't even know whether these effects happen, and also at that point to model. I mean, for example, not exactly this, but similar modeling frameworks are relevant in ecology, for example, when you have the interaction between two species be mediated by a third. Naturally, that's what I already said. I mean, there's this problem about predicting simple complexes, and for example, I don't know if you're familiar, but there is some recent work about embedding structural connectomes and using those to predict functional connectivity. If you envision the possibility of having higher order interactions, you need to be able to do that. And that actually means that instead of going node to vec, you need to do simplex to vec, and there are ways of doing that. And we propose one, we're doing the prediction, seems to be working well, but there is, a lot of open space up there. But really the real sort of money problem is this one. So if I have a network, there are many simple complexes that might be compatible with the same network structure. So one question is, how can I infer the most likely structure depending on the data that I have? For example, we're doing this in a Bayesian framework with Tiago Peixoto, and we have some nice results, but for example, the type of framework we have does not lend itself to be used, for example, in this, if you have time series to begin with, right? So this is an open problem. I'd like to talk to anybody that is interested in this. And finally, if you were interested at all, this is a shameless plug, take a look at this editorial, especially at these articles. This is a focus feature on network neuroscience that came out in May or June, I don't remember exactly in the end. And there are many different applications of these ideas, from the macro scale to the micro scales, going through data analysis and modeling. So this is a good reference for you to go there. And thank you for your attention. This works.
Thanks, Giovanni, for this very interesting talk. I have it's more than a question, it's a curiosity. Are you aware about the null models? Is there any null model oriented, for instance, to assess the significance of the homological fingerprint?
Super good question. The answer is maybe. There are some random models. We developed one configuration model to exactly for that reason. Because when you have groups, at least you can say, well, I have got groups. There are some not old, but sort of previous works whose attempt is basically to put when you have the persistence diagrams. Let me see if I can get them here. It's probably so. The conventional wisdom is that when you have, Jesus Christ, sorry, is that when you have stuff that is close to the diagonal, they're blips, right? They're noise. So the conventional wisdom is that you should not really look at them and you should neglect them. And so what you do is that you can sort of bootstrap the analysis of a band here and say, okay, everything that is inside the band doesn't count. Thing is that depending, I mean, the description I gave you works because if you use Pearson correlations, those are ametric, so that can kind of work. But for example, you could create that long sequence of simplicial complexes from anything, and sometimes small changes make a lot of sense that are important. Just to give you an example, if you do the same thing with a weighted network of passengers on the airport, transportation network, and you run this thing by thresholding for each weight and then constructing the simplicial complex accordingly, you find a bunch of things that are on the diagonal. Okay? Then you take the configuration model for networks. Those are networks, so you can do it. And you find that everything leaves all the way to the end. Okay? So what it's telling you is that in the original system, there is a lot of ordering between the weights that you're losing when you create a configuration model, even the state-of-the-art ones. Okay? So in that case, yes, they are short-lived, but they are important. So depending on the applications that you have, as always, the null model needs to go along. To go back to the original question, there are very few ones. There is a paper by Danny Bassett in which they don't really do, and Sizemore is the first author, they don't really do null models, but they do generative weighted networks, and they classify them according to this. It's an attempt. The problem is that homology in itself is very tough to go through. I mean, it's a rigid box. We're working on that from the point of view of linking the spectrum of the original matrix. If you have the precision homology, so you can work behind. But there are a few. Typically, what you do is that you create a lot and you just do it again.
Thank you.
For the moment, at least.
I guess we can pass it over here.
Thank you, Giovanni, for the presentation. I like very much the simplicial approach. I think that perhaps the next step, one of the next objective would be to give a meaning to the simplicial that you have, at least for a network neuroscience. I mean, to get really the impact on the community. I feel that we miss something on that part, or maybe you have something that you can explain me. But if you, for example, just you take into account the node degree, you make the comparison between the node degree. You know that there is the number of connections of each node, so you have a difference in the way that region is connected to all the others. So you have a neurophysiological, let's say, meaning, a biological meaning. Now, for the simplicial, for me, it's harder to find this explanation and then to...
I've got two answers to that.
Yeah.
The first one is, yes. If you think about, for example, I did not show you any localization in particular, right? And that's because, so those correlation matrices that one usually looks at, right? They are basically metric spaces, okay? So what you're looking, when you take a certain degree, is really choosing the distance, the scale at which you're looking at the system, right? Because you're deciding who you're attached to. So if you're looking at the shape of this space, you need to make a choice. What this does is that looks at all possible choices. So you lose the kind of, this is highly connected with respect to the other one. But what you can, for example, do here is to see when you start being involved in some of the topological features early on, and through. So that's one. And I forgot. And the second one actually it's constructive. So one of the things that we lack for the moment, and we have ideas, and actually today there were some contributions that might solve the problem, is what if instead of building it this way, I actually build directly a weighted simplicial complex. So I calculate the weight for the edges with the usual correlation, suppose, and then I have a 3-body correlation, and a 4-body correlation, and a 5-body correlation. At that point, you just define the strength of your node in your 3 strength, 4 strength, just by summing as you would normally do. So I think some of for example, the ideas that Mario was mentioning earlier might work. Maybe some information theoretic approaches there. We were discussing with Tommaso Gili about using some old StatMechie, like your cell functions, that kind of stuff. And that would be constructive, right? Because here what we do is that we take the topological data analysis perspective. So we have a space, and from the space we build a simplicial complex, right? And you can do this without a space if you have a weight that you put on the simplices themselves. And I think that would be a lot more meaningful in the direction in what in the direction you're saying. If you talk to people, they don't think that their correlation matrices are actually metric spaces. They think they're networks, and they have to threshold them, right? It's actually not always the truth, depending on the goggles you put on. But having that, what I just said, would help with this, exactly with this. And would help also with the issues at the end, right? Because you will know where you have the tree interaction, where you don't, whether it's trivial or not. So yes.
Well, I'm sorry, but I think I need to cut the discussion here. Giovanni will be around, so you can ask more questions during the coffee break or whatever.
Thank you very much.
I'm sorry for that, but I think we need to move on to the last speaker of this session. And so, the last speaker of the session. And he's the one who is actually working on multi-layer networks, so everybody who has so far told that somebody will later explain what are these multi-layer networks. Now Mikko is the one who is actually going to explain. And he has been the first author of that famous multi-layer network paper that is called Multi-Layer Networks. And his talk today is also called Multi-Layer Networks. You know, Finns are really straightforward people, no fancy titles. But it's great to have you around, and welcome.
Thanks. Yeah, I'll maybe use this one. So I'm going to talk about multi-layer networks. And this is going to be sort of like a generic talk on multi-layer networks, and not so much about the brains. In fact, I think in the next slide, this is probably the only picture I have on the brains. And so basically, yeah. So the starting point here is that in, like, I'm a network scientist. I have been working on many kinds of systems and analyzing them as networks. So here's just like a few examples of them, and of course the brain is one of them. But you can think of other things like social systems or ecological systems, or even the sort of the regulatory systems inside the cells. And the nice thing about this is that you can represent sort of the complexity in all of these as networks. So you have some sort of agents, so some like brain regions, although now we know that we shouldn't trust on them, and then some sort of links between them. And okay, we again know from today that we shouldn't maybe trust the links either. But anyway, you can do this for the same thing for all these different kind of complex systems where you have some sort of nice, interesting emergent behavior and so on. And then you can analyze them as networks, which are basically just graphs, so you have nodes and links. So the nice thing is that you can use the same tools, same ideas. So many of the things that we have mentioned today, like the clustering coefficient, I don't know, the small mode phenomena, all of these, you can analyze the same concepts and tools in all of these systems. And this has sort of been a success story of this network science I would say, since last 20 years. So why do we need multi-layer networks then? So if networks are so amazing and they are everywhere. So here I would like to think in terms of social networks, because those are much simpler to understand than, say, brains, because we are all sort of familiar with the basic concepts and there's not so much confusion. For example, we all have a brain, but it's, I don't know, it's you are not so much aware of your brain as you are of other people, maybe. And it's also more simple to define, say, what is a node in a social network. That's like a single person. You don't have to sort of think about these sort of questions. But even in these social networks where things are supposedly more simple, there's a problem that if you take a social system like this,
and you represent it as a graph like this, then you're clearly losing something. So you don't just have people and connections between them. But say in the social system, you could have something like the connections could change. There's different types of connections, there are different types of people, there's different social contexts, and so on. So it's not only that you can represent things as simple graphs like this with nodes and links. And actually, this is not like something that is very new if you think about analyzing networks. So these are pictures from the 1930s of social networks that they constructed back then, and these days still called sociograms. And here is just a single social system inside a factory where they have different types of workers, and then you have different types of relationships between these workers. So already here in this early analysis, people saw that, okay, it's not enough just to represent this as a single graph. And of course, when this social network literature developed, this is from a book from 1990s, this had already developed to much further. So here they call these kind of things as multivariate social networks, and they had already sort of tools to analyze these. They were even, in social networks, they were even sort of very complicated kind of structures or representations. And this is from a social network where you have two types of relationships, advice and friendship relationship, and then for each person in the social network, they also have an internal representation of that social network. So you actually have, because you have 21 people in the network, you have 42 different networks, just sort of giving a picture of the same system from different perspectives. Then there's, of course, things like temporal networks. So this is a picture of social network where you have recorded mobile phone calling between people. So the red arrows flashing there are mobile phone calls between the nodes. And there's a time running, and this makes a huge difference for example, for the dynamics on top of the networks. Moving away from social networks, you can think about infrastructure networks. This is a sort of from an influential paper from around 10 years ago, where they showed that this kind of connectivity problems in infrastructure networks really need this sort of perspective of having multiple networks of networks of networks. And the idea here is that they showed how to simulate these blackouts here in Italy, so that you have a network of power stations and networks of computers that control the power stations. And so you have two networks, and those networks are interconnected or interdependent. So if a power station fails, then some of the computers fail, and this means that more of the computers can fail and more of the power stations fails. So in order to understand this system, you need to understand how these sort of systems of networks work. You can also think about other types of infrastructure networks. So here's just a picture of networks that are used in England to predict sort of how are the damages that, for example, flooding could cause spread using the different networks that are interconnected. So basically, this is sort of the idea that I wanted to give you here is that there's many fields where there was this kind of need to come up with something more than just a simple graph representation in order to model many of the real phenomena that is out there. And this was sort of the starting point of this review article that we wrote that is already now 5 years old. So there's actually a lot of development that has happened since then already. But I'm just now going to go through the sort of the basics that we have here, and we can discuss maybe later on more of the recent developments. So the idea that we had is that back in that day, like 5 years ago, the situation was pretty much like this, that we had all these different network concepts that people were inventing in different fields. So all these application fields, they had noticed that, okay, they need something more. So they came up with different kinds of ways of generalizing graphs. And okay, there were many of those, and here's just a few of them. And the idea is that we could somehow maybe develop something that could be used to represent all of these. So we could talk between the fields and use the technology and different measures from the different fields to analyze them. And this is what is called multilayer networks. And here I'm just going to show you what we did in that review article. How did we define multilayer networks? There's some other definitions out there also. So here's the sort of the most general kind of multilayer network that we needed to do this, to get all these different representations from the literature. So basically the only thing we added is this sort of layers. So in addition to nodes and links, we had layers. And so yeah, you have nodes, you have links between them, and then you have these like boxes here that are the layers. But there's few things you should notice here. So first of all, the same node, for example, node 1 is in multiple layers. So that can happen here in this definition. The node can be connected to itself across the layers, or actually to any other node layer pair that you have there. Then what we also needed to have is that you have this sort of like multiple dimensions or aspects, we call them, for the layers. This is actually not, usually you only need one of the aspects, but for some cases you would need more. Okay, so you can also formalize this in the interest of time, because the coffee break is actually over already, so according to the schedule. So I'm just gonna skip this, but you can just define this. There's nothing too difficult there, just complicated. And now that you have this sort of thing, what you can do is that you can now start mapping things to this multilayer network framework. So for example, you could take something like that has been used in the transportation networks. You have different cities, and then you have different modes of transportations. So maybe you would have been thinking of this as this sort of networks where you have multiple edges, and each of the edges could have a color in them. So this kind of multicolored, multi-edge colored graph. And you could do that, and you could map it to this multilayer network formalism in a way that for example, that each of the modes of transportation can become a layer, and then you just connect the cities inside the layer in a way that if there's a connection in that mode, then you connect them inside the layer, and then in order to represent the fact that you can transport between the different modes, you also connect the node to across the layers. So you can change from the airline to railways, for example, inside the city. Okay, and you can do this. I mean, it's not very difficult, and actually you can then represent many of these real systems from different fields as multilayer networks like this. So for example, here's a social network where you have an account on different social networking websites, and then you're connected to other people inside the website, but also you can sort of connect yourself across the different social networking sites, and to represent the sort of flow of information, say, between the social networking sites and inside the networking sites. Okay? And this is just a picture of the UK transportation network built with the same idea. Okay, so now you can do this. So you can map different types of networks. So this is sometimes called multiplex networks, and that I just explained. And you can map it to this framework of multilayer networks. And now, of course, this is somehow limited the mapping. So in a way that not all the different kind of multilayer networks correspond to some kind of multiplex network. So if you take a point, some network in multilayer networks, you probably cannot find any multiplex network that would map into that. So it's in that terms, these multiplayer networks are sort of like a more narrow term, or like a multilayer network is a generalization of multiplex networks. Okay? Now what you can do, you can take these different concepts that people have defined in other fields or for other kinds of systems. So for example, node-colored networks. There might be a few different ways of doing this transformation sometimes, but usually there's only one, or at least some clear one. And then this is something that you get. So some of the things that you map into the to multi-layer networks, for example, multiplex networks and multi-relational networks, actually will result in the same set of multi-layer networks, and these concepts are somehow then equivalent. Other times they might be somehow overlapping, but different concepts. And now what we did in the review article was that so basically went through literature on different kinds of concepts. And this is the result on this table. Each row in this table is one concept. And there's 40 rows in this table, so there was quite a lot of things. And now the ticks here correspond to the different features of multi-layer networks that you have. So, so basically, the row of ticks here defines the set of multi-layer networks that you have had in the previous slide. So if you give me the exact set of ticks here, I will tell you what is the set of multi-layer networks that this concept can span, right? So put it in another way, if the set of ticks here is the same, then the concept is exactly equivalent in this transformation. And now there's something, so okay, so we can do this, and you can see that there's something that are the same and others that are just similar. And in fact, if you look a bit closer, you can find two sort of main categories here. So one is this something that now is often called multiplex networks, and another one is that is sometimes called networks of networks, or if you don't know these terms, then maybe these are just slightly easier names. So this box here is networks that have colored edges, and this is a box of networks that have colored nodes. But of course, there's like a slight differences in many of the definitions. So this is not so clear, but this is sort of the main two categories. Okay. So now that you have this multi-layer network structure, what do you do with that? So how do you analyze this? Because I mean, this is in the end what we want to do. It's nice that we can represent things in the same way, and we can talk about different like network generalizations from different fields and from different people. But in the end, you need to analyze them somehow. And what is sort of the new thing that this will bring to you? So, especially back then in the literature, what you could easily find is that people often constructed these networks. And what they then did is somehow aggregated the data. So you had this amazing multi-layer network data, and then you just aggregate it because you don't know how to deal with the full multi-layer network thing. So and then you analyze the aggregated network. Now things have changed a bit, so there's many more multi-layer network methods out there, so that will actually take into account the full structure, but still. Yeah, this aggregation is quite popular, and sometimes you even do it sort of implicitly, so that effectively your method or measures that you're doing are working on the aggregated network. Okay, yeah, so here's some of the things that have been done. So mostly especially in the beginning, what people have been doing is generalizing different methods and tools that are out there. So take you decrease clustering coefficients, centrality measures, community detection methods, everything has been sort of done in terms of generalizing. Often this has been done in an ad hoc way. If you, for example, think about the clustering coefficient that, again, I think you are using, then even for this multiplex type of networks, this has been generalized in many different ways. And at the time, I was able to find at least 6 different definitions of the clustering coefficient for the same type of networks. They were, of course, from different fields, so one could be from social networks and one from transportation and so on. And often they were done in a sort of ad hoc way that you were just they were just staring at the formula of the clustering coefficient and then somehow adding stuff to it, adding the multi-layer network things to it. What I think would be a nice way if you want to go through this generalization pathway would be to start from the sort of the first principles of the tools or methods that you are using. And I'm maybe giving you a few examples of these. So one of the things that you can do is looking at the linear algebra. And this has been a very powerful tool in network analysis. So what you do normally for graphs is that you represent them as adjacency matrices. And then if you calculate stuff for the adjacency matrices, these will tell you things about the structure of the graph or on the dynamics on the graph or so on. For multilayer networks, the similar concepts are either the adjacency tensor, which is sort of multilinear construct, or like a generalization of a matrix to higher dimensions, or something that is called the supra-adjacency matrix, which is basically flattening of this adjacency tensor. And then you get nice things like you can directly use, for example, tensor decomposition methods, or some of the centrality measures you can get out of this. And there's, for example, from the tensor decomposition literature, you get to already see what is sort of the fundamental difference between these normal graphs and multilayer networks, because they have, for example, multiple ways of generalizing tensor decomposition, matrix decomposition methods to tensor decomposition methods. So there's this added degree of freedom. It just gives you different options and different ways of looking at the data. Yeah, so here's just a sort of a representation of this, like what I mean by that, the fact that the adjacency tensor is just a tensor flattening. So if you would take a multi-layer network and forget about the layers, so that each of these sort of node layer tuples would become a node, and then you just add the link and take adjacency matrix of that is the supra-adjacency matrix. Or if you take the adjacency tensor and flatten it, that's the same thing. Okay. Another way of starting from this first principle is to look at the walks and paths. So these are often the starting points for many tools. So if you think about cascading failures, modularity, so many centrality measures, clustering coefficients, these can all be defined in terms of walks or paths. And so what you can do then is that instead of having like a single type of walk, you can define walks in multi-layer networks. Depending on what is exactly your system, this will give you the sort of inspire the definition, what do you mean by a walk? And then once you have defined this walk, which can be or a path, then you can just plug it in to these definitions and get out something that is more like it gives you or you put in the definition of the walk and you get out the measure that you want. Okay. Yeah, I normally give an example of this clustering coefficient, but I think I will now skip over that. Instead of, I will talk a bit more about the graph isomorphism. So again, this is a very simple concept in graphs, and which is underlying many methods that people are using to analyze networks. So basically, it just says that, or tells you which graphs are structurally equivalent. So here, for example, these two graphs are the same, even if you add these close to the nodes, because you can just remap the node labels in a way that one becomes the other, right? In multi-layer networks, this becomes slightly more complicated because you now also have, in addition to remapping the node names, you can remap the layer names. Or you have to decide if in your system it makes sense to remap the layer names. So for example, if you would construct a multi-layer network where you would have, say, these brain networks of, so maybe in this case, you would want to remap the layer names because you want to think that these are all sort of the same. But if you would have something like the structural network, brain network, and functional brain network, maybe you don't want to remap these because you want to keep the identity of these two networks, right? So it's inspired by the sort of problem that you have in hand. And once you have these, you can immediately almost get all of these different kinds of analysis tools that you have. Of course, for example, for motives, you would also, in addition, need to have some sort of null model for your network. Yeah, here's just a teaser on something that we are doing related to other talks, with one by Onerva and one by Iiro, where we looked at the this kind of multi-layer network motifs in Iiro's data, and using the technique that Onerva explained earlier. And yeah, I'm not going to go too much into this detail here, just show you that this is the kind of things that we are working on now. Yeah, here's if you want to work on multi-layer networks and don't want to code your own tools, which is, I wouldn't recommend it, then you can use, for example, this tool that I have developed for analyzing multi-layer networks. There's also other tools out there, but yeah, this is a Python. It's integrated with NetworkX, so you can use all the monoplex stuff from there, so all the like normal network things from there, and then it has some additional things. You can also draw this sort of nice pictures if you want. Okay, and this is sort of the summary here. So the literature on these things, this kind of more generalized concept, has been quite messy, but there has been actually quite a lot of work in very different fields, and I think this is because there's a need to go beyond graphs. Hopefully it has been getting better, so people are maybe now talking about multi-layer networks, or at least using the same kind of words, for example, multiplex networks, to talk about this specific type of network. So you no longer hear things like overlay networks, which is basically a multiplex network. Yeah, and if you want to work with these generalizations, then you probably want to start from these fundamental concepts and work your way to more complex things. Okay, yeah, thanks.
Thank you. Fascinating. So you mentioned, of course, many things, but I guess there are also concepts for directed graphs in multi-layered networks, because you can imagine two layers, but if there are two processes occurring in this node and in this, that's not symmetric interaction, not necessarily. So there are also, I assume, tools for directed graphs, right?
Yeah. So I didn't mention that's very trivial to have directed multi-layer networks. Yes. So you just have direction on the links. Yeah.
Yeah, no, just at the end, you're talking about the motifs. And I wonder if you can explain a bit further, just because I couldn't understand what you were doing there.
Yeah, so this was just a reference to the previous talk. So there was this way that Onerva constructed the multi-layer networks. Do you remember this? Yes. From the earlier talks? So there was basically one time slice was one layer.
Motifs you mean by you counting the building blocks of this network?
Yeah, so then we count this kind of...
in different nodes, or?
Yeah, so then we count, for example, this kind of like a isomorphism classes, so the same way you would do in normal networks. And then, of course, you need to have some null model or somewhere to compare to, right? So you just count how many of these building blocks there are. Okay.
Okay.
A little bit crazy question, but can you actually infer from, even if it's one layer network, can you infer a necessity or presence of another layer, which is required? Because you analyze it, and then you think, no.
Yeah.
That requires another layer. I'm just curious whether we can infer something, the existence of higher levels. From
Yeah, so if you just observe a single, there has been some papers on this. This, of course, you need to have some sort of model or understanding, like what is, like you in general you cannot, of course, do it. But if you have some sort of idea what could have produced it, or what kind of multi-layer network model could have produced the data, and then you only observe the single layer network, then you can try to do the inverse problem. And there has been some things like this in the literature. Yeah.
Okay, thank you.
Yeah, thanks.
And let's now have a break of around 15 minutes, and then let's really try to get back.
It's very interesting meeting, and I want to also to thank the speakers of the morning session, Onerba, Massimiliano, and Fabrizio, who introduced many aspects of this kind of system of the fMRI data that I will describe in this talk. So, does it work? No. Yes. I will, the summary is very brief, and so basically I will treat mainly with magnetic resonance data, in which I was involved initially for structural problems about brain, not functional. And on the functional data of magnetic resonance, I will introduce a sort of a percolation process in order to solve an important problem, to tackle the important problem of the existence and meaning of intrinsic thresholds in correlation matrices in order to detect connectivity structures and topological structures. And then I will also present a brief study of application of the concept of maximum spanning tree and forest in order to study the branching or chain-like structure of the most functionally connected areas of the brain. And then I will apply this kind of framework to the distinction between healthy and schizophrenic patients at resting conditions, and then I will give some conclusion. So, but let me start with a brief historical introduction that maybe most of you will know. But anyway, that's interesting, you know, because the real first people talking about functions of different regions of the brain were the phrenologists. Phrenology was a pseudoscience starting at the end of the 18th century and lasted for more than 1 century, and basically concluded when the detection of the electrical activity of the brain and the isolation of the neurons changed page basically in the history of the study of the brain. But anyway, that's interesting. So I don't want to enter the details. To give a look to the original books is very interesting. But anyway, so starting from Franz Joseph Gall, this bunch of people divided the brain basically in a certain number of organs, regions, or we now call regions, but they were organs, and each organ was in charge of a particular task. Not only usual brain tasks that now we study in our researches, but even more refined, for example, emotional, intellectual features of for example, the murder organ. So if you have well-developed murder organ, I hope you are not in this room now. But anyway, and it lasted very long. So one of the most important books was at the beginning of the 20th century. And they were convinced that it was a scientific theory, and that because they repeated thousands and thousands of observations, millions and millions. And at the end, they collected very many predictions. For example, in this book of this Bode's Practical Character Reader, you can read about all the regions, about practicality, persistence, self-confidence, not far from what we do now, but then with strange application that, like, if you have a bump here, you're a genuine father, and many others, for example, a genuine husband, an unreliable husband, danger, it's Al Capone, clearly, you can see immediately. And moral lady, and then spiritual individuals against the material one, and etc., etc. So now we laugh about that, but that was the beginning of the functional study of the brain in different regions. And so there are many issues and many problems behind this first approach. But anyway, I think that the most important error they did is that they completely underestimated the importance of connectivity between different regions. They were, for the phrenologists, separated organs, and each organ worked by itself. So that was, I think, the big step we did with this machine, and the previous one with the EEG and the MEG. Well, with this machine is to study the connectivity, the functional, the structure, and then the functional connectivity between different. So every one of you know how it works, the magnetic resonance works, but basically there is a parcellation of the brain voxels that are about 100,000 up to 1 million, so all over the brain, including the white matter, and in the cortex between 10,000 and 100,000. And in each, in a parallel way, in each of these voxels, you perform different measurements. You can measure chemical species, abundance of chemical species, spectroscopy, or oxygenated hemoglobin, and etc., etc. Okay. So there are many, many applications, diffusion of water. So that, but clearly, for what concerns the functional activity, you can study in time the density of oxygenated hemoglobin. So with the Bolt signal. So every 1 or 2 seconds, you measure in a parallel way in all voxels what's the density of this oxygenated hemoglobin. With the other kind of measures, you can reconstruct images of the brain or the connectome when you study the diffusion of water in each voxel. Okay, in our case, instead of for functional, you basically are able to determine for each voxel a time series, and then you can apply different statistical tools to answer the question: how similar are two different time series related to different regions of the brain? There are many, many tools, but clearly, because the dynamics in fMRI is very slow, you have a measure every 1, 2 seconds, 3 seconds. So the correlation matrix is by far the most used statistical tool. And I will do the same, in particular resting condition. So basically what you do, we collect in each voxel at the time series of the density of oxygenated hemoglobin, and we relate the density to the activity, even though there are discussions about that because it's not a direct measure of consumption. And then we introduce the simplest statistical tool to say how much these two time series are similar. And in particular, we introduce the covariance, and then from the covariance, the Pearson coefficient correlation, when the correlation coefficients are between -1 and 1. And you can see these correlation coefficients between, clearly, this can be done at voxel levels, but as I was saying this morning during Onerva's talk, it's completely inconvenient to study too many time series. The discrete time series are short, because if the number of time series exceeds the length of the single time series, by just combinatorial effects, you create a lot of spurious correlations. So you cannot disentangle the real correlations from the spurious ones. So in order to avoid this problem, we also collected and clustered. We operate first algorithm of clusterization by reducing the time series to larger region of interest, in particular the one of the AAL atlas, anatomical atlas, so in which you have 116 different regions in the brain, including the limbic system and the cerebellum. Okay, so the cerebellum is that part there. Okay, then you can see, so now you have 116 different nodes in your network, and each pair of these can be seen as linked by a weighted edge. And the weight is the correlation. If you take the absolute, in particular the absolute, for the first approach, you disregard the sign, just look at the absolute value of the correlation, you can see as a completely connected network, weighted network, and with weights between 0 and 1. So this is typically the beginning of the story of most of articles in fMRI. And then the next step is let's visualize and study the clustering. Let's see if it is possible to cluster these 116 regions in subgroups which operate in a more coordinated way than with others. So the first is but in order to binarize, the first step you have to do is to binarize the network. But in order to reach this binarization, it is necessary to introduce a threshold R between 0 and 1, and putting to 0 all the edges, so cancel all the edges with correlation, modulus of the correlation smaller than R, and putting to 1, so existing binary link in the other case. The first problem we face is what is the threshold? What's the meaning? Because clearly, also choosing different thresholds put the question, what's the meaning of these thresholds and why you choose this set of thresholds and not other ones? Okay, this is, so we started our study with the fMRI of 40 different healthy individuals. And so that's the top row of pictures. So this is the average, the arithmetic average of the correlation matrix. This is the variance, and this is the coefficient of variation. So basically the standard deviation divided by the mean. So that's, and these are instead for the second group, which is 44 schizophrenic patients. Okay, so that's the problem. How to choose this threshold? So that's and once you have chosen this threshold, typically you apply all the machinery of binary graphs. First of all, modularity detection and clusterization, community detection. Okay, so a first problem is in this modularity, the choice of the modularity function. You can show that for correlation. So this morning also I said I don't remember to Benvenuto Massimiliano. One of the problems with the correlation matrix is that it is subjected to a strong non-local constraint. It's definite, semi-definite, semi-positive definite. So all the eigenvalues have to be larger or equal to 0. So this imposes to the structure of the matrix a strong constraint. Okay, so using for the null model in the modularity function, the usual configuration model is completely wrong, because the configuration model doesn't determine a correlation matrix. So this point was faced in this nice article by McMahon and Garlaschelli some years ago, in which they found the null model for the correlation matrix, the right one, okay, which is related to the study of random matrix theory. But anyway, I don't face this, so I don't face this, and instead, anyway, if you instead of binarizing, you apply the directly community detection on the correlation matrix a la McMahon and Garlaschelli, at the end, you end up with just 3 communities. So it's not very informative. Clearly, you can apply in a telescopic way to each family, but anyway, when to stop or not is the problem. So we decided another route, and in particular, coming from percolation theory in statistical physics, that I studied for many years, we started to think of how to apply the percolation algorithm to the correlation matrix, fMRI correlation matrix. So percolation, for those of you that have never worked in, is very simple. Imagine to have all a lattice where initially all the cells are white. Then you choose a probability P and you color in black randomly each one with this probability. So at the end you have more or less this fraction of black dots in. Then you increase the probability. If you take this probability too large, at the end you have almost everything black and some small white lake. In the middle, and so the opposite situation in this, in the middle there is a situation in which you have percolation, in which basically you cannot find a path from one side to the other, which is just done over following a continuous path over white cells. That's percolation transition, and that in the lattice. Now we decided to generalize this algorithm to the correlation matrix where we take the absolute values of the correlation matrix. So we have numbers between 0 and 1 for each pair of regions of interest in the brain. Then we choose a threshold between 0 and 1, R, and we cancel, as usually done, cancel the number, all the links that are weaker than this threshold. Okay, and then instead of choosing some, we taking into account all the possible thresholds, and we see what happens to the number of connected components of the network. Okay, so at the beginning you have just one single connected component. Then you choose this threshold, and then this, always one. At a certain moment, there is a first rupture and you have 2. Then you go on, and then 3, etc., etc., etc. And then correlation pairs. Then you reduce this threshold, taking only P8 is the first threshold, intrinsic one. Then this is the second, decreasing, and you integrate up to the complete integration of the brain. And you can study how the hierarchy of configuration appears, a hierarchy of telescopic clusters of the brain appears without choosing from outside a particular threshold, a discrete one. Okay, the second algorithm is instead chosen, but we decided to modify it slightly, the algorithm of the maximum spanning tree, in which basically that can be reformulated in a very simple way. For each region of interest, you look only for the other region of interest with the maximum correlation. Okay, clearly it's a directed relation. Clearly I can be most friend with David, but David can be most friend with etc. So it's a directed network. But anyway, if you perform this game, you end up with a sort of set of bushes, 3 small trees, and when you give a look to the real brain one, immediately you have a feature that emerge. The brain is very hierarchically organized, and in particular you see that there is a sort of chain-like structure. Clearly the color is the different macro regions of the brain, so there are only some regions that share main connection with different macro regions. This is the cerebellum, but what you see is that you have almost all chain-like structures, not branching. That was the first time we saw this. We get really struck because we didn't expect that the brain was so hierarchically organized. And indeed, when you randomize, the color here have no meaning, but when you randomize in any way, you get instead much more branched structures. Okay. This is the spanning tree. You can verify that. Okay, this is a really strong property by studying the histogram of the real network, the red one, and the other one, the null model, the blue one, where you have much more branched branched bushes due to this long tail. Okay, let me go to what we can do with LT. Can we apply this sort of machinery in order to distinguish LT from schizophrenic patients? So what we did is we performed the percolation over each individuals in the LT group and in the schizophrenic group. Then we took the average, the distribution of these sigmoids over the LT population, and the same we did over the schizophrenic population. So we got an average sigmoid for the LT, an average sigmoid for the schizophrenic. And then we put the 95% confidence interval around this curve, and they separated. So that was something also unexpected. And so it looks that in average, the mean, the brain, the functional brain of schizophrenic patient is more resistant to breaking, to rupture, to percolation than than the healthy one. One can think that this is due because they have stronger correlation, for example. But if you study the histogram, that's not true. They are more like one is a little bit broader than the other, but there is not a shift of the maximum that can explain this. And the explanation instead is different, is that the brain of a schizophrenic patient is typically more functional, from the functional point of view, more homogeneously distributed, more homogeneous than the healthy one. In the healthy one, there are more, can you say, compartmental structures, while in the schizophrenic, there are strong connections between these different clusters. That's why you, with a given, after following a stimulus, you can have different reactions in the schizophrenic, just instead of a single one. And this is given by, for example, by these two temperature plots in which we studied the variation of the degree distribution of the regions at increasing of the threshold. And we see that there is a stronger heterogeneity given by the lines in the healthy population with respect to the schizophrenic ones, and also the histogram of these, basically these curves is much broader for the healthy population than it's much more homogeneous. They are more similar in some sense. And this gives another. Let me just in the last minute, in order to go deeper in the study of this homogeneity among population, we wanted to study basically a topological exponent introduced in statistical physics in allometry and also in the study of repeated networks many years ago, that is able to distinguish more or less branched backbones of the correlation matrices for the population. Basically, in river networks, basically you can imagine that here you have one drop of water. Here there you have to be two because one has to go here and one has to stay here, and etc. And then you have, but at the end, you have to also to have, if in order to have 1 year, if you have a flux, you have to be a total cost of transfer that is 2 plus 1. You can do this game, and you can, for each node, and you can give to each node two quantities. One is the resource exchanged at node I, and that is the total cost. That's typically in allometry and in river networks. You can do this game. There is no physical meaning for the brain. It's just a way to detect how branched is the structure, because it is possible to show that typically there is a power law relation between these two quantities, and this exponent is between 1 and 2. And 1, you get 1 when your structure of n nodes is a star. And you get 2 when it's a chain-like. Okay? Then we did, okay, there are some examples. We did for, clearly you have, the first step is to transform the network in a tree. And clearly there are, for each node, for each region of interest, you can have a different tree. So we have an exponent over the LT population, different for each region taken as seed of the tree, but anyway, more or less, it's more or less of the same, apart from this region, there's more reduction. But what is important is that if you do this for the LT population, the schizophrenia, you find the same star. So it looks that there is no difference. Okay, these are the maximal spanning tree, basically. It's a sort of maximal spanning tree for the LT population and the schizophrenic one. There is no difference. But if you remove this from the completely connected network, correlation network, you remove these trees and you go to the next generation of these trees. You do again this work, okay, of finding the same tree and studying the exponent. You see that while the schizophrenic stay more or less at the same level of the exponent of 1.5, the LT population have a reduction of the exponent, meaning it's more star-like, more random. So what? And you can take the third generation, the fourth generation, the fifth generation. What happens? This means that you can see that these different generations have the most important pattern of connection, the second important, the third important, and in schizophrenia they look all the same, while in the healthy population, they look to become more and more random. So that's a study of the homogeneity. I will conclude here because I expired on the time and took also a couple of minutes. That was the community of collaborated to in this work. Let me just announce that from 2 to 7 July 2020, NetSci will be in Rome at the Sapienza University. So for those of you, and we are the organizers next time. So, thank you very much.
Thank you for the talk. So when you do the percolation kind of approach just to see the number of components, so this is done on the population matrix.
Okay. This is done over the average brain. Meaning that we take the Fisher transform, so we do in the proper way the average. We took all the correlation matrices for all the 40 individuals. We Fisher transform the matrix, then we take the arithmetic average, and then we Fisher transform, take the inverse transform to get the unbiased mean in this case. In this other case, instead, we took we performed the percolation for each individual brain, and then we studied the distribution of the curves among the population.
Okay, so can you say in one individual, just by the way it percolates, to what group will go?
No, there is an overlap. If you study, there is an overlap. There are basically, that's the situation of the two distributions. I think that would be too much, no? To have clinical.
And another very interesting.
But anyway, the group are not big, are 40. They are not 5,000. They are just 40 individuals. You can distinguish two populations of 40 individuals almost exactly.
I really like that. So another thing which is very important for the clinical world is that you should. So with this approach, can you kind of say what kind of circuits or brain areas are the more different in percolation, the component less? You have to say something about that.
We have not done for the population at the individual levels. Anyway, this gives you for at least for the healthy population in average.
No, I mean in the pathological group.
In the pathological, we have not yet done.
So
because we have to do individuals by individuals, with the stratification of different ages and the level of the schizophrenic.
If there is any component which is more different than the other, this would be very good.
Okay.
Yeah, I really enjoyed that. So I really like the idea of the forest, of the minimum spanning forest. But you didn't explore, or you couldn't explore that much. But so this was in functional connectivity.
That's the mean brain. That's the forest for the mean brain of the healthy population.
Have you tried to explore this in the structural connectivity matrix, in the connectome as well?
Yeah, we have done, but the problem is that we would like to relate the two. The problem is that it's not simple to relate correlation matrices to structural properties, mainly because of the small eigenvalues. When the structural can be seen as a sort of inverse of the correlation, at least in the Gaussian approximation, and the small eigenvalues create big instabilities in what you see. So usually they cover completely the signal if you want to derive connections from correlations. That's the big problem.
Yes, so
we are working to this point anyway.
So another point that you were mentioning the percolation, and I thought you were going to talk about the phase transition or percolation threshold, and yeah, you didn't go into that, but the minimum spanning tree kind of touches a little bit on that, except that you remove the loops. But can you explain what's
so it's not a simple cancellation of links. You have also the imposition, no loops and connectivity.
So what's the advantage of the minimum spanning tree with respect to the percolation threshold itself?
No, I say the spanning tree and the spanning forest have additional information, not only the connectivity, but also the lack of loops And we have just one connection for each node, basically. That's what you impose. So it's not simple to see from the percolation point of view where the spanning tree is.
I find your general vision of schizophrenia as a hyper-connected syndrome quite at odds with with the general image of schizophrenia, which doesn't necessarily mean that it's incompatible, but it's different.
Usually, structurally, there is a depletion.
Mm-hmm.
Of connectivity.
Yeah, it's often presented,
but that can create a more homogeneous patterns.
Mm-hmm.
Okay,
yeah. So it's interesting.
Functional patterns.
So it's interesting, and maybe seen at different scales.
Yeah, we are investigating now the relationship between the structural and the functional patterns, DTI versus fMRI, basically.
Yeah, on this topic, as I also today in the morning presented, did you look at all at, so to say, univariate properties of individual voxel activities? Or
not yet, because clearly there is a large variability.
It's amazing
and
that
it covers
that in MRI community
we are not allowed to go in the clinics, basically individual diagnosis.
But you have data. You have time
we
courses.
have data. These tools they permit to distinguish small communities. If I give you a small community of healthy and a small community of schizophrenic, and I don't tell you which are the schizophrenic, I can distinguish the two communities.
I just think that,
but not the single individual.
For tomorrow, maybe that's might be one of the topics for that. Usually the properties, the dynamics of the time courses are never studied in conjunction with spatial connectivity. But in neurophysiology, we know that the state of receiver and transmitter define the connectivity, and it's completely kind of ignored. To me, this is an amazing state of affairs currently.
Yes, definitely. Okay, I think it's, we'll thank Andrea.
Thanks a lot.
So our next speaker is Jesus Cortés, and he's from Basque in Bilbao, and he's going to talk about brain predictors of aging and lifespan, I suppose.
I will use this one.
Okay.
Okay, thank you everyone for being here. Thank you for inviting me, David and Javier Uldu. I really appreciate. So I'm going to speak about something we have been working in the last, let's say 3-4 years. It's a combination of two specific papers, but they follow the same line of research. So this is the people have been working in this talk. I'm going to speak today is Asier Ramuzbe, did the PhD with me. Now he's in Jerusalem. Paolo Bonifaci now became professor as well in Bilbao in the same lab. Myself, Beatriz Mateos is a radiologist. Ibai Diez did a PhD. Now he's in Harvard. Iñaki Escudero, radiologist, and Javier, who is a physicist. So, two classes of brain networks. Today, other speakers already introduced that. I'm going to focus on anatomical connectivity and functional connectivity. Although many people already said something, I really think still I can do a little further. So anatomical connectivity, you can see most of these are connectivity by eye. So it's these white matter tracks that communicate and connect different areas. So this is so what is the cellular substrate of this kind of tracks that I can see by eye in the millimeter scale? So it is supposed to be that there are like thousands of different axons that they can align together and make this kind of fibers or white matter tracks that people can reconstruct with the computer. So indeed what we can do is for each voxel in the brain, we can get a diffusion tensor, in which this is a symmetric tensor, in which we get three different eigenvalues, 3 different eigenvectors. So diffusion, when there is an axon, and when there is white matter tracks, the diffusion is highly anisotropic, there is a direction. When there is not, the diffusion is highly isotropic. So we can follow let's say the numbers in which this anisotropy is higher and make the continuation. So from these tensors, we can get different 2D images or slices. So some of the eigenvalues there, some of these different images are very important to study different kind of properties in the pathological brain. So we know that fraction anisotropy is a good measure of fiber integrity, so the higher the better, but there are others that are typically used in clinical routine. So let's say the algorithm that get from one seed the different voxels that are aligned together with a tract is called tractography. And we get these kind of figures. That means that what we can color is the different fiber groups in different colors. So projection in blue, we get association in green, or commissural crossing corpus callosum in red. So we have been working on this for a while. So when we speak about anatomical connectivity, we can get a matrix, as we have been seeing this morning, related to the number of fibers that are connecting to areas, but also other kind of properties of this kind of fiber connectivity. We can get the volume they occupy, the density, the length, the anisotropy, so the healthier the better, and on the higher the value, mean diffusivity, and any others. So functional connectivity is something which is nothing related to the other, in terms that the images from which you calculate these connectivity matrices as compared to the anatomical, the images are different, but also the processing you use in order to get these matrices. So what we can calculate is the blood oxygen level dependent. So it means that we can see when the deoxygenated hemoglobin, so in areas where the oxygen has been consumed, we get a high signal. So two areas of the brain, although they don't have any anatomical connectivity between them, we can get that when looking to this ball signal, they are highly similar. So we say that if this is so similar, they might participate in the same network. That means that they are functionally connected. So it's symmetric in general. It's also a connectivity which is highly dynamical. I like the anatomical connectivity, but also very important, some areas here, they can be in the same network, participating in the same network with anti-correlation with the other two. So the three classes of clusters are participating in the same network, and there are different measures to get that. So what we typically do in the lab is getting what we call triple acquisitions. It's just by default, we always take the anatomical or structural imaging, the functional imaging, and the diffusion tensor imaging. In such a way, we can get the two classes of networks. So this also have different preprocessing. People here didn't pay too much attention on this in this workshop today, but typically in order to get functional connectivity matrices, you also need these anatomical ones for co-registration, segmentation of gray-white matter and cerebrospinal fluid, and also you get some brain extraction, smooth intensity normalization, regressing out, bandpass filter, diffusion tensor. You have a very different process, and finally you can come out with two connectivity matrices of the same person that they must be related in somehow, and this is what I'm going to speak today. So first paper we published is this one, and I'm going to tell you what we did. So the result of the paper, you can use also this atlas, and you can download. If you have any questions, further question, I will be happy to tell you. So we have one person, we have this class of networks, and the idea is how the two can be related, no? So this was already underlined by Carl Friedson in Science, where he said that this is a very complicated problem in system neuroscience, and the main reason is that we have many functional states consistent with a single connectome or single anatomical graph, and this makes this relation problem very hard. So this is what we get. This is one subject. This is the anatomical connectivity between 2,500 regions in the brain. They occupy the cerebral cortex, also subcortical structures. And this is the functional connectivity matrix of the same person of the same regions. And we want to address is how much this matrix is related to this. So we didn't work at this level. This is not a voxel level. This is a, let's say, super voxel level. So it's about 90 or 60 in average voxels per region. So we work at the module level. So what we did was, I can tell you the details, but basically we define per region what is the connectivity pattern to the rest. And we did this for all the regions and then built a distance matrix over which we can calculate hierarchical clustering. So after reordering this matrix with the labels of this module, we can get this kind of a structural, more structure in modules matrix of this previous person. So then, by using this order, we can go back to the original structural matrix and order again. So here, if we do a zoom, we can see how just by applying modularity in the functional and reordering the anatomical, we can see that the two matrices that in the beginning they were not very similar to each other, at the aggregated level, at the module level, we can see highly similarity between the two. Okay, then we need to quantify this in terms of different metrics, and this is what we did. But this is the main observation, is that those two matrices can be very similar, but you have to go to some aggregated level. It's not at the voxel level. So this is another small thing. So these connectivity matrices, although in the neuroimaging community people don't like too much, there are negative correlations. And this has been a huge discussion, what this or negative correlation come from, and is it preprocessing artifacts or not. What is clear, we published a small paper in which we show that classical Ising models with conserved dynamics, what is called Kawasaki dynamics, when you apply constraint conservation in the dynamics, you can get this kind of negative correlation as a result of this kind of homeostasis activity. So this is just to summarize. We can get the functional connectivity, applying hierarchical clustering there, reorder the structural one, and when we zoom, we can see that some modules in the function appears in the structure very clearly by eye. So the big question now is, okay, this is hierarchical clustering. You go from the original matrix, from one module, which is the entire brain, you go dividing by 2 in each step until the node level. So at what level of the tree I should stop in order to get the maximum similarity between the modules in the function and the modules in the structure. And this is what we solve in this paper. So we got that the maximum similarity between structural modules and functional modules are achieved at the level of 20 modules. Why is 20? Is that a driven problem? We don't know, but this is the maximum, and I will tell you based on what metrics. So after that, that purely data-driven, we can see what modules are those, meaning that these modules are modules in the functional matrix, but also modules in the structural matrix. So these areas are functionally homogeneous, similar dynamics, but also structurally integrated, meaning that they have intra-module connectivity higher than to the rest of the modules. So we can see here, for instance, module 9 and 10, this is the classical deformal network that we know that incorporates the cerebellum, but we separate, the deformal network is separated into the frontal part to the rest. Also we can get classical sensory motor auditory areas, subcortical ones, and others. So this is the best partition we can do in order to get modules that are functionally important and structurally important. So optimal in what sense? What is the metrics? So then we created what we call cross-modularity. Cross-modularity is just, I can come here and say this is modularity as a function of the level of the tree, and then this is modularity in the function, reordering the function, modularity in the structure, reordering the function, different classes, and just to see the level of modularity that the different partitions have when we go down in the tree. But also the similarity that this module has with this one, based what is called Sorensen index, is intersection between the two, the mean similarity between all the modules across the tree. So we want a partition that is modular at the function level, a partition which is modular at the structure level, and also that the modules in one representation are similar to the other. Then we multiply the three, and we get the cross-modularity, and we try to stop in the hierarchical tree at the level in which this correspondence is maximum. So visually, I can just see, so these are the different modules. I can plot the intra-module connectivity in the two representations, and it is high, but also they match each other. And this happens module by module. So taking this, let's say, partition, I can guarantee that dynamics is similar, and also there is an intra-connectivity higher in that module to the rest. So then we identify the different modules. Some have a very well represented function, or some represented anatomy. So this we have been working in different data sets, and we are getting similar results, even at the individual level. So in the paper, we also did a complete anatomical description for each of the modules. And we can, you can see there. If you have any question, you also can ask, and I will tell you after. So this is the optimal brain partition. As you can see, we can the 20 modules, just data-driven. We see that most of them are bilateral, so they are well represented in the left and the right hemisphere, the symmetrical part. So some regions are highly compact. This is just a medial visual is an area. Other modules are more a class of a network, and what we can guarantee with this method is that each individual region or module is structurally wide and functionally similar. Okay? So now we are going to use this formalism to kind of work in network predictors of aging. And I'm going to tell you what we did here. So this was published last year. So just very quick data on lifespan. If you know this, if you don't know, I can tell you more in more detail after. So the total volume increases 25% from childhood to adolescence. The run keeps constant for about 3 decades and later decays down to childhood size. So it's something like U-inverted curve in the volume. The gray matter volume decreases less than white matter. There are regionality patterns in the two. And also some areas, as hippocampus, prefrontal, or parietal, are the ones that they get faster deterioration or atrophy with age. And then the ventricular volume increases, and this is indeed a very good radiological indicator of aging and other classes of dementia. So this is a paper by Messer Lab in Jerusalem. So when you look to different kind of white matter tracks, how this changed from a lifespan 8 years to 80 years, most of the anatomical growing patterns in the brain follow what is called U-inverted patterns. So we have less integrity in the tracks, a maturation level, and then a deterioration again. This happens in anatomical connectivity, also happens in the function, also happens in cognitive tasks. Most of the tasks people perform better when they are like 20 until 50s, and then they start to decrease. So this is a really general pattern of what is called healthy aging in the brain. So what we wanted to do here was the following. So can we use brain connectivity to measure brain maturation? So can we understand the age we have by looking how our brains are connected? If this is true, there are two concepts, different concepts of age. The age that our connectivity pattern has, and the age that we really have in our passport. This is important because if they are different, what is bigger? If you have 60 years old, but your brain is 50, this is good for you. But if you have 60 and your brain is 80, this is bad for you. And also the contrast, the contrary pattern is when you are a kid. If you have 8 but your brain is 20 or 12 or 13, this is good for the neurodevelopmental of this kid, but also the opposite. Second motivation is the following. Can we alter the brain connectome age by a treatment or therapy? So, for instance, by increasing the level of physical activity using a drug or rehabilitation program. And the last question, which is very important, if this is true, if I can predict the age from the connectomics, what is the circuit that predicts the age the most? This is an important circuit when we age. Okay, this is the three questions that we solve in the same problem. So, can we use brain connectivity to measure brain maturation? This is kind of brain training for many here. I know you played when you were younger. So after some time playing, based on reaction time, the machine was telling you are 79, and you were pissed off. And then you try to play again, and you were reducing this reaction time, and you were like younger, no? So indeed, the first person doing this was Rutan, which introduced in the 70s what is called the brain age quotient as a measure of age-related cognitive functioning. So based on how you function at the cognitive level, I can get your age. But honestly, this has not been used too much, although Rutan is one of the fathers of the modern clinical neuropsychology. So brain age based on morphological features of the brain, volume, gray matter volume, white matter volume, ventricle volume, and many others has been studied. But our approach is just strictly based on the connectivity. So this is important to tell you. This is just, imagine, this is the ventricle of a person of 17 years old, this black thing here. This is the average in our population of 155 subjects, and this is the ventricle volume of a person with 72. So just the volume of the ventricle is a good proxy of the age. But we didn't do like this. What we did was to exclude all the regions belonging within the highest ventricle, just to see the connectivity of those regions which are not affected by that. So I'm going to go to the methods. So first thing we did was to use the brain hierarchical atlas that I explained you before. Okay? And then, rather than using the 20-module representation, meaning that at this level, structural and functional modules are the most similar as possible, we work in the multi-scale approach. So it doesn't matter the level, we consider all of them. And for each module, at each level, we calculated 4 classes of descriptors in the machine learning terminology, which means in the functional matrix, the internal connectivity that this module has. In the functional matrix, the external connectivity, that meaning that the sum over all the connectivity that that module has to the rest of the brain. This is another feature. And then this is in the structural, and in the functional we did the same. So eventually we have, for each module, 4 classes of values that represent the connectivity. And then we did this for all the modules at all the levels in the hierarchical tree. This is the possible features or descriptors. Then we did the following. This is the population we have from 10 to 80 years old, 155. And then we did for each. So this is the number of possible descriptor or feature I have for the regression problem, which is to predict the age that the subject has when he's in the scanner. Okay? So we have 4 classes: functional internal, functional external, structural internal, structural external, times the number of different modules per each level in the tree. These are the possible. Then we did something that we did like feature selection, but we did was something like we did. We want just to use those modules such that the correlation of the functional and the structural, the multiplication of the two, is significantly correlated with age. So we don't want modules structurally correlated with age, functional correlated with age, independent of each other. We want modules that they are correlated with age functionally and structurally. This is important in terms of performance, it's in the paper, but also because we wanted to address a structure function circuit of this aging. And then also you have to do Monferroni correction because you have many levels, many comparisons. So this is just divided by the number of comparison we have. For instance, starting 20 modules to 1,000, this would be the correction that we have to do. Then this is the method we did for the prediction. So basically what we did was a regression problem. So this is the age, different subjects. Here we have the values of the different connectivity classes 4 times the number of modules less because we wanted the structure and functionally correlated. Then we estimate based on a test population, we estimate the error, which is just how good your model is capable to regress the variable, the continuous variable we wanted, which is the age. This is the maximum likelihood estimator. It's a simple way of doing this. So finally, we were getting as a metrics for the performance what is called mean absolute error. This is chronological age, connectome age, and we did cross validation in different ways. It's in the paper. If you have questions, I can tell you after. But basically, you use for training the model, you use some data and testing, calculating the metrics in the other. So it's very important this. So the model depends on how many features I want to put here in the sum. So we did some optimization method. Is in the how many time I have?
Just a couple of minutes.
Okay, so you have question, but basically we were doing some kind of nested correlation. We were capable to get the optimal number of features to be considered in the model. So these are the results. So we were getting in relation to external connectivity. So in blue, I'm representing the area such that the structural anatomical connectivity reduces with age and functionally increases with age. So somehow network compensation between the two. In green, I'm plotting the region such that the external connectivity, anatomical and functional, both decreases with age. So as you can see, with regard to the external connectivity, both classes of phenomena occur. Internal connectivity, we don't get any area increase in function to compensate the structure, but also we get the two decreasing with age. So this is the performance, this is machine learning, it's in the paper, 3 years of work, but this is test data, this is chronological age, this is the brain connectome age. So we can get correlation 0.9 and mean absolute error of 5 years. So without knowing the age of the person in the scanner, we can, from a population that goes from 10 to 80, we can know the age that the participant has with an error of 5 years. Okay? This is very important because motivation number 3 was what is the circuit that performs such well. And the circuit is the frontostriatothalamic circuit. This is an important circuit. I will tell you after what it does, but it's thalamus, basal ganglia, and frontal and orbitofrontal areas. So this is important because in the aging literature, people are having focus a lot in the hippocampus circuit, because in the pathological aging, in most of the dementias, the episodic memory and classes of memory are heavily altered during age. So in our method, the hippocampus connectivity also plays a role in the estimation, and the connectivity decreases with age, but our circuit, the decrease of connectivity due to age is higher than the hippocampus circuit. Okay, so the brain connectome age is multi-scale structure function estimation of the chronological age, can work as good as other brain age estimators using morphological descriptors. Our approach reveals that the circuit participating the most in age prediction is the frontostriatothalamic circuit. We suggest that when studying healthy aging, frontostriatothalamic circuit is the major circuit dominating the process, but when studying pathological aging, the hippocampus circuit is the gold standard in both human and animal studies. Frontostriatothalamic circuit mediates motor skills, cognitive control, executive function, also regulates dopamine that control reward, motivation circuits, serotonin, mood and emotion release. And this is important. The discrepancy between chronological age and connectome age might work as a biomarker for quantifying deterioration as a result of disease or improvement after some treatment or therapy. So this is something we are working now with Stefan Swinen in Leuven. I will be sitting on Thursday to him. So we also have some scales that tell the level of physical activity that these people are doing. So now we are calculating for those people that have very high physical activity, what is the difference between this is the older brain, so chronological connectome age older than chronological. So we get that these people have younger brains as compared to people with low physical activity. So the difference can be 4 years, 2-3 years younger. So this is an indication that we could use this physical activity in order to some rehabilitation program in age population or something like this. So finally, thanks to all the people that work in this and funding, and all of you for your attention. Thank you.
So, any questions?
In the modularity study of the functional network, which kind of modularity function did you use?
In that result paper is standard Newman modularity.
The standard modularity.
Yes. So I was aware on this Garlas-Kelly paper later, and now we are also using this for other studies, but not for this one.
Thank you. Thank you, Jesus, for this very interesting talk. My question is very naive. What is your opinion about, I'm trying to imagine which are the mechanisms that produce this inverted U curve? I mean, for the different regions that you, modules that you show it.
This in aging is very typical. So, for example, when you are a kid, you have some procedural learning. You need some time to do this. Then you get optimal. You learn, and then there is physiological aging cascade that make you to perform worse. This behaviorally, I think, is clear. Anatomically, I think that behaviorally we behave like this because our brain is also doing that. So the brain is growing when you are a kid a lot, but then in adolescence start to stop. There is also connectivity. There is all this growing. There is planning. So I think this kind of developmental U inverted patterns have been observed from cellular level to behaviorally a lot, I think.
Very quickly.
Thank you for your presentation. I have a question for the brain age, chronological age versus connectome age. So at a certain point you say that your results are as good as the one you get with morphological features. So now it's true that you say that for the connectome analysis you excluded the regions close to the morphological features that are mostly affected by the atrophy, let's say. But in general, I'm assuming...
We were capable to exclude at least the ventricular bias.
Sure. But I'm guessing that in general you have atrophy all over the brain with age. So I was wondering to what extent morphological results are correlated with the one with the connectome one. And in general, I was wondering if you can combine the two to be more precise in the prediction.
So this is something that indeed we did. So we were combining not only morphological descriptors, but also psychological outcome in different tests. So surprisingly, you get like a bound in which you cannot reduce more than these numbers. So what this means is that maybe morphologically you can get the same number, and the function, neurological function, is related to the connectivity somehow. We don't know the connection, but you don't do better because you add a structural kind of constraints, neither neuropsychological outcome. So just to tell you, many other papers that people have been showing, for example, there was a release in UK Biobank for aging dataset. So people were doing 2-3 years mean absolute error, but that was released from 40 to 80. It's not important only the error, but also the range of the age in which you calculate. This is 5 years in a range of 70 years. So other people do, but in a range of 30. So eventually, is this ratio the one that we perform as good as other models?
Okay, we'll discuss that. Anyway, thank you for your talk. Doesn't seem to be there. Oh yeah, here he is. It's a very brave task because he's the last one, I suppose. So let's move away from your computer, which I think. So Ignacio Echegoyen is at the Polytechnic University of Madrid. He's a student. Fourth organizer of this event, and he'll talk about some multi-layer related stuff in Alzheimer's disease. Is that correct?
Yeah.
Okay.
Yes, it's a bit. Yes, yeah.
Okay.
Okay. Yeah, hi everyone. Thanks for having me here, giving this talk, given that I'm in my third year of PhD. I'm going to talk about a project I'm currently working on with Javier Martin Buldu, my supervisor. We are trying to understand Alzheimer's disease from a network neuroscience perspective, and more concretely, we're trying to apply a fully multi-layered network approach to try to get new insights on the disease. This is an ongoing project, so I'm going to show just preliminary results. As I guess all of you know, Alzheimer's disease is one of our age diseases, and many efforts and money are now on trying to understand how we can stop the disease and understand the delaying principles that drive it. It comes with basically memory loss and spatiotemporal disorientation, but also with some other symptoms as anxiety, aggression, mood, and personality changes, which makes the disease not only difficult for the people that suffer it, but also for the people that are taking care of them. So nowadays it's a huge problem. But also from a psychological perspective or a psychiatric perspective, things are more or less clear, and if you have a good expert on the topic, he's always going to give a good diagnosis, and things are clear more or less. When we talk about time series or networks, things are not that clear. And first, I'm going to outline some results, mainly from the start when Stam and colleagues started to do research on this topic around the 2000. There has been many efforts done in trying to understand the time series from a time series analysis physics perspective. There are some papers on entropy and complexity made mainly on sensors. Here I'm going to focus on MEG and AEG data because I work in that modality. So the results refer to those modalities. And the point I want to make here is that results are somewhat contradictory and counterintuitive. In this case, talking about broadband signals, if you study complexity, you can get different results depending on the measure you're using. Also, it will change if you're using sensors or ROIs. I'm going to talk about it later. And something similar happens if you go to study correlation between the time series that you use to build the network, the edges of the network. I'm not going to talk a lot about it because it's been said before. But again, it depends on the measure you're using. You will get different results. If you use coherence, you will get different results in the different frequency bands in which you can divide the signals from the brain. Also, as you can see here, Stam is probably the guy that have studied this the most, and he get different results depending on the measure he's using, which is quite worrying if you really want to understand what's happening here. And I'm not going to do an exhaustive exposition on this. The key point here is that people are usually focused on one synchronization measure and usually in one or two bands. And this is just a small part of the whole picture, and it's not useful to understand what's going on and to develop new biomarkers to understand how the disease evolves. So I'm going to present some results we have on MEG signals. We'll try not to make the same as everyone. And basically we have resting state, eyes closed data for 4 seconds at a frequency sampling of 1000 hertz. And we also have 30 samples or trials or repetitions for each subject. The data guys did some stuff on the time series, anti-aliasing filters. At the end, we have a signal between 2 and 45 hertz, which is in sensors at the scalp level. I think Leonardo talked a little bit about this, the MEG data is basically information, magnetic information at the scalp level from cortical neurons, and is more or less common, although Honorva doesn't like it, it's more or less common to get the source reconstruction and get information at the ROIs level, which is kind of problematic as we saw before. But again, here as the data guys in our lab are used to do this, I'm going to work also at this level. Some information about it. They have a forward model of 1 centimeter spacing, which is just a grid in which they project the information from the source, from the sensors to more than 2,000 sources, and then they get the average for the centroid, which yields 62 regions from the Harvard-Oxford Atlas. So at the end, we have more than 100 subjects. 48 of them are in the control group. 46 suffer mild cognitive impairment, which is a prodromal stage of Alzheimer's disease. It has a rate of 10% per year of conversion to Alzheimer's disease, and 17 people with Alzheimer's disease. As you can see here, there's a sampling problem. I will try to fix it statistically. I will talk about it briefly. And we have information at the sensors level, 102, because because of the machine we have there, and 62 estimated ROIs regions. And with every time signal from both modalities, we do a frequency decomposition analysis to get the classical delta, theta, alpha, beta, and gamma bands, and to ensure that we are just keeping the edges that are significant, we do a permutation testing. In this case, we are getting the correlation with mutual information. I'm not going to talk much about it, but basically what we try to get here is the network with homogeneous links, meaning that we want the same measure for intralayer links and for interlayer links, which has been a problem in clinical neuroscience because typically you use one measure to get interlayer links between layers, and a different measure to get the links inside the layers. In this case, we're using just one measure, mutual information, which has the advantage of having the same units. It's everything in bits. So we can use it to measure interlayer and intralayer links. Because we're building a full multi-layer network in which each layer is a band previously decomposed from the signal. Before talking about the results, first of all, we will conduct some analysis on the time series. We also conduct some analysis on each individual band, typical network parameters, and then we will try to get some information about the whole full multi-layer network, which is the final goal of this project. First of all, I will talk briefly about these statistical methods. We do a surrogate method. In this case, we are applying some sort of block permutation testing. We basically get the correlation between two time series, which is going to be the edge. We then cut some part of the time series of one of them and put it at the end of the time series and get the correlation. The length of this block in which we're cutting the time series corresponds to the cycle of the autocorrelation function. So that way we ensure we are not randomizing too much things. And basically, we do this for 200 times, 200 iterations, so we get a distribution of values of, in this case, mutual information of the correlation under the null hypothesis of no temporal correlation, because what we're trying is we're trying to break the temporal correlation, keeping everything more or less the same. And then we use this null model, this distribution, to make a hypothesis test and test that way every edge. So we keep only the edges that are 2 standard deviations above these null model distributions. Okay, about the data, the groups, as I was saying before, we have a sampling problem because we just have around 20 subjects with AD and more than double with mild cognitive impairment and control group. So what we do to get these results, here I basically plot the results of a Kruskal-Wallis test, which is a non-parametrical ANOVA test for 3 groups. And what we do is basically, as we can get a value in this case of complexity per subject per sample, we have basically a vector of values, and we do a pooling. We pull some values from each vector in each group. That way we can match groups. We do this Kruskal-Wallis test, and we repeat the same test 1,000 times. So in this case, if you see stars above the bars, is because we find here statistical a significant difference more than 800 times. We did this. We take from the whole data just a group, we do the test, and we take again data to do the test again. And as you can see here, results are not the same if you do the procedure on sensors or on ROIS. Fortunately, the direction of the changes are more or less the same, but as you can see in sensors, we have much more differences than in ROIS. In this case, for complexity, we expected different results. Indeed, we expected the opposite result, which is that in Alzheimer's disease, complexity decreases, which is what we're used to see in literature. Basically, in alpha band, we would expect that it goes down, which is exactly the opposite of what we're getting here. And as you can see, the pattern of alterations is not the same at every band. So if you focus just on one band, you're not getting the whole picture, which is that if you believe this, you should believe that the differences are not the same at each band. Regarding sensors and ROIS and the difference between them, if you are a detractor of ROIS, you would say that you're losing some information when you do this process. But if you're in favor of ROIS, you would say that sensors are basically noise, and what you're getting is spurious correlations, and then you're getting just the real correlations. I'm not going to interpret this because it's not clear what does it mean. But if you were to classify subjects as in one group or another, it's better to use sensors instead of ROIS because you're getting much more differences. At least things are paired between complexity and entropy. That's more or less good. And we can do some basic network parameters on each individual layer from each subject. And again, we're finding results that are not in agreement with the literature. In the case of strength, for example, we would expect that it goes up in mild cognitive impairment. It's been proposed as some sort of mechanism trying to go against some sort of recovery mechanism. It's been observed that strength goes up in mild cognitive impairment, which something that will recover for alpha band, but then Alzheimer's disease goes up again, which is against the literature. But in this case, again, sensors and ROIS show really, really different results. So you cannot take just one part, just one set sensors or ROIS and one band, because you are not going to get the whole picture, which is something really hard to understand and to interpret. And as we're finding this and we are not sure how to interpret and what to think about it, we would like to have just one parameter that is able to capture the difference or is able to distinguish between groups, just to simplify things, because this is hard to interpret, and this is when the multi-layer approach might be useful. And I'm going to talk about really preliminary results and on the multi-layer network. Here, in this case, we're working with the second smaller eigenvalue of the Laplacian matrix. For those that doesn't know, the Laplacian matrix is basically a matrix with the degree of each node in the diagonal minus the adjacency metric. So it has information about the modules inside the network, but also about the weights, because in this case the network is weighted. Theoretically, well, it depends on the number of interlay links, on the number of layers, and also on the differences between the weights in the difference in the interlayer links. Theoretically, what we would like to have is some sort of transition, some sort of abrupt transition, as you can see there. Basically, the second eigenvalue, the algebraic connectivity, is talking about the different groups inside the network. If you take one network without just in one band, this eigenvalue is going to talk about the number of communities inside that network, and indeed there are methods trying to find communities based on this value. In the case of the multilayer, it's speaking about the integration of the bands. It's speaking about the integration of the layers. And what we would like to find here is a difference in this parameter, which would be interpretable in terms of integration between these layers. Theoretically, as I put here in a paper from Radici and Arenas, if you increase the value of the interlayer links, which is this p value here in the x-axis, if you increase it, what you get is a change in this eigenvalue, in this lambda 2, until it reaches an abrupt, it reaches an asymptotic value, which is exactly the mean between the eigenvalues of the network taken as a whole, the aggregated network, which basically is telling us that at that point you cannot talk about two different systems, but a system that is not distinguishable. The bands are not distinguishable. It's completely integrated. And what we are trying to explore now is, first of all, there are differences in this second eigenvalue, which could tell us information about the integration between the layers. And also, if we do a simulation on of these interlayer links, basically taking the networks we have and increasing the value, could we see differences in this critical point, in this abrupt transition point? Well, first of all, it seems that we can. At first, here I just plot for the not for the full multilayer with all bands, but just with alpha and beta, theta and gamma, and then for the full multilayer, not all possible combinations, but we're thinking of trying all possible combinations to see which one carries more information. At first, the conclusion is that we can see groups. In the case of the alpha and beta band, we can see a clear fashion in which Alzheimer's disease show a smaller value of this lambda 2, which is telling that layers are less integrated. We could say are more independent functionally. Something that doesn't hold for in ROIs or in the other network just with theta and gamma. Something similar we can say with the full multilayer, although in this case the mild cognitive impairment group has a little bit higher value, which we don't fully understand. We have to look at this more carefully. And we would like to use this as a biomarker to see how it changes with age. In this case, we need longitudinal data so we can understand better how these parameters change. And just to conclude, this is the results. These are the results for the simulation. It may seem from the plot that there are differences, but running this test, as I was saying before, 1,000 times doesn't give results. Although it may look from the plot, statistics tell us that it's not different. So this is quite disappointing. We don't know yet what's going wrong, and maybe it's because it doesn't have any clinical meaning, and it's not useful to do a biomarker, but we have to do more research on this. Basically, to conclude, results are somewhat contradictory, counterintuitive, depends strongly on the methods we're using. If we are estimating information in ROIs, the measure we're using to get complexity and entropy, the measures we're using to build the networks. And from my viewpoint, we should get as many parameters as possible to see the big picture, the picture as broad as possible, and try to keep those that are more informative. And apparently the multi-layer network approach could give one useful parameter at least, but we still have to work more in this. Thank you.
There's no question. Oh, maybe. Well, I'll go with mine and then I'll. So why do you think there's so much inconsistency in results?
It might be because of the. I think it's mainly because of the methods. We've seen here today that it depends on the methods you're using. You're overestimating parameters, underestimating parameters. I think that as we don't really know what's going on dynamically in the brain, every result that is able to distinguish between groups is good enough. So it's published, and you get more and more and more papers saying, oh, we find a difference here. And the thing is that we don't really know not only what we're doing, but also what is going on. So.
When you say consistency, why you said inconsistency?
No, well, because there are many studies saying basically the opposite from the prior one.
What is it exactly? Because we publish in Alzheimer's disease information higher than control.
Sorry, sir?
So we publish also that information, total amount of information, Alzheimer's disease versus control was higher in the pathological group. There are other people saying this as well. And so this is why was my question. In what sense?
Yeah, for example, complexity. The trick here is that complexity is calculated with a different measure in each paper, but it is supposed to measure complexity. So either we're not measuring complexity or.
Yeah. So I have another question. So when you do from sensor to region of interest, so there is one slide which is something very similar we found, which is that you find more differences between the groups in the sensor space.
Yeah.
So for us was the same, but then working in the ROI level, rather than going for the average signal, we were calculating PCA, further components, and we were getting very nice differences in second and third component, but not in the average. So if you look in the ROI, rather than taking the average, which is the first component, you go to the second component, and we were finding a lot of differences at the ROI level, but second component and third component.
Similar to the sensor, so it was more consistent with the sensor.
Yeah, it's higher than the average, but it's you know, it's a lower scale than the sensor. So it's something in between. You can let's say correspond the two results by going to the more than first component in the ROI.
Okay.
Comparing this.
Yeah, that would be really interesting to see the steps and see how it changes with each step. Okay, yeah, we should talk about it later.
Thank you for the presentation. Counterintuitively, there could be some possible explanation for your MCI cases, because there are increases in connectivity between thalamus and medial temporal lobe in MCI subjects differently than Alzheimer patients. So that's one explanation. And also there are some papers saying the increase in structural volume in MCI due to some edema in white matter. So there could be several explanations for your findings.
The thing is that what we expected was finding this raising in mild cognitive impairment, but then going down in Alzheimer's disease. I mean, these people doesn't know where is their home. They have like holes in the brain. I can't believe that they have higher values of complexity if we're really measuring complexity. It's hard to understand. I mean, I don't know what's going on, but they're in a very final stage.
But complexity is redundant. It's bad complexity. So, you know,
then
pathological brain can be highly complex in your measure, but doesn't mean anything because they're kind of communicating each other like a mess, but nothing is happening.
Then it would be entropy. I would expect that entropy. The measure I'm using here with complexity is not exactly entropy.
What is it? Sorry.
It's the statistical complexity that makes entropy with divergence, the Kullback-Leibler divergence.
When you were showing that the matrix, the connectivity matrix, you had many concatenated, basically. Yeah, I wanted to ask you: is this cross-frequency coupling? Let's say when you have beta alpha, is this cross-frequency coupling?
No, it's probabilistic because it's based on mutual information.
Yeah, but you directly calculate the coupling between two bands. Yes.
But yes. Just with the time series.
Yes, using bandpass filter data, not amplitude envelopes, because in amplitude envelopes you demodulate signal and then you're okay. So here you don't demodulate signal with Hilbert wavelet. You directly have bandpass filter data, and then you have these mutual entropy calculations. Okay, but then I'm wondering, you have different frequency content. How would it work actually? Because for some, if it's integer-related NM coupling, you can calculate cross-frequency, or for amplitude envelopes, arbitrary interactions.
In this case, we're just using mutual information, which doesn't have like a real interpretation, apart from the fact that things are happening more likely than you would expect by chance.
Okay.
Yeah.
Okay. Any other question?
So, thanks for all the speakers. So basically all of you, almost. For discussions tomorrow. And also now we have time, if there's still some question somebody wants to ask. Or if you already have something in mind you definitely want to discuss tomorrow, you could also bring up like 5 minutes, 10 minutes of open word. I'll pass the mic to whoever wants to say something.
Firstly, very interesting talk. And okay, you compare very well the two kind of ages, but what's about, did you use, because perhaps I didn't follow well, did you use specific tasks in order to evaluate the output in terms of cognitive, emotional, or action? Because this is the real parameter, in my opinion, of aging evolution.
So this data is we didn't use, but this is what most of the cognitive neuroscientists do. And Stefan Swinen in Leuven has a lot of this data on many motor learning tasks, and cognitively and other things. So this is a different approach, and I agree this is behavioral is very important in the task. So we could extend this kind of approaches by using also this kind of functioning. But this has been studied a lot in attentional task and cognitive control. No. Yeah, no, yeah. This is something that could be extended very, very nicely, but we didn't do yet. Yeah, thank you.
I had a question for Giovanni. So I was very surprised by one of your results, actually, which is the inexistent relation between Ricci curvature and topology. Given the number of relations between curvature, not necessarily Ricci curvature, but Gaussian curvature, and let's say global aspects of topology such as Euler numbers and so on, the Gauss-Bonnet theorem, but and so many other generalizations. So I was wondering.
Do you hear me? No. Okay. That actually works when you have. So let me start somewhere else. Usually, people say that topology is global, right? And that's actually true when you're considering full-on classification of surfaces. For example, if you take the classifications of surfaces in 2D, then the genus is sufficient. And at that point, the curvature is related to some of those things. In this case, actually what you have is that there is something that is multi-scale and highly non-trivial in terms of topology. So the curvature that you measure that way is actually extremely local, and it can change from point to point because it's really.
Yeah, but this is always the case.
Well, but the relationships usually don't hold when you have a generic space of that type. Right? They hold when you have very specific spaces. Well. And that's the thing. That's the only explanation I can find for that one. At the same time, you have some global. For example, if you think about Euler characteristics as a sum of the Betti numbers, but actually what happens there is that you have a different Betti number per step through the data filtration. So you're aggregating the whole information while the curvature that you get basically literally from the relation values, right? It's one step on. So that's another possibility why that actually is not picking up the same type of.
No, it's interesting. I don't deny. The result is.
No, yeah.
So.
No, but we too we're expecting to find a little bit there, and we expected them to be sort of correlated. To be honest, for me it's harder to understand. Yes, you can calculate it. You can make argument about flows used by using the curvature. For example, as in the papers they introduced this, but really what they do there